मुझे फिर चेक करो। बाहर को आ गया। अरे
I hope the entire session will be enjoyed by you guys. Program will be starting shortly. All dignitaries and guests have reached. We will be entering soon the auditorium. As they enter the auditorium, keep continuing clapping, please, until they take their respective seats.
तो सर अब मतलब टाइट शीट नहीं है रिलैक्स वगैरह। टेबू लगा लो। अभिषेक पसूरी में ऑन करना है वो।
A very warm, a very warm and happy good morning to you, one and all. Until here, I, Dr. Prem, standing for you to welcome you all on behalf of the Institute of Pharmacy, University on today's International Conference, New Paradigm in Pharmaceutical Research and Development 2023.
All delegates and participants, our esteemed guests have arrived. They are outside auditorium. They will be soon arriving amongst us. So as they enter into the auditorium, please stand up for their honor. As they enter, just not now, as they enter into the auditorium, please be stand up at your places. I request all participants, please be seated. Once again, I request all delegates and participants, please have your respective seats at the earliest. I request all participants and delegates, please keep your mobile in either switch off mode or in flight modes. Either you may also keep your mobile in silent mode. I request all delegates and participants, please keep your mobile in silent mode or in switched off mode. Ladies and gentlemen, once again, I, Dr. Prem, feel privileged to welcome you all on behalf of Institute of Pharmacy, Bundelkhand University, Jhansi. for today's international conference, New Paradigms in Pharmaceutical Research and Development. Hello. I request to all delegates and participants, please have your respective seats. Our dignitaries and guests will be soon arriving among us. Please don't rehearse right now. Don't rehearse. Good morning, ladies and gentlemen. Once again, I welcome you all participants and delegates who are here. All dignitaries have reached into the auditorium. They are just entering into the auditorium. So as they entered, please keep continue your clapping. As they entered, just for a while. Please try, don't move here and there during the session. During the inaugural program, please don't move here and there in between. वही रहा क्या ना? Chief guest dignitaries just going to enter. Welcome all dignitaries. Welcome Professor Pia Vogel ma'am, who is here. I cordially welcome on behalf of Institute of Pharmacy, Bundelkhand University, Jhansi. Welcome ma'am. Welcome. Welcome ma'am. I welcome you all on behalf of Institute of Pharmacy, Bundelkhand University, Jhansi. Ma'am. I request to Professor Pia Vogel ma'am to please directly come on stage and have a seat. We have amongst us Honorable Member of Parliament, Shri Anurag Sharma ji. We are pleased to have among us Shri Anurag Sharma ji. He is an Honorable Member of Parliament amongst us. So big hand to him, please. Keep continue clapping. Sir, please take your distinguished seat, sir. I welcome you all dignitaries, all esteemed guests, Professor S. Scott Hall, Professor David Terrero, sir, Professor Dr. Amit Tiwari, sir, Professor J.B. Ramapuram, sir, Dr. Ranjit, sir is here amongst us, Honorable Vice Chancellor, Soviet University. So I welcome you all on behalf of Institute of Pharmacy, Bundelkhand University, Jhansi. Please have a seat. I request to all dignitaries on dais, please have a seat. Please keep continue clapping once again. A warm welcome should be there. Thank you guys, thank you very much. I request all dignitaries on dais, please have a seat. Amongst us, we have our three officers are here. Registrar sir is here. Shri Vinay Kumar Singh ji, Mohammad Wasi sir, who is finance officer, controller for exam, Shri Raj Bahadur sir ji. Honorable dignitaries, learned delegates, respected teachers, beloved students, a very warm and healthy good morning to one and all. I, Dr. Prem, feel privileged to have and feel privileged to welcome you all on behalf of Institute of Pharmacy, Bundelkhand, Jhansi, for today's international conference, New Paradigms in Pharmaceutical Research and Development. Ladies and gentlemen, as we all know, no program can be started without remembering of Almighty. So we have a lamp lighting ceremony. I request all dignitaries on dais, please proceed for lamp lighting. I request our boys and girls to present Vandana of Ma Saraswati. As a tribute to Ma Saraswati, the goddess of knowledge. Hello. Thank you, ma'am. Thank you very much, ma'am.
Asari sanini sani papa ni pama 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 pani dana sa papa pani mama pama pama pani sa. Ah. Ma Saraswati Sharidi, Ma Saraswati Sharidi, Purneshwar Gyani Udati Se, Purneshwar Gyani Udati Se, Veera Pani Tarde, 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 Ma Saraswati Sharide, Ma Saraswati Sharide. Saraswati, Saraswati, Sharide, Ma Sharide, Sharide, Ma Sharide, Surya Brahma Shiva Vishnu Gajanan, Jin Charanon ko Devate, Surya Brahma Shiva Vishnu Gajanan, Jin Charanon ko Sevate. हम शिशु हैं अज्ञानी माता, इन चरणों का प्यार दे, वृंदा पानी तार दे, तार दे, तार दे, माँ सरस्वती शारदे, माँ सरस्वती शारदे, 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 Sharide, Sharide.
Thank you, all dignitaries and esteemed guests. Ladies and gentlemen, the world is full of diamonds and gems. Some of them are here with us. The galaxy of intellectuals are sitting amongst us. So we have to welcome them. So for this, I request our patron sir, Professor Mukesh Pandey sir, to please to present a token of love to our chief guest, Honorable Member of Parliament, Sri Anurag Sharma ji. Once again, I request to our Honorable Vice Chancellor sir to please present a token of love to our presiding guest, Professor Piyush Trivedi sir. Now I request Professor Avarna Raj ma'am to please to present a token of love to our Honorable Vice Chancellor sir. Okay. Moving forward, I invite Professor Archana ma'am to please present a token of respect to our respected guest of honor, Professor David Terrirosa. In this sequence, I request to Dr. Bhavana ma'am to please present the flower of love to our guest of honor, Professor Pia Vogel madam. Now I request Professor Saurabh Srivastava sir to please to present a token of love to Professor F. Scott Hall. Now I request to Dr. Sovi Singh sir to present a token of love to Dr. Amit Kumar Tiwari sir, who is Assistant Professor of Pharmacology, the University of Toledo, USA. Now I request to Vijay Singh sir, Dr. Vijay Singh, to please to present a token of love to Professor Jayachandra Babu Ramapuram. Now I request to Dr. Alok Mahor to please to present a bouquet to Professor Ranjit Singh sir. I request to Dr. Alok Mahor to please to present a bouquet to Dr. Ranjit Singh sir. Once again, I request to Dr. Vengesh Dikshit sir to please to present a token of love to head of the department, Dr. Piyush Bharadwaj sir. Now I request to Dr. Nandlal Singh, please come on dais and present a token of love to Professor Sunil Prajapati sir. Now we have our patron sir, who I think shifted our token of love to Dr. Bogal ma'am. Now I request to Dr. Devesh Nigam sir to please to present a token of love to our patron, Professor Mukesh Pandey sir. Now I request Sri Dr. Rishi Kumar to please to present a token of love to Dr. S.K. Jain sir, who is sitting off the dais, first row of dais, Professor S.K. Jain sir. Now I request to Dr. Upen Sharma sir to please to present a bouquet to Dr. Sailen Saraf sir. Now I request to Dr. Sasi Alok to please to present a token of love to our registrar sir, Shri Vinay Kumar Singh ji. Now I request to Dr. Vijay Singh sir, please to present a token of love to our finance officer sir. Now, last but not the least, I request to Dr. Ram Narayan sir to please to present a token of love to our controller of exams sir, Shri Raj Bahadur Singh ji. Thank you very much sir, all dignitaries and guests who are presented here. We are delighted to have amongst us. Now, I request Dr. Piyush Bharadwaj as a convener of the today International Conference New Paradigms in Pharmaceutical Research and Development. I request Dr. Piyush Bharadwaj, please come on dais, sir.
Namaskar to all of you. A very warm welcome to all of you present over here in virtual mode as well as physical mode, because this conference is also on online mode too. This is very first international conference of this institute of this university. The title of the conference is New Paradigm in Pharmaceutical Research and Development, that is NPPRD 2023. Firstly, I welcome our patron, Honorable Vice Chancellor, Professor Mukesh Pandey ji, our chief guest, Honorable Member of Parliament, Sri Anurag Sharma ji, and presiding guest, Professor Piyush Trivedi ji, former Vice Chancellor of Rajiv Gandhi Technical University, and Honorable Vice Chancellor of Shobit University, Professor Dr. Ranjit Singh ji. I also welcome different expert and resource person from different institutions. Professor Amit Ketiwari from University of Toledo, USA, and Professor Akshot Hall from University of Toledo, USA. Professor Pia Vogal, Southern Mother State University, USA, and Professor Sanjay Jainji from Sagar University, Madhya Pradesh, and Professor Jai Ramapuramji, Alwan University, USA. Professor Shailendra Kumar Sharafji, APJ Abdul Kalam Technical University, Lucknow, and Professor J. Tabit Terrido, Alwan University, USA. Professor C. Karthikeyan from Indira Gandhi National Tribal University, Amarkantak. I welcome administrative authorities of this university, Registrar Sri Vinay Kumar ji, Finance Officer Sri Wasi Muhammad ji, and Controller of Examination Sri Raj Bahadur ji. Now I welcome all the deans, director, heads, faculty, members, non-teaching staff, and members from the House of Media. In very short, I would like to inform that this international conference is of 2 days. In first day, that is on 27 today, there shall be 3 expert talk session, and the postal presentation of the conference shall be held in the Institute of Pharmacy. And in day second, 28th February, there shall be 5 expert session, and after this, the evaluation of the poster session shall be held, and interactive talk with the expert session shall also be held. After this, cultural and valedictory session shall be held. As all the session of this event are for basically for better health and knowledge, so I would end my welcome talk with two mantras, that is, Vidya Dadati Vinayam, Vinayam Dadati Patratam. And the main focus of our profession is on the better health. So at last, I would say, Sarve Bhavantu Sukhina, Sarve Santu Niramaya. Dhanyavad. Thank you to all of you.
Thank you very much, sir. Now I request to our patron, sir, Professor Mukesh Pandey, sir, who is Honorable Vice Chancellor of Bundelkhand University, Jhansi, to please come up on stage, sir.
Thank you so much. In the premises of Bundelkhand University, Jhansi, and in Bundelkhand area of Uttar Pradesh, India, on behalf of the Bundelkhand University, we are ready to welcome you all on the inaugural function of the 2 days international conference on new paradigm in pharmaceutical research and development. It's a moment of great privilege and honor and remembrance in the history of the Bundelkhand University that we are organizing an international conference in which from the various parts of the country and also the US and other countries, the experts have come and they are going to deliver the keynote speech. On this occasion, the very distinguished dignitaries, very dynamic, energetic, enthusiastic, always having a cheerfulness on his face. With honor and respect, I welcome very vibrant and dynamic Member of Parliament, Chief Executive Officer of the one of the leading Ayurveda Company of India, Baidyanath Group, Managing Director, सभी लोगों के लिए चिंता करने वाले, सभी लोगों के लिए आसानी से मिलने वाले, having a flexible approach, हमारे बड़े भाई श्री अनुराग शर्मा जी का मैं स्वागत, वंदन, अभिनंदन करता हूं. He is very busy person, traveling abroad in various parts of the country. So when I requested him कि some very high level scientists are coming from the US and other part of the world. So as they are visiting India and they are coming to Bundelkhand University, so your presence is very much requested. He has given the immediate consent and really we are grateful and thankful to you to be present here today. My mentor, my guru and my former Vice Chancellor, very great academician. He is always working as a guiding force. He has given the motivation and the various directions for my capacity in career building. I worked with him when he was the Vice Chancellor more than 8 years in Rajiv Gandhi Technological University, Bhopal. And we are very much delighted to his presence in the premises of the Bundelkhand University, along with the brigade of the experts from the pharmacy field, Professor Piyush Trivedi ji. We are honored to have your presence in the premises of the Bundelkhand University. Also, at this conference, भारत की जो सांस्कृतिक परंपरा है, ladies first. So, on behalf of the Bundelkhand University, I am also welcoming Professor Pia Vogel. Please stand up and namaskar. Yeah. She is also a leading scientist and holding very key position We are also thankful and grateful to Professor F. Scott Hall. Please stand up so that they will join. Thank you. Bundelkhand University is also welcoming innovator having a very scientific approach in all the discoveries and having a more than 40 drugs in the market designed and developed and innovated by Professor J. Ramapuram. Thank you. I would like to welcome Professor David. He is also a leading scientist in the pharmacy. Please. And very young chap of the 5th generation in pharmaceutical field, Professor Amit Tiwari. Yeah, we do have with us also Professor Saraf. Professor Ranjit Singh. He is also the Vice Chancellor, one of the university in India. So really we are thankful to my international friends from traveling from USA to Jhansi. In this auditorium, we do have the other experts from the pharmacy field from the Pan India, Professor Sanjay K. Jain from Sagar University. Professor Shailendra Saraf. I am also welcoming very young scientist, Dr. C. Karthikeyan. Senior deans, senior professors, and the head of the department of the various departments of the Bundelkhand University who are present here, and the UG, PG students, especially from the pharmacy and other programs also, the members of the electronic and print media, and our participants and delegates from various institutions, various universities, and various parts of the country. All are you have been welcomed in the premises of Bundelkhand University. So as the time constant is there, because our honorable member of parliament has to go somewhere for some function. So just I wish to that Bundelkhand University is a 46-year-old university, my dear friends. And this university, the main mandate of this university is to teach, train, conduct research, collaborative research, innovation, and having the vision and the goal for generation of the new knowledge for the community, nation building, and for the students' empowerment. The university offers the diploma, undergraduate, postgraduate, and PhD program and various streams and the programs. The territorial jurisdiction of the Bundelkhand University is in the 7 districts of this Bundelkhand region and covering 368 institutions in the area. In these 368 institutions, more than 0.3 million students are studying. In the university, in the education field, 9 faculties are there. In the management, education, agriculture, arts, engineering, education, forensic, physiotherapy are the some different courses. We do have 29, more than 100 programs in the campus. And as far as concerned with the courses, we have more than 2700 courses in which the students are studying. The in-campus strength of the students, more than 12K in the campus. The intake capacity in all the programs in more than 5,000 each year. This university having a NAC accreditation by the highest body of India. We do have a ISO certification, and I am proud to share that the Institution of Pharmacy of Bundelkhand University is having a 73rd rank in National Institutional Ranking Framework, NIRF, 73. So, and the students are getting a good placement from the pharmacy institutions, and certainly this universities also have a more than 110 MOUs. In the last 1 year, we have signed more than 100 MOUs, and out of that, more than 15 MOUs from the various countries like the Germany, Canada, USA, etc. And I am happy to share today also we are signing one MOU with University of Toledo in his presence. As far as concerned with this particular conference, more than 800 students are participating, and the various topics and sub-themes are covering the latest area, and certainly that will open the new avenues and the new opportunities for the students in the drug discovery and the designs. As far as concerned with the Government of India, because of the new policy, the medical healthcare system in India is increasing day by day, and we are also focusing for the export. And in this Bundelkhand region, the pioneer pharmaceutical industry is from the Ayurveda, and the chairman and the CEO is here. There are ample employment opportunities in the pharmacy, lot of opportunities for the entrepreneurship and the incubation startups. And because of the policy of the Government of India, India's position at the 3rd in the world startup. The conversion of the startup into the unicorns and the transformation rates is very high. And Bundelkhand University have also established one incubation center in which some startups are working. With the support and help of our dynamic member of parliament, this university is the only university in the state of Uttar Pradesh who has received the approval for establishment of the technology enabling center. That's a very prestigious program. Through that center, we will work for the technology dissemination and deployment in the nearby area. We will try to convert the products into the commercialization by the tie-ups with the industry. So this is so many things and the academic excellence achievement of the university. But considering the time constraints, once again, I welcome all our foreign experts, all the participants, all our distinguished delegates. And I wish you a very pleasant stay here in Bundelkhand University and here in the lush green campuses of university. Certainly your stay will be very productive and successful, and I wish my grand success, every success for these grand achievement, outcomes, fruitful outcomes in the pharmacy field after end of this deliberations in the conference. Thank you so much.
Thank you very much, sir, for your kind words for us. This will definitely encourage us. Now we have a short speech of our distinguished guest. So first and foremost, I would like to invite Professor Madam Priya Vogel. She is Professor of Biological Science, Southern Methodist University, USA. Now I request to Priya Vogel ma'am on stage, please. Priya Vogel ma'am.
Good morning, everybody. I'm very honored to be here. Thank you guys for coming. Thanks for organizing and hosting this. I will be talking about my research a little bit later today. And I just can say thank you. And I'm, as usual, since those last 10 days in India, completely overwhelmed by the hospitality that you have shown us.
Thank you very much, ma'am. Moving ahead, I request to our guest of honor, Professor S. Scott Hall, Director of Pharmacology and Toxicology, the University of Toledo, USA.
I would like to echo Dr. Vogel's comments. Thank you very much for the kind invitation to be here. I'm very much looking forward to the next couple of days for the science, for meeting all of the people here, and for meeting the students as well. These types of conferences have a number of purposes. It's to bring us all together. It's to learn about science, but it is also to promote the education of young people. And we want to move forward on all of these things while we're here. Thank you very much.
Thank you very much, sir, for your kind words. Now, I cordially invite to our esteemed guest, Professor David Terrio, sir, who is a professor of pharmacology, Auburn University, USA. Okay, now I request to Dr. Amit Tiwari, sir, who is an assistant professor in USA. So I request him to please come on stage, sir.
I think he got my title wrong. It worked very hard to become a professor, so it takes several years and a lot of effort to grow in life. And you guys know, who are faculty here, what are the challenges. Okay. Good morning again. Thank you for having me. I feel really honored and privileged to be able to come and talk to you. And Jhansi is really, really important to me because मेरा गांव बहुत पास में है यहाँ से. चित्रकूट के पास एक गांव है भौरी, तो अभी भी वहीं पे सब खेती बाड़ी करते हैं. और आई फील वेरी ऑनर्ड कि मैं जहाँ पे आ पाया हूँ, यहाँ पे आके आपसे बात कर पा रहा हूँ. आई एम रियली रियली थैंकफुल फॉर द ऑर्गेनाइजिंग कमिटी. एंड just so that you know, it takes a lot of effort to build a conference, an international conference, and bring delegations who are world-renowned expert in their field to come and talk to you. Okay, so this conference is really for the students and the faculty and of course scientific exchange, but use this opportunity to talk to people who are here in next few days. And so very important people have taken a lot of their time to be here, to share this experience together. So I really want to thank the chief guest, who is very busy handling the whole entire region, Shishal Sharma ji. Thank you so much for being here. And of course, all the very important persons on the dais, and some sitting in the front row, who are pioneer mentors and leaders who have created more leaders. So again, thank you so much. And I cannot thank enough Professor Trivedi sir for always mentoring young generation. And of course, the Vice Chancellor, the patron, who has this vision to actually have a conference of this size. We have been talking, and I'm really impressed by how much people have vision to think far and beyond, to bring science and technology to this place. And I would request and take this opportunity, because I know the chief guest is going to leave because it's very busy, is to somehow create a center of innovation and research here, provide that infrastructure and build an infrastructure in this region. So, I will do my best to bring people from United States together and some other places together to build some sort of technical center here to be advanced. And somewhere in the region, in the area of phytomedicine, because people do phytomedicine, but there is not a very, very solid program anywhere in the world which can actually facilitate that. And maybe this would be one of those centers where it will be from molecular mechanism to all the way to technological development. And I think, that request would be taken fairly. And another thing is, I really hope that this conference can continue every year so we can benefit and more people can come and join us. So again, thank you so much. It was supposed to be a short speech. I took too much time, like I always do. So being a professor, that's a problem we have. We talk too much. Again, thank you.
Thank you very much, sir, for your profound words and speech. Now, moving forward, I would now request to our esteemed guest, Professor Jayachandra Babu Ramapuram, who is a professor of pharmaceutics over University, USA.
Good morning, everyone. I'm very humbled, honored to be here for this wonderful conference with a lot of delegates. And I'm very thankful to our organizers have me here, invited me here to participate in the conference and share some of my experiences in the form of a lecture sometime today or tomorrow. So I am from Andhra Pradesh. I did my Bachelor of Pharmacy from Madras Medical College, but as Dr. Tiwari said, I have some roots here. I did my PhD, MPharm and PhD from Banaras Hindu University. So मैं भी इधर का हूँ, and I met a lot of eminent pharmaceutics faculty yesterday, and I'm looking forward to interact with pharmaceutics faculty as well as students in the coming days. So technologies are emerging. There are several newer pharmaceutical technologies compared to the conventional pharmaceutical technologies. There are new areas like biopharmaceutical development leading to biopharmaceutical drugs, which have got delivery challenges. And there are so many thousands of molecules, which are about 1,000 molecules are in clinical trials belonging to biopharmaceutical sector. And it will be very timely to have this kind of platform to discuss those challenges and current knowledge. And I'm looking forward to interact with you all. Thank you very much again for inviting me.
Thank you so much, sir. Now, we are very delighted to have our presiding guest, Professor Piyush Trivedi, sir. Before inviting him, I request to Ms. Princy Rai to please introduce him as a deep insight of him.
Thank you so much, sir. Professor Piyush Trivedi is presently serving as Director Honorary of the Center for Innovation and Translational Research, CITR, a visiting faculty at the University of Toledo, USA. He has graduated from Indore University with bachelor's degree in pharmacy 1979 and then obtained his postgraduate degree in pharmacy from Sagar University in 1981. Then received his PhD in pharmaceutical chemistry from Indore University 1985. Professor Trivedi started his career in teaching from Holkar Science College, Indore, 1987. He then advanced to the post of professor in 2000, and in 2003, he moved to RGPV, where he became the founder head of the School of Pharmaceutical Science, a university teaching department. In 2008, Professor Trivedi was appointed as Vice Chancellor, RGPV, Madhya Pradesh, a state technological university, where he was the longest serving Vice Chancellor, RGPV. Professor Trivedi has filed 2 US patents and 7 Indian patents related to new drug discovery and development. He is also a recipient of several prestigious awards, and the most recent being the Innovation Award for the Discovery and Anti-Cancer Molecule at the 3rd National Madhya Pradesh Education Summit and Awards organized by CMAI Association, Madhya Pradesh, India. Thank you.
Thank you, Princey. Now I would request to our honorable presiding guest, Sri Dr. Professor Piyush Trivedi, sir.
Good morning to all of you. Chief guest of the function, Honorable Anurag Sharma ji, the Member of Parliament from here, and a dynamic leader in the pharmaceutical industry who is present over here, Dr. Mukesh Pandey, the patron and the Vice Chancellor of the Bundelkhand University, and Dr. Priya Vogel, Dr. Amit Tiwari, Dr. Scott Hall, David, Babu, other guests on the dais, the members of the faculty members of the Bundelkhand University, Registrar, and other members who are sitting over here, dear students, guests, and members of media. I'm very happy to be here. I mean, I can't express because I wanted to come 1 year back when Dr. Pandey became Vice Chancellor. So he was always telling you come here, but we were trying to find out some time when I can be here and then see. So it's a time when we are at a very big event where this conference is being organized. I must congratulate Mr. Bharadwaj and entire team of the pharmacy department who has organized such a wonderful event on the very relevant topic, new paradigms in pharmaceutical research and development. You know, the whole thing as far as pharmaceutical sciences and healthcare is changing. What is required is that we have to have the research which should benefit and help out the patient care. That is the most important area. Right now, as we are discussing with the Honorable Member of Parliament, that what we do is we are mostly in a manufacturing of generic medicine. We are the third largest producer of the generic drugs. Volume-wise, maybe we may be the highest. But what else? When we consider any other thing, as far as technology is concerned, we have, just please, yeah. When we consider the technological part of it, when we look here and there, that from where we have to bring the technology. We have been working hard for so many years. We have seen that there were some companies which were engaged in the research of new drug discovery. Almost everybody has closed down it. Almost everybody has closed down it. Drug discovery is now not a subject in India. Why most of the organization, almost none of them is doing, because of the very high cost of production, about 3 billion of rupees, which are involved to bring one single drug, and only 5 drugs come out in the market when 10,000 more drugs they go to the clinical trials and that. Looking at such a huge investment. So what can happen? What should we do? How we are getting technology from the university, from the other developed countries? How it is coming from the US and European countries and other countries? We have to look forward and change our perception. Since the policymaker and the member of parliament and the member in the deputy council, general council is here, it's very important to bring to your attention, sir, and all of you who are sitting over here, we have to change our perception. The first is the change of perception that what we should do and how we should do. If really we have to produce some technology in the entire US, not only the technology is coming from the industry, but the technology comes and flow from the universities. So it is time when Dr. Pandey has organized such a good conference on a very relevant topic, what is to be done in time to come. And what and how the people who are sitting in the universities, various universities, the teachers, the students, the researchers, how they have to do it and they have to move forward the things. There are things which are to be done. One is discovery. The other one is development. The third part are the instruments which are there, the implants and other things which are there. And more than that, I am looking forward one thing which can be done very easily. And people should think, we are not thinking in that direction at all. We are very slow in thinking in that direction. We always look at that, let us see that formulation can do this. We modify the formulation, we repurpose the molecule, we repurpose the drug molecule, and all that we are doing, biosimilar drugs and all that. But there is one thing which is very important, which is lacking, particularly in the institutions, that let us see, we look at the diagnostics. Instead of treating the patient with a drug, if you have some very important diagnostic tools which can really detect the disease at an early stage, the dangerous diseases like cancer, which cancer is one of the very important disease, which is, India is suffering from that. We are not prepared for that to treat cancer, such a huge investment. We invest in money in treating it. If we do something different, let us see, it should alarm and ignite the minds of the young people. If we have some early stage diagnosis of the disease like cancer, Dr. Scott Hall is here from the neurological disease, because now you look at the average age of people has increased in India. It has gone over 65, 70 years. When you have more average age, there are diseases like Alzheimer's, neurological disease. So early detection at any point of time to these diseases will save a lot of money, one thing. One is that you are going to save a lot of money in case of the treatment, because once you detect at the immediate step, you are able to start the prevention of that disease to occur. And second part is that we contribute to society where, this is such a large society, we can just help out the people at the different ages. Now, as far as the other part is concerned, we are looking a lot of change with the entry of AI and machine learning. Artificial and machine intelligence and machine learning is another area which has come to drug discovery and development side. Now, how to look at it? I have been reading so many papers and so many things. This was started from they have collected data bank in Carnegie Mellon, CMU, USA, and they started working on that. Followed by a number of companies in the IT field, like IBM has come with the Watson model for drug discovery and part. What they have done is that since we have a big data, we have very large data. If we know the data, even for the patient, even for the treatment, even for the disease, even for the molecules, even for the clinical research and everything, that data is being stored at one place. And if that data is being processed, the market, which is very high, it can be reduced if you make use of IT tools, the artificial intelligence, and the machine learning. So a lot of automation. This is not only one thing, but very recently I was reading an article which is published by Lady Bala from Wipro. They have said that how we can proceed actually in optimization, preclinical trials, even up to clinical trials. Even up to clinical trials, what should be the set of action? Except for certain inhibitions are there. So I think all this, the new things which are coming up, emerging us, like the 3D printing and novel delivery and all that, we have to change ourselves. Let us see. We move from theoretical research. We move from publishing only the papers, academic papers, fulfilling the requirement from the to the translational research. Look at the molecular research. Look at the molecular drugs which can come out. You look at the things which are taking place at the molecular level, understand the biology, deep biology, and see that how can you really design a drug, delivery drug, and everything can be done. I hope that the deliberation in this conference, with the presence of the experts from United States of America and other experts from India, going to yield a lot of things. At least it storms the minds of the young people. The only one thing is there, that what we can do is that we can direct to the people, we can convey our message, what they have to do. If two of you can opt for that and really grow, that will be the real success of the conference. I thank you, the organizer of the conference. I thank Dr. Mukesh Pandey and Honorable Chief Guest of the function who has spared time for us, and I'm thanking for inviting me. All the best, my best wishes for the conference. Thank you.
Thank you so much, sir. What a great moment and what a great program for this history of Bundelkhand University. I thank sir. Thanks for blessing all of us with your kind words. This is such a great opportunity for Bundelkhand University and all the officials of Bundelkhand University to honor the people around us who are contributing a lot in the welfare of society and who are giving a different level of development for the society. Bundelkhand University administration and today's chief guest, Professor Anurag Sharma, with Professor Ranjit Singh sir, that Bundelkhand University has decided to honor our guest with few awards. I would like to request Honorable Vice Chancellor sir, Honorable Chief Guest of the ceremony, and Professor Ranjit Singh sir to please come forward to the stage for these award distribution. The first award goes to Dr. J.B. Ramapuram with the title of Distinguished Pharmaceutical Investigator Award. So can we have a huge round of applause for Dr. J.B. Ramapuram, the Distinguished Pharmaceutical Investigator Award. We are giving this honor to you, sir, but we all are really feeling honored to give you this award. आपकी तालियां अवार्ड के सापेक्ष नहीं है। एक जोरदार तालियों के साथ हम डॉक्टर जे.बी. रामापुरम जी का अभिनंदन करते हैं। द डिस्टिंग्विश्ड फार्मास्यूटिकल इन्वेस्टिगेटर अवार्ड, डॉक्टर जे.बी. रामापुरम। The next award goes to Dr. F. Scott Hall for his contribution as a Distinguished Neuroscientist Award. So can we have a huge round of applause for Dr. F. Scott Hall, who is getting this award titled Distinguished Neuroscientist Award, University of Toledo, USA. Today, Bundelkhand University is really feeling honored to honor such a great personality who is today with us, and this is really a great moment for us. We have the gracious presence of Professor Piyush Trivedi sir, and we all are feeling honored to honor him as the Excellence in Academic Leadership Award. Professor Piyush Trivedi sir, who is continuously contributing and guiding the academics to be more precise. Sir, so can we have a picture with all the award winners, a combined picture with all the award winners? And to all the spectators, I want to request, just give a huge round of applause for all these three award winners of this ceremony. Now, I would like to request Professor Amit Tiwari sir to please come up. He want to do some announcements, so please listen carefully.
Thank you so much again. And since it's award ceremony sort of it began already, we have two special things that we want to give. One is for the patron who I think has been visionary leader, and I think he has impacted the life of a lot of people. He's an innovative thinker. So at the University of Toledo, we would like to provide it with an award. So I will ask Dr. Hall here on the stage to announce the award.
So we're honored to present the Excellence in Academic Leadership Award to Professor Dr. Mukesh Pandey. It is presented by our department chair, Frederick Williams, myself, and Dr. Amit Tiwari. It is in recognition of your dedication to transforming lives through innovative technology and science-based education.
Thank you. So I'd like to ask Dr. Pia Vogel, Dr. Babu to be also there to please provide the award.
We all as a teachers, we all as a students, we really are noticing this transformation in Bundelkhand University. He is getting this leadership award. The Honorable Member of Parliament is there. Professor Piyush Trivedi sir is there. And Professor Mukesh Pandey sir is getting this award. Again, a huge round of applause for the great leadership of Bundelkhand University. This cap is not only a simple cap, it's a cap of responsibilities, a cap for the great leadership and visionary person. Thank you so much.
I just have one small thing to present to the chief guest. He has taken so much time to be here. So we brought a special thing for him from University of Toledo. Toledo is called a glass city because a lot of artifacts that has been designed there. So Dr. Pia Vogel will come and Dr. Hall will present that token of appreciation. Thank you so much, Mr. Sharma ji, for being here with us.
So directly from the University of Toledo, we are getting some special gift to our Honorable Member of Parliament, Sri Anurag Sharma ji. I'm sure this mutual respect, the mutual contribution to each other will provide a quality education, greater opportunities to the world. So all the award winners and distinguished guests are requested to please line up for a group photograph. Guys, can we have a huge round of applause for this frame? These are few lifetime frames which have been recognized even after the decades in the history of Bundelkhand University. Thank you so much, everyone.
Thank you very much, all dignitaries and esteemed guests. Now we have amongst us Professor Ranjit Singh sir. I would like to call him on stage. He will deliver some speech on the theme of today's international conference. Okay, actually, due to some inevitable cause of our Honorable Member of Parliament, he has to go somewhere. So we are pleased to have Sri Anurag Sharma ji. He is Honorable Member of Parliament in Government of India. He is a leading industrialist in the field of Ayurveda. Moreover, he is a CEO of Ayurvedic of Vednath Industrial Group, Jhansi. So I would like to call on stage Honorable MP sir.
Namaste. I'm delighted to be here. The International Conference on New Paradigm Shift in Pharmaceuticals Research and Development. It's one of the few subjects which is completely close to my heart. I come from the same industry. And I'm so very glad that it is happening in my university. I'm a student of this university. It makes me feel very, very proud. Let me first congratulate Professor, the Honorable VC, Pandey Sahab. Sir, I know how much effort it takes to actually hold a conference, and on top of it, you're holding an international conference with so many dignitaries here. Congratulations to you and the entire department and the team which has put this together. आप सबको बहुत बहुत बधाई दिल से. बहुत समय लगा होगा, बहुत मेहनत करी होगी. I'm also very, very glad that we have such an eminent and a distinguished panel of professors. मंच पे बैठ के बड़ा अजीब सा लगता है, सबके सामने प्रोफेसर लिखा है, मैं अनपढ़ सा लगता हूँ. तो हिंदुस्तान में, अच्छा, first let me actually also welcome all of you to the land of the Rani, our first warrior of Indian independence. आप सभी का स्वागत है रानी की धरती पर. It's been an honor and a pleasure to meet Professor Piyush Trivedi ji, to meet somebody हमारे खुद के और अपने अमित कुमार तिवारी जी, चित्रकूट के बेटे हैं आप, तो बहुत खुशी हुई. प्रोफेसर संजय कुमार जैन साहब, रंजीत साहब, प्रोफेसर स्कॉट हॉल, प्रोफेसर जे रामा प्रमाणी जी, प्रोफेसर शैलेंद्र कुमार सराफ साहब, प्रोफेसर विया वॉगल, प्रोफेसर डेविड टेरेरो, Professor C. Karthikeya ji, and Professor Piyush Bharadwaj ji, and of course, Dr. Sunil Kumar Prajapati ji, who is the co-convener of this conference. Distinguished professors, eminent scholars, scientists, and my dear students. I'll keep this very brief. मुझसे तो सीखने को आपको ज्यादा मिलेगा नहीं, सीखने को तो आपको यहाँ मिलेगा. मैं आपको कुछ ही चीजें बताए जाता हूँ. हिंदुस्तान में हम एक चीज कहते हैं, इन इंडिया वी से वन वर्ड, इट्स कॉल्ड वासुदेव कुटुंबकम. वर्ल्ड इज आवर फैमिली, एंड नथिंग प्रूव्स दिस more than the Prime Minister's vision of one world, one vision, one earth. And today in this conference, I would also like to add the words, one humanity. One humanity, because that's what pharma serves across the world. I would also like to congratulate the university on some extremely exemplary work being done here. Very, very proud of the work which is done here, and especially for scientific research. I'll keep my speech a little short. I had a lot of points. But students, I'd like to tell you one thing. I was privileged to spend 10 minutes in the VC Saab's room. I met some of these distinguished professors. In 10 minutes, believe me, if you keep an open mind, I learned so much. I learned about transdermal patches. I really didn't know they existed, except हमारे आयुर्वेद में पैच होते हैं, where we actually use them as painkillers, but here you were talking about Terastron, you were talking about nanomolecule delivery systems. I learned a little about cancer research. I learned so much about new work and new technologies being done. So please, when you are here, when you hear these, make notes, learn. Today you are one of the most exciting times in the world, in any industry. There are three industries which actually drive this economy of the world. One is energy. Unfortunately, most of it is polluting. But new energy developments will take place. India is committed to being a carbon neutral country by the year 2070, which is the Prime Minister's vision. The other big industry we all know is facing a downturn, which is the IT. But pharma industry ek aisi hai, whether you have a pandemic, it goes up. When you don't have a pandemic, it goes up further. So this is an exciting time. Pharma will always do well. But what is more exciting is, for the first time in our history of this country, where it took us 70 years to get the first vaccine to come to India, where the last vaccine came to India in 20 years. But during the pandemic, Indian scientists, Indian students, research fellows, our leading laboratories, universities, when the world said there will be over 34 lakhs, 3.4 million deaths in this country. When the world said 140 billion people will not survive this pandemic, India is going to be the weakest chain in the link. We proved to be the strongest. There is nothing you can't do. Today, Indians are recognized the world over for the finest brains you have, for the work ethic you bring to the table, for the dedication our scientists have brought, where it took us less than 1 year to not only develop a vaccine, but to administer it in terms of 220 crore doses across India. Today, because of vaccine Maitri, we made Indian vaccines available across 150 countries. No other country in the world did it. And this was made possible only by the finest scientific pharma minds in this country. I would like to congratulate all of you, and I would like to encourage the students, please listen to the sessions you have today, make notes, understand it. There is a huge exciting opportunity in the Indian pharma sector. Today, India accounts for virtually 20% of the global trade in generics. But like Professor Saab said, unfortunately, we don't do new molecule discovery. We're more into biosimilars. We actually repurpose drugs. It's time where Indian scientists and Indian institutions went forward and showed the world that if we can develop a vaccine for COVID inside 9 months, we can and do breakthrough research work across the entire pharma spectrum. I think there is a lot of opportunity which exists. We can all do it. I'm also very grateful to the Prime Minister for actually bringing the first WHO traditional center in the world for traditional medicinal knowledge center, a research center. WHO has only one in the world, and that's been brought to India. It's just been set up. I had the honor of being present at that meeting too. I will not go through all the figures I have, but I am going to try and tell you some things which are very, very interesting. And since we have a lot of eminent scientists from abroad, the government of India takes the development of pharma sector extremely seriously. We are one of the few countries in the world where we've accorded production-linked incentive schemes for domestic manufacturing of key critical ingredients and starting materials, KSMs and DIs, and APIs in India. Promotion of bulk drug parks. Production-linked incentive for promoting domestic manufacturing of medical devices. Professor Saab just mentioned diagnostic devices. The first park is actually going to be coming up in UP in Noida. The government of India has granted that to us. That's a very, very exciting field to be in. Today, my watch does a lot of diagnostic work than was possible 25 years ago and going to a lab. We've got health ATMs. I was shocked. We put up one small health ATM. I didn't know what a health ATM was till we put it up in Bar, a small, tiny little village inside Lalitpur. मैं पहली बार गया भैया health ATM क्या होता है? So moment I go and we use that machine, it gives me my BP, it gives me my sugar, it gives me my long-term sugar, it gives me my cholesterol, my lipid profile, it tells me my temperature, it tells me my height, my weight, my BMI. So a small, tiny machine like that, not very expensive, can be put up inside a village, does not require too many people to handle it. Just one small technician, doesn't require a doctor, and gives me an entire printout of all my immediate requirements of diagnostics, of blood especially. And it's all for free. So can you imagine if we can do this in a small village, we can do this across the entire country? So it's a very exciting field. I'm also very happy to say that We are planning to do 3 more things, which will be very good for you, especially the students. Under the Smart City program in Jhansi, we had our first floodlit stadium yesterday. So children can come and play hockey there on an AstroTurf field. It's floodlit. You can come and play at night. But more than that, I'm also happy to announce under the Smart City program, I have also decided to make sure Jhansi gets a new innovation center where young minds can come and participate. We will have teams from Smart India, Invest India to help guide you and your path forward. The other announcement we will make in some few days, but with the VC Saab's blessings, Baidyanath Group has decided to do a scholarship program, especially for drug and molecule research and for Ayurveda and bioactive ingredients, worth 40 lakh rupees for students as scholarships to be given in the university here. I have also insisted, and we are also including smart classrooms for our students here. I'll be very happy if you make a proposal and send it to me. So we will make sure under the smart city programs, we will put up smart classrooms in Jhansi. So that'll be another very exciting field to be here in. I have requested 2 departments. In fact, we can make a presentation to even NIPA, but I've requested 2 departments. One is CSIR, they have some world-class labs, and the Department of Biotechnology to come and fund universities, especially young people in tier 2 and tier 3 cities. Tier 1 cities में तो पैसा बहुत आ जाता है सब. They get a lot of money. IITs, IIMs, but it's universities like Bundelkhand where new innovations can take place. जहाँ हमारे स्टूडेंट्स में वो भूख है, आज उनमें चाहत है, वो काम करना चाहते हैं. आज आईआईटी में बच्चा बहुत बार चले जाता है, वो सोचता मैं आईआईटी पहुंच गया, मेरी जिंदगी सेट हो गई. पर जब बुंदेलखंड यूनिवर्सिटी में बच्चा आता है, उसको पता है कि हमको आगे भी मेहनत करनी है. आप में ही हिम्मत होती है और आप में ही दम होती है ये करने की, क्योंकि आप में ही वो चाहत है और वो भूख है और वो पैशन है. वो जज्बा छोटे शहरों में ही रहता है. जब हम गांव से निकलते हैं, जैसे चित्रकूट से निकले अमित जी, ऐसा जज्बा तभी देखने को मिलता है. So we are planning and designing and I'm hoping कि बुंदेलखंड यूनिवर्सिटी से हमको कुछ प्रोजेक्ट्स मिलें which I can and take to CSIR and DBT. So एक बार हमारे बच्चों के लिए you will get an opportunity to present your thoughts, your innovations, your ideas. And we've also requested them to handhold you, that means mentor you, what is called angel funding as well as mentorship. Both can happen with these departments, and I'll be more than glad to take you there, introduce you to the right people, and make sure that they come here for any such work. I would also request the eminent scientists, since I come from the field of Ayurveda, मेरी छोटी सी रिक्वेस्ट है, don't ignore Ayurveda. Ayurveda is one of the oldest science in the world. Some of you, to tell you, Ayurveda, the word itself means Ayu, life, and Ved means science. It's the science of life. And the science of life doesn't require any clerical processes to prove itself. We have enough history of 5,000 years of empirical evidence of one of the largest human trials in the history of civilization being done on some of these drugs. They've proven themselves, but yes, we need to revalidate them. I wouldn't say there are going to be new drug discoveries, but they will need to be revalidated and put in a new scientific format so the world can understand what Ayurveda does. So it'll be a pleasure and an honor to host you sometimes. And I would request the VCs to ask me to show you some agricultural, herbal gardens. So we will try and arrange a visit for you either today or tomorrow. आप सबको पुनः धन्यवाद कि आप लोगों ने मुझको ये वक्त दिया, समय दिया, अच्छा लगा। इन्हीं शब्दों के साथ अपने बच्चों के लिए, उनके भविष्य के लिए मैं पितामह माई से कामना करता हूं कि आपका बहुत उज्जवल भविष्य रहे। जय हिंद, जय भारत।
Thank you very much, sir. I cannot speak in words or express in words for your profound and wonderful speech. Now I request to our dignitaries, because we have to release one our abstract books and two faculty books. This is actually a proud moment for us. So I request our girls to please come forward for releasing the abstract book. For releasing the abstract book, please girls come forward. I request all dignitaries to please release our abstract book. Thank you very much, sir. Once again, I request to our girls to please come forward to raise our two of our faculty members' book, which is written by Dr. Bhavana ma'am and Dr. Upay Sharma. I also request him to please come on stage to raise your books. Thank you very much, sir. We have one more book to release. I request all dignitaries, and I also request to Dr. Sasya Alok to please come on stage and to release the book, please. Dr. Sasya Alok. Thank you very much, sir. Thank you very much. Now I request to Dr. Anupam Vyas, sir, please take over the mic.
Okay, so before doing the next announcement, I would like to request So before making the new announcement, I would like to request the registrar sir, FO sir, and controller of exams sir to please come on the stage for the next announcement. Please, sir. Today's date again will be marked. Today, Bundelkhand University and the University of Toledo, USA, College of Pharmacy and Pharmaceutical Sciences is going to sign a MOU for the mutual growth. This is again a very big task in the ceremony. I would like to request the Honorable Vice Chancellor, sir, to sign this Memorandum of Understanding between the University of Toledo, USA, and Bundelkhand University, Jhansi, India. So this is a historical moment you all are seeing. Honorable Vice Chancellor, sir, is signing. Professor Amit Tiwari, Professor Skorhal, and all the other dignitaries are witnessing this immensely important moment. Now the pharmacy students will get a chance to get more exposure and do the research work all around the globe. I am thankful to all the office bearers of the University of Toledo, USA, for giving this concern and signing a MOU between Bundelkhand University and University of Toledo, USA. Friends, can we have a huge round of applause for this memorandum of understanding? I request Honorable VC sir and the other officials to please show this MOU in front of all the spectators of this great moment. I request FO sir, Registrar sir, and Controller of Exams sir to please join this important moment. This is not a simple memorandum of understanding, but is a bunch of opportunities which all the pharmacy students are getting. Thank you so much. Thank you so much once again to all the office bearers of the University of Toledo, USA. Because of some prior engagements, Honorable Member of Parliament, Shri Anurag Sharma sir, has to leave the program. But before leaving, I would like to request Honorable Vice Chancellor sir to please present him a memento, a token of gift and gratitude from the Bundelkhand University to Honorable Member of Parliament, Shri Anurag Sharma sir. भारतीय सभ्यता में सम्मान का प्रतीक शॉल, शुभ का प्रतीक नारियल और हम सभी के इष्ट भगवान राम का राम दरबार माननीय कुलपति प्रोफेसर मुकेश पांडे जी द्वारा हमारे माननीय सांसद श्री अनुराग शर्मा जी को भेंट स्वरूप दिया जा रहा है. This is really a great moment. And once again, I am thankful to the Honorable Member of Parliament to take out his time in his busy schedule just to honor this occasion, interacted with all of us. Thank you so much. Thank you so much. एक बार जोरदार तालियों से हम विदाई करेंगे आदरणीय श्री अनुराग शर्मा जी की. And thank you for all the announcements and the support and the hand holding for the Bundelkhand University by the Vednath Group.
This has been a wonderful time for us, no doubt. We are one step ahead, signing memorandum of understanding. Now I would like to request Honorable Vice Chancellor Professor Ranjit Singh sir. He will deliver some speech on new paradigms in pharmaceutical research and development. So I would like to request Honorable Vice Chancellor sir. Professor Ranjit Singh sir, please come on stage, sir.
Thank you very much. Honorable dignitaries on the dais, office bearer of Bundelkhand University, faculty colleagues, and dear participants. At the outset, let me thank the organizers, particularly the patron, Professor Mukesh Pandey, the patron of this conference, and Dr. Piyush Bhardwaj, who made it possible for me to be a part of this grand inaugural function of this new conference on new paradigm in pharmaceutical research and development. I am really happy, elated, humbled, and honored. Thank you very much, sir. Such conferences are definitely the backbone for scientific exchange and giving a platform for exchange of views and to nurture, to make new associations and collaborations, which is must for utility-based research. I'm very happy to see that this conference was organized in such a grand manner. When I entered this auditorium in the morning, it was full of energy. Still it is full of energy. And I was really elated to feel that energy here. The other thing that I have noted here is that a group of scientists from USA and India, they're already working together under the dynamic leadership of Professor Piyush Trivedi ji, who is a renowned scientist, pharmaceutical scientist, with very clear vision and mission. So collaboration association is already there. And what we need to do here is, and what I feel, but the outcome of this conference, I am very much sure that this will be a very useful and yielding conference, especially to the young participants sitting across. And just a call to these young participants that this is a time where we need to learn new skills, we need to upskill, so that we can move ahead with time. You have to be future ready. And for that, such collaborations and associations are the life. I congratulate the organizers, especially the patron, for taking this initiative and organizing conference of this intensity in Bundelkhand University. I am happy to be here, and right from the morning, during interactions, I have learned many things from the scientists from USA, from the scientists who are really working here in my country and fulfilling the dreams of our Honorable Prime Minister, that is Atmanirbhar Bharat. I call upon the young participants especially to participate in the coming sessions sincerely, honestly, listen to these scientists who are doing wonders in their field, who are there, who have given already the products which are in the market. You will learn many things from them. And these 2 days, I am very much sure, will be full of learning to all of us. I wish a grand success to this conference. And again, thank you, the organizers, for giving me this opportunity. I wish you all a happy learning during these 2 days. Jai Hind, Jai Bharat.
Thank you very much, sir, for your kind speech, kind words for us. Now, moving forward, I would like to request Professor David Terrio, sir, who is our esteemed guest. He is a professor in pharmacology, Auburn University, USA. Sir, please come on stage. David Terrio, sir. David Taylor, sir.
Excuse me. Morning, everyone here. Good morning, good afternoon, Mr. Mukesh Pandey. Good afternoon, all of you teachers and students. It's an honor for me to be in this important international conference, which put in evidence the needs of collaboration work. I think the most important thing what we can find on heart in this conference is the way how to collaborate. And the second one, or as important as the one, is how to encourage the student to engage in science. I really celebrate that you have this kind of institution. I really celebrate that you have a politician as Dr. Anurag Sharma, which really understand the importance of science. Let me tell you this. The most important increase a point in science in Spain, it was because one politician which was scientist could push their influence to have more level in science. So again, I congratulate your effort to do this type of event, and I really feel honored, and I really feel delighted to be part of this. Thank you very much.
Now, I would like to request Professor Sunil Prajapati, sir. He is a senior most faculty of our department, to propose his vote of thanks.
Good morning, all. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. I feel honored and privileged to get the opportunity to propose the vote of thanks and acknowledge the contribution of those who work really hard to make this event possible. I, Professor S.K. Prajapati, on behalf of the Institute of Pharmacy and entire fraternity of the institution, extend my sincere thanks to the Almighty for making today's event a grand success. On behalf of my organizing committee of NPPRD 2023, I extend a hearty vote of thanks to our Honorable Chief Guest, Sri Anurag Sharma Member of Parliament, who spared their precious time from his busy schedule to grace the occasion. Today we had an opportunity to hear your thoughts, and this will surely encourage us in all our future endeavors. Your thoughts have enlightened our minds and have shown us a new path. I, on behalf of the organizing committee, convey my deep regards and hearty thanks to our Honorable Vice Chancellor, Professor Mukesh Pandey. Without their kind support, this event could not be possible at any cost. I am also thankful to the authority, Registrar Sir, Sri Binay Kumar ji, Finance Officer, Sri Wasim Mohammed ji, Examination Controller, Sri R.V. Singh ji. They helped us in obtaining the necessary administrative
approval for organizing the event in our campus. They supported in all possible manner to organize this conference. I want to thank our presiding guest of honor, Professor Piyush Trivedi ji, for his active participation in our event. You truly made the day enlightening for us. Your deep and intellectual way of imparting knowledge has added to the glory of this event. Today, we have got a lot of information which will truly help us in our life. We have no words to offer gratitude for your valuable presence here. You gave deep insight into the topics and revealed some interesting facts. I also want to extend thanks to all the respected speakers who have entertained us with their important and worthy opinions. I want to appreciate their efforts for making the events informative for all of us. I must mention our deep sense of appreciation, Professor David Terrero for his explanation of natural product with their application to pharmaceutical industry. I would like to express our sincere thanks to Professor Babu Ramapuram for giving excellence coverage to his idea. We are all inspired by your great words. I would like to thank all of the honorable delegates, Professor David Terrero, Professor Pia Vogel, Professor F. Scottal, Dr. Amit Kumar Tiwari ji, Professor Ranjit Singh ji, who blessed us with their presence. I am also very much thankful to all Program Advisory Committee members and the invited speakers, respected Professor Sanjay Kumar Jain, Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, University Sagar, Professor Shailendra Kumar Saraf sir, Director BBD, Northern India Institute of Technology, Lucknow, Dr. C. Karthikeyan, Indira Gandhi, National Tribal University, Amarkantak. Words are not enough to thank constant guidance and support to save the conference. An event like this happened overnight. The wheels started rolling a week ago. It requires planning a bird's eye for detail. We have fortunate enough to backed by a team of very proactive and dedicated staff. I am in short of words for their involvement and their willingness to take the completion of tasks beyond their comfort zones. Special mentions to our head of the department, Dr. Piyush Bharadwaj, for being the catalyst that stimulated us to do our best. With a deep sense of appreciation, we thank our teachers and non-teaching staff for their untiring efforts. Our sincere gratitude goes to all the students, research scholars, teachers, and participants from the different parts of the country and abroad for their presence in the campus of Bundelkhand University, Jhansi, who really made this event successful. I would like to thank from the bottom of my heart to our budding volunteers that made this event successful and their impressive performance. Your ideas and day of hard work were evident in your acts. I would like to thank all the sponsors and respective representatives from the pharmacy colleges and pharma industry who have blessed us with the financial support and their presence. I am thankful to all the staff of the university for their efforts and arranging this function. All the speakers have beautifully added to the glory of this event. I am gratified to all the audience for coming here and for their best wishes for this event. Last but not the least, a big thank you to each one of you who made the celebration memorable. We thank you for being with us. It has remained a pronounced pleasure. Finally, I would like to thank Dr. Kausal and his media team, representative from the various printed and live media, who shall summarize and cover the thoughts of our dais dignitaries, speakers, and motives of this international conference to make it successful. Have a great day ahead. Thank you very much again. Thank you.
Thank you very much, sir. Last but utmost important moment is to be held because it's time to honor our esteemed guest, who are sitting on stage or dais. So to honor him, I would request to our Professor Mukesh Pandey sir to honor our presiding guest, Professor Piyush Trivedi sir, to present Saul's rifle and memento to him. I would like to invite all officers, registrar sir, finance officer sir, or controller of exam sir to please come upon dais and share these moments, please. Once again, I request our Honorable Vice Chancellor sir to please to present Saul's rifle memento to Professor Amit Kumar Tiwari sir. Now I request Professor S.P. Singh sir to please come on stage to present a token of Saul's rifle memento to our Honorable Vice Chancellor sir. Professor, right, there is. Please, Scott Hall. Professor Scott Hall sir. Okay. Now I would request to Vice Chancellor sir, all officers of the dais to please to present a memento and Saul's rifle to Professor Pia Vogel ma'am. Now I request all officers of the dais to please to present a memento to Professor F. Scott Hall. Once again, I request to all officers of the dais to please to present a memento to our esteemed guest, Professor Jayachandra Babu Ramapuram sir. Now I request all officers once again to please to present a memento to Professor Ranjit Singh sir. I request all dignitaries to please to present a memento to Professor Ranjit Singh, sir. Now I request all dignitaries once again to please to present a token of memento to our Dr. C. Karthikeyan, sir. Prof- Chalan, sir, आप. Now I request all dignitaries on dais. Now I invite Professor Sailen Saraf, sir, to come up on dais, sir, please, for to take a memento from our officers. Now I invite on a stage Professor S.K. Jain, sir, to come on dais, sir, to take memento from our officers and dignitaries.
मैं तो अतिथियों से निवेदन करूंगा कि एक ग्रुप फोटोग्राफ के लिए हम लोग मंच पर इकट्ठे होंगे. I would like to request all the honored guests that we will gather on the main stage for a group photograph. I think it will be better if you will carry your memento with you. मैं request करूंगा, I would like to request everyone to please carry your memento with you so that it will look means more official, more honored, and everything is good. I would like to request to please carry your memento with you. If you got some award, you can take the award with you. This. So friends, can we have a huge round of applause for this moment where we are honoring And even after honoring others, we ourselves feeling honored. I would like to request the organizing team members to please come up as soon as possible. All the organizing team members are requested to please come up for a group photograph. I think the dignitaries can sit at their places, and we will all line up behind you if it is possible. I would like to request all the dignitaries to please take your seat on the dais, and all the stage committee will be stand behind you. So all my dear students, now you can see the team in front of you who worked really hard to just put their efforts at the maximum level to make this so possible. इस टीम के एफर्ट्स के लिए जितनी तालियां होनी चाहिए उतनी नहीं है. एक बार जोरदार तालियां इस ऑर्गेनाइजिंग टीम के लिए, डिपार्टमेंट ऑफ फार्मेसी के लिए, जिन लोगों ने आपके सामने इतने बड़े प्रोग्राम को सफलतापूर्वक आयोजित कराया. I really want to congratulate each and every team member under the visionary leadership of Professor Mukesh Pandey sir to make this event possible. And after seeing this program, we really feel that Bundelkhand University is spreading its wings really in all the dimensions to give you a new fly in your career. Great frame. Thanks a lot to all the organizing committee members. Thank you. All the faculty members who are present here, either they are from pharmacy department or not, they are requested to please come over the stage and for a group photograph. All the faculty members who are not, especially belongs to pharmacy department, they can please come up. ऐसे सभी टीचर्स जो दूसरे विभागों से भी हैं लेकिन अभी सभागार में उपस्थित हैं, आप सभी से निवेदन है कि हम लोग मंच पर आकर इस एक ग्रुप फोटोग्राफ के लिए आएंगे. All the faculty members. Sir. So here we can see the diversity of Bundelkhand University. The teachers of different departments, computer applications, Hindi department, management department, architect department, and all the departments are lined up here. A physiotherapy department. So this is the USP of Bundelkhand University, that we have unity in diversity. As Honorable Vice Chancellor sir has mentioned in his speech, 9 faculties, 368 colleges, around 3 lakh students. This is the diversified Bundelkhand University. So can we get a huge round of applause for this diversified education provided by Bundelkhand University? So now we will stand at our places for national anthem. Okay. हमारे कार्यक्रम के स्पॉन्सर्स अगर हमारे मध्य हैं, if I am audible to the sponsors of this international conference, Do we have the presence of sponsors?
अलग अलग कॉलेज, फार्मेसी कॉलेज से जो भी रिप्रेजेंटेटिव आए हैं, जिन्होंने स्पॉन्सर किया है, और इंडस्ट्रीज से जो भी स्पॉन्सर हैं, उनसे निवेदन है कि वो आ जाएं, कृपया स्टेज पर आ जाएं.
So once again, we are ready for the national anthem. An anthem which gives us a goosebumps feeling. Dubey ji.
भारत माता की, भारत माता की, भारत माता की
जय.
मातरम, मातरम.
So these are the two new words for all the our foreign delegates, this Bharat Mata Ki Jai and Vande Matram. We are treating our country as our mother. So that's why we always say that Bharat Mata Ki Jai. Jai stands for the victory. So we are trying to make our India victory everywhere. So that is why we are always praying for Bharat Mata Ki Jai. And second one is Vande Matram. It means we are folding our hands in front of our country and trying to put our best gratitudes to make our country progressive.
This is time for lecture session. So for this, I would invite Professor Sanjay Jain sir to please to present the session. Chair will be done by Professor Piyush Trivedi sir. इसको लगाओ फटाफट.
Okay, so let me inform to all the participants of this international conference, all the dignitaries, that now immediately after the inaugural ceremony, we are starting the first session of this international conference on new paradigms in pharmaceuticals, research and development, NPPRD 2023. In this first session, although we are running late by some time, but still we are trying to optimize the time limits. The first session, the expert talk by Professor S.K. Jain sir, and the session chair will be Professor Piyush Trivedi sir. Once again, I would like to request and inform to all the participants and the dignitaries from the various parts of the country that now immediately after the opening ceremony, we are conducting our first session, and in the session, the expert talk will be delivered by Professor S.K. Jain sir, and this session will be chaired by Professor Piyush Trivedi ji. So now we have the expert of today's session, the first session, the expert talk by Professor S.K. Jain sir. Read out to I would like to inform all the participants of this international conference that now immediately after this opening ceremony, we are starting the first session. This first session talk will be given by Professor S.K. Jain sir, and this session will be chaired by Professor Piyush Trivedi sir. So before requesting Professor S.K. Jain sir to deliver his talk, I would like to request Shubham to please come up and just read out the citation of Professor S.K. Jain sir.
Thank you, sir. Hello, everyone. Dr. Sanjay Kumar Jain, sir, is a professor of pharmaceutics presently working in the Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, Central University, Sagar, Madhya Pradesh. At present, he is also holding the position of Director, Planning and Resource Generation of the university, Chairman of the Council for Industry, Higher Education Collaboration, and Chairman of the University IPR Cell. He is a former head of the Department of Pharmaceutical Sciences and also the former Dean of the School of Engineering and Technology, Dr. H.S. Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. With over 33 years of experience in teaching and research and 2 years in the industry, he has 197 research publications. He has also written a book, Elsevier, and more than 40 book chapters in different national and foreign reference books. He has one US, one European, and filed 3 Indian patents. His main research area is the design and development of various drug delivery system for the treatment of disease associated with the gastrointestinal tract and brain. In 2018, he has been conferred with India's most prestigious and highest award, Visitors Award 2018, for his excellence research in basic and applied science. He has also been ranked consequently twice under 2% World Scientist by Stanford University, USA in 2020 and 21. Professor Jain sir has also been awarded thrice with the Indian Drug Manufacturing Association Award for
Now I would like to invite Professor S.K. Jain sir
All students are requested to take their seats and keep silence. This is very important session for all the pharmacy students. Please come in the hall and take your seats.
So, hello. So, may I request you to keep silence, and I request Professor S.K. Jain to throw light on his topic on which he is going to discuss today's talk. Yeah, please, Dr. S.K. Jain.
Good afternoon, everyone. At the outset, I would like to thank organizer of this international conference, especially the patron of this conference, Dr. Mukesh Pandey, Vice Chancellor of this university, and Piyush Trivedi and his team for inviting me and share my views about nanotechnology-based colon-specific drug targeting. When I received this invitation from Piyush Trivedi, Piyush Bharadwaj, then I thought what I supposed to share with our young budding pharmacist. Then I asked Piyush Bharadwaj ki kya karna chahiye. Then he said, sir, aap something say about the colon-specific target drug delivery system, on which I received the President Award in 2018. Actually, I'm working on the development of drug delivery system, especially for delivering drug to those organs where it is difficult to deliver drug, especially the colon, that is the last part of our gastrointestinal tract, and the brain. So in both organs, delivering drug is a really difficult one. So then here I will discuss something about the colon-specific drug delivery system. How will you develop colon-specific drug delivery systems for the treatment of the diseases associated with the colon? Say next one. Actually, when you talk about the development of any drug delivery system, then you will require to know that what will be the concentration is required at the target size or disease organ. So that is very important. If you know that this much amount of the drug to be delivered to the target organ, or we can say the drug which is effective in this concentration, and that to be delivered, only then you can think about the designing of any drug delivery system. Something technical problem, slides are not changing. So well, in most of the textbook of biopharmaceutical, you might have seen a curve that is showing after the administration of drug through the oral route, a maximum plasma concentration will reach, and there will be a minimum effective concentration, and that there will be a maximum safe concentration. And your intention is to keep drug concentration in this above the minimum effective concentration and below the maximum safe level. And that is known as therapeutic window. You are required to keep drug concentration in therapeutic window. Being the scientist of pharmacy, you have to know about what will be the therapeutic window of the particular drug of which you are going to develop the delivery system. So, well, you will require to keep drug concentration in the therapeutic window. That is the minimum effective concentration and maximum safe concentration. And here the drug delivery scientists play a very important role. If he knows about the concentration that to be delivered to the target organ or disease organ, then you are required to manipulate with the drug delivery system so that the effective concentration can be delivered to that particular organ. So after knowing the therapeutic window, you should also know that what are the challenges. After the administration of the drug, whatever route of administration you select, whether it is intravenous route, whether it is oral route or intranasal route or whatsoever it is, the main challenge is that we face the prevention of drug in the biological system after absorption of the drug. Being that this drug is the foreign molecule, body will try to throw it out from the body. And it will degrade, it may degrade the drug. So main challenge that we face, you have to protect the drug in the biological system from the degradation. Then keep drug concentration in the therapeutic window. And you are required to extend product life. This will not only extend the time duration of taking that drug, but same time it will also reduce the cost, right? So that is related with the shelf life of that drug delivery system and maximize drug utilizations. Maximize drug utilization means whatever drug you administered, it should be 100% utilized. If it is 100% utilized, sometimes what happens, say, if you have administered 100 milligram of the drug, and only say few nanogram is reaching to the target organ, and other remaining drug is eliminated from the body, means it is not 100% utilized, only 0.001% utilized. If you develop a drug delivery system which will deliver drug effectively to the particular organ in 100%, means 100% drug if it is utilized, then that will be the best drug delivery system. Then effective targeting that related with the utilization of that drug, if you make drug delivery system target specific, whatever drug you deliver, if it is 100% is reaching to the target or disease organ, that will be the best one. Then patient compliance, no doubt, whatever you deliver, it should have compliance with the patient, and it should be affordable one. This is only possible when you have integrated knowledge of chemistry, biology, and material science. So, well, if you have knowledge of biology, material science, and link with the chemistry, then you will get a pharmaceutical product or drug delivery system. If you know you should know about the biology, what you should know. You should know the molecular level of the life. Then only you can manipulate with your drug delivery system its release that will be reaching to the target organ. Next. So you should know the biology. Say here, we are concerned about to deliver drug to the colon, then you should know the biology of the colon. Next one. As you know, the next, this colon is the lower part of the GIT. This is the last part of the GIT. And it is 5 feet of the large intestine, and rectum is 6 inches. And if you see the colonic membrane, it is totally different from the membrane which you are having in the upper part of the GIT. When I say upper part of GIT, it means the small intestine in the stomach. So colonic membrane is totally different. And because of its complex nature, because of presence of so many barriers, very limited molecules are absorbed from the colon, right? So it has a number of layer like mucosa, submucosa, circular muscular layer, longitudinal muscular layer, and serocell. So if drug is to be absorbed and to be delivered to the blood circulation, it has to cross these muscular layers. Next one. And if you see the, there are 2 routes of delivering drug to the colon. The rectal route, that is the most shortest route by which through you can deliver drug to the colon, and that oral route, that is the most convenient route by which you can deliver the drug to colon. But if you deliver drug through the oral route, then drug has to pass through the different environment of the different parts of the GIT. Like there is a lot of change in the pH. If you see the pH of the stomach, it is ranging from 1.5 to 2, and the residence time of any content in the stomach is 1 hour to 3 hours. And when the content will go into the small intestine, then their pH is gradually increasing from 4.5 to 7.5 from duodenum to the ileum, and the residence time of the small intestine, that is about 3 to 5 hours. So if you want to deliver any drug, because after the small intestine, the colon will come, large intestine will come, right? So if you want to deliver drug to the colon, then your drug delivery system or drug has to pass all these barriers of the variation in the pH, right? Starting from 1.5 to 7.5. And when the content of the small intestine, that is the last end of the small intestine, ileocecal region, when the content will go from ileocecal region to the colon, ascending colon, there is a sudden drop in the pH, starting from 7.5 to 6.4, 6.2, or 6.5. And this drop in the pH is because of the fermentation of the polysaccharides like lactose into the lactic acid. And because of formation of lactic acid, the pH is dropped. Next one. And this is the variation of the pH we can use for releasing drug is specific to the colon, right? Similarly, if you see the microbial content in different part of the GIT, then you will find that the colon is full of microorganisms. It is having aerobic and anaerobic bacteria, microorganisms, right? And if you see the microorganisms present in the upper part of the GIT, means stomach or small intestine, then you will find only 3 types of the microorganisms. And when you see the microorganisms present in the colon or large intestine, it's a long list of the microorganisms present in the colon. And the enzymes released by these microorganisms can be used for triggering the drug release from the developed colon-specific drug delivery system, especially the azoreductase enzyme or the polysaccharidase enzyme that can be used for digesting the drug delivery system in the colon. And because of presence of the lot of microorganisms, your feces is containing about 30% weight of the feces containing the microorganisms. Next one. So if you see the count of the microorganisms, bacterial count in the different part of the GIT, then you will find that stomach is having 0 to 10 to the power 3 colony forming unit per ml. In the per ml only 10 to the power 5, 10 to the power 3 colony forming units. But if you compare it with the microorganisms present in the colon, say 10 to the power 11 to 10 to the power 12 colony forming unit per ml. So in 1 ml, lot of bacteria will find, and these bacteria will release following enzymes, hydrolytic and reducing enzymes, and these enzymes can be used for digesting your drug delivery system and releasing the drug. That will help to release the drug exclusively to the colon. Next one, please. And the enzymes which are released by the bacteria present in the colon that are carrying out following metabolic reactions like hydrolysis, reduction, deocarboxylation, dealkylation, esterification, acetylation. As I told you that the most important enzymes that is being released by the bacteria of the colon, that is the azoreductase enzyme and the polysaccharides. These enzymes, two enzymes, generally we have used for triggering the drug release from the developed drug release. We have used such type of the polymer which will be digested by either azoreductase enzyme or the polysaccharides enzyme and release drug exclusively to the colon, because these enzymes are not present in the upper part of the GIT. Next one, please. And these are the barriers. These are the barriers present in the colon. If you want to get absorbed drug from the colon, as I told you, there are a number of layers. Apart from that number of muscular layers, these are also barriers are present, and you have to pass out these barriers if you want to get in the drug from the colon lumen to the systemic circulation. Say it is having the small surface area. If you compare with the small intestine, that is having the large surface area, but the colon is having the small surface area. The specific and non-specific binding of the drug or colonic content, and that produces hindrance for the transfer of the drug from the colonic lumen to the systemic circulation. High viscosity of the content, because colon, most of the water present in the colon that is left and reaching to the colon is absorbed from the colonic membrane. So the content is highly viscous one. So this is the another barrier for the transfer of the drug from the colonic lumen to the wall of the colonic membrane, from where drug will get absorbed. Mucus layer, that is also a protective layer of the colonic membrane. And in the colon, the thickest membrane is present, mucus layer present, and drug has to cross that membrane also. Then unsterile water layer, that you will find in the colon only, that is just below the mucus layer or the epithelial membrane. You will form a aqueous layer, water layer, unsterile water layer, that is produce hindrance for the transfer of the drug from the colonic lumen to the systemic circulation, especially for hydrophilic drugs, especially for the lipophilic drug. And then colonocyte and presence of the occluding junctional complex. The membrane cells are tightly bound by the occluding proteins. So then paracellular transfer is not possible from the colon. And the epithelial surface, that is lipophilic one, so that is produce resistance for the transfer of the hydrophilic drug. Next one, please. Now, after say, so many barriers, after we know that so many bacteria are present, there is a lot of variation in present. lot of transit time. If you give it any drug delivery system or dosage form through the oral route, if you want to deliver it, you will require, drug will take at least, say, 5 to 7 hours to reach to the colon. So there are chances of degradation of the drug. And say, if you want to deliver drug to the colon there through the oral route, then either you increase the dose of the drug so that after the absorption of drug, whatever drug is remaining unabsorbed that will reach to the colon, or you can add deliver drug after the absorption and through the circulation, it will reach to the release drug into the colon through the colonic membrane. So if we deliver drug into the conventional form or dosage form of the drug, then what will happen? The drug will get absorbed and from the systematic circulation, it will reach to the colon. Just like in case of the metronidazole, for killing the metronidazole, you will, for killing the amoeba, you will require metronidazole in a microgram concentration. But you do know how much amount of the metronidazole we are administering, say 1.5 gram to the 2 gram per day. Because for killing the drug, for delivering effective concentration in the colon, you will require to increase the dose so that some of the unabsorbed metronidazole will reach to the colon. Because most of the drug, metronidazole is absorbed from the upper part of the GIT. So this is the drawback with the conventional dosage form. So we design colon-specific drug delivery system, directly deliver drug to the colon. It will not get absorbed from the upper part of GIT. It will release only in the colon. Next one, please. As you know, colon is the potential site for the drug delivery because of it has low hostile environment, because it has long colon transit time, means the content in the colon will remain more than 20 hours. So you can manipulate with the drug delivery system. You will have sufficient time to manipulate the drug release. Unique microbial flora, and that can be used for releasing the drug specifically to colon. Highly responsible, colonic membrane is highly responsible for the absorption enhancer. And as you know, the route of administration is oral and rectal, and oral route is most convenient one. Next one, please. So which type of drug we can use for incorporating in colon-specific drug delivery system? Those drugs which are having the poor bioavailability from the stomach and which are having a deleterious effect on gastric mucosa, like non-steroidal anti-inflammatory drug, or drug required for the treatment of the diseases associated with the colon, that is the amoebiasis, colon cancer, ulcerative colitis, and inflammatory bowel disease. Next one, please. These are the some of the drugs that are being used for the local action or topical action or systematic action. The most popular example is sulfasalazine, that is the prodrug of 5-aminosalicylic acid with the sulfapyridine, right? It is having the azo bond, and azo bond is exclusively cleaved in the colon, right, by the azoreductase enzymes. So that product, these are the product ipsalazine, valsalazine, or mesalazine. These are all product of the 5-aminosalicylic acid. Next one, please. These are the characteristics of the colon-specific drug delivery system. If you design any colon-specific drug delivery system, you should have following characteristics like releasing exclusively to the colon, increase colonic bioavailability, enhance therapeutic efficacy by localizing drug to the colon, reducing systematic adverse effect by reducing the absorption of the drug from the upper part of the GIT, maximize drug utilized because drug exclusively delivered to the colon, reducing the dose of the drug, maximizing drug retention time in the colon. Next one, please. These are the different, next one. These are the different approaches which we can use for designing the colon-specific drug delivery system for releasing drug exclusive to the colon, like the prodrugs. Prodrug, and the prodrug means azobond drug, a prodrug, glycoside prodrug, cyclodextrin prodrug, dextran prodrug, size-dependent drug delivery system we can develop, and surface charge dependent drug delivery system, pH dependent drug delivery system, and biodegradable drug delivery system means some polysaccharides, some gums we can use for fabrication of the colon-specific drug delivery system. Redox drug delivery system, especially in case of the inflammatory bowel disease, where the ROS level, reaction oxygen species level is very high, that can be used for the oxidizing the drug delivery system, releasing the drug specifically to the colon. Active targeting, this is the approach on which I received the President Award. Right, and the nanocomposition-based colon-specific, dendimer-based colon-specific drug delivery, and time release. Time release is just delaying the drug release, right? It means if you are able to delay the drug release 6 to 7 hours, then by the time drug is about to release, your drug delivery system will reach into the colon. Next, please. This is what product of sulfasalazine, and it is this is the sulfasalazine product, and it is the product of 5-aminosalicylic acid and sulfapyridine. And this azo bond and double bond end is cleaved by the presence of active reductase, and you will get one molecule of 5-aminosalicylic acid and sulfapyridine. The toxicity of this product is because of the release of the sulfapyridine. So another product are being synthesized. Next one, please. That is the balsalagine, ipsalamine, salazide, or osalazide. So these in balsalagine and ipsalazide, the amino acids, instead of taking the sulfapyridine, amino acid is conjugated with the 5-aminosalicylic acid with the help of the 5-azo bond, right? So the azo bond will be cleaved, and you will get a biological friendly molecule of the amino acid and 5-aminosalicylic acid. And in case of the osalazine, this is the another product of 5-aminosalicylic acid, where 2 molecules of the 5-aminosalicylic acid are conjugated with the help of the azo bond. So when this azo reductase enzyme will be acting, you will get 2 molecules of the 5-aminosalicylic acid. Next one, please. This is the glycoside product. Next one. These glycoside prodrug or cyclodextrin prodrug or in the colonic lumen by the colonic bacteria. Next one, please. Next one. This is a dextran prodrug is being synthesized with the help of the dextran and the dexamethasone and methylprednisolone, right? Next one, please. This is a size-depending nanodrug delivery system. If you reduce the size, the more the residence time will increase, the EPR effect will increase. Next one, please. This is a charge depending, means whether your delivery system is positively charged or negatively charged or neutral one, both charged particles will get absorbed from the colonic membrane, especially in case of the inflammatory bowel disease, where the mucus layer is thick. So the positively charged particles are adhered to the colonic membrane. Next one. And if it is negatively charged particle, then positively charged protein are overexpressed. In case of the inflammatory bowel disease, like eosinophil cationic protein or transferrin, so their uptake is increased. Right, so negatively charged particle uptake is more as compared to the positive charged particles. This is the pH dependent particles, as we know that the last part of the small intestine having the pH 6.5 or 6.4, right? So we select such type of polymers which will dissolve at this pH or more than this. So by the time it is getting to dissolve, delivery system will reach into the colon. So by taking the pH sensitive polymer, we can fabricate new drug delivery system. Next one, please. Biodegradable polymers, by taking these as we know that azodirectives and polysaccharides enzyme are released in the colon. So if we take azoaromatic polymer means in the skeleton of the polymer, there are number of azo bond, right? And this polymer will be degraded in the colon by the azoreductase enzyme. So your polymer become porous one and drug will get released. Or we can use only 2 minutes I will take to just conclude. And alginate, pectinate, or dextron. Hello. 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 Yes. Thank you very much. And after delivering, we have a lot of work on the colon drug delivery system. After designing or developing the colon specific drug delivery system, we have performed some in vitro studies, in vivo studies, and say if you just to ascertain that the drug will be released in the colon. Drug has to release in the colon only. It should not release drug in the upper part of the GIT. So we perform different studies by taking the simulated gastric fluid, simulated intestinal fluid, simulated colonic fluid. Especially when you perform studies in simulated colonic fluid, you are required to mix the cecal content, means colonic content you will require to mix, or you are required to induce the enzyme which is responsible for digesting that particular polymer. So that is being mixed in the colonic fluid, right? And we perform this in vitro release studies. Then we, after the administering drug through the oral route, we dissect the animal and isolate the different parts of the GIT, and we observe that how much drug is there unabsorbed, right? That is being released by the released by the colonic specific drug delivery system. Next one. Thank you. Thank you very much. So, okay, na?
Right. Thank you. Thank you, Dr. Professor Jain. So thank you for your important lecture on the colonic delivery, specifying various pH in which the drug to be delivered, use of various polymers, prodrugs, cyclodextrin, dendrimers, and other polymers which can be used, and use how the enzyme can degrade. And now I I'll open this for the questions that Dr. Jain will be able to answer your question. Thank you.
Thank you very much for such a informative presentation on colon-specific drug delivery system, sir. I have one quick question. When you have nanoparticles to be delivered at the colon site, is there any possibility of getting nanoparticles engulfed and absorbed instead of releasing the drug and release drug get absorbed? Because
From the upper part of the GIT.
upper part of the DJT, is there any possibility, any absorption?
Yes, if it is in nanoparticles, there are so many chances of absorption of the nanoparticle as such that will get absorbed from the upper part of GIT. That is why what we have, that it is not a simple, it was not simple nanoparticle. It was further coated with the pH sensitive polymer so that if it is not coated with the pH sensitive polymer, then we filled in the capsule, coated with the pH sensitive polymer so that drug will be released when the coating will dissolve at the distal part of the small intestine. By that time, this capsule will dissolve and drug will, nanoparticle will reach into the colon.
So much for a very good talk. Just a simple question I have is, you know that gut microbiota plays a major role, and it's actually, you have also shown that number of these bacteria have different enzymes which can metabolize drugs at different. Microbiota can influence, and especially the pharmacokinetics of the drug. You know,
say from the, as you know, the gut microbiota, especially in the colon, there is a number of microorganisms and number of enzymes are released of different category, and they are showing the different reactions, right? And naturally, they will affect the your drug molecules if it is released, right? But these enzymes we have used for the digesting our drug delivery system. Digesting the drug delivery system, say azoreductase. If we the drug delivery system, whether it is nanoparticles or microparticles or not beads or tablets or coated with the tablets, right? Coating of the tablets, right? So when it will reach into the colon, the microorganism, azoreductase enzyme will be released and that degrade the azo bonds that are present in the skeleton of the polymer, and you will get the porous polymer, right? And drug will get released. So these enzymes, most of the enzymes that are present, they are beneficial enzymes. They are beneficial bacteria. These are the beneficial bacteria for use for the digestion of the the content, food contents, right? Which is not digestive in the upper part of the GIT. These are used for digesting the food content. And these enzymes we use for digesting our drug delivery system.
And I am a student of B Form final year from SRIP BUJHANSI. And I have a question you for that. You were saying that colon targeted system is administered through oral route, but oral route so fast metabolic. So how can it is possible?
When will you get first pass metabolism? When drug will get absorbed, only then you will get the first pass metabolism, right? So, okay. So drug is not getting, our intention is not to get absorbed, drug should not get absorbed from the upper. If it is from the upper part of GIT, then it will go through the liver or like that first pass will be there. First pass metabolism will be there. But if it is absorbed from the colon, then we can bypass the this liver passes, right? First pass, bypass will get it.
डॉक्टर सर सर आप तो हमारे टीचर भी रहे हैं Actually, sir, in your presentation, there was one mention in slide that dendrimer also can be employed as a colon targeting in colon drug delivery. Sir, sir, please some highlight about it. How dendrimer can be used for?
Yes, dendimer as a colon-specific delivery system. As you know that dendimer is having number of amino groups, right? So in the skeleton of the dendimer, whether it is the first generation, 2 generation, or the 4, 5 generation, then in the skeleton of the dendimer, if you, instead of amino groups, if you put the azo bond, right? By using the, say, divinyl azobenzene as a cross-linker. So during the fabrication of the different generation of the dendimer, if you use this, if you put the azo bonds in the dendimers, then naturally it will be degraded, exclusively degraded, because it is having the azo bond. It will not get absorbed from the upper part of GIT. It will get absorbed, or it will cleave only when they will get the suitable enzyme like azoreductase enzyme to cleave that bond. So to destructure the dendimers, a drug will be released.
Thank you. Last question.
Yeah. Sir, I have two questions. So
Last question, 2 questions.
Last 2 questions.
Last 2 questions.
any difficulties with amino-containing compounds, diazotization reactions, if that amino group is present in the drug or in the excipient?
Amino groups present in where?
Drug or in excipients used.
If the say a drug or your excipient is having the amino groups, then it will be very good for fabricating the this colon specific using the carbamide chemistry, right? So it is good. We are interested in the amino groups. If the drug is having amino group or our polymers are having amino, that is good. Otherwise, what we are doing, we are producing amino groups in the API, either API or in the polymers.
Second question is,
Generally, we face problem when we incorporate some peptides, all right? उनमें आपस में conjugation हो जाता है, वो आपस में cross linking हो जाती है. So that is sometimes it becomes difficult one to protect so many amino groups, right? And only few groups can be used to convert into the azo bonds.
second question is for the sake of the students.
Pardon?
My second question is for the sake of the students. Can you shed light on the strategies to retain the delivery system in colon?
Retain what?
Delivery system in the colon. How can you retain this? Because
से वो क्या होता है कि presentation जो है वो बहुत fast का है. You might have escaped that slide, and you could not see it. The residence time of colon, you are not required to retain the delivery system in the colon. It will automatically retain there. The residence time of the colon is more than 20 hours, up to 48, 50 hours. Your content is there. मैंने बोला था, so that you can manipulate its release. है ना?
So, thank you, Dr. Sanjay Jain, and for your answers and use of modified polymers, linkers, ligands. What you wanted to focus that all for the delivery of drug in the colon, and look at how best it can be used. Thank you so much.
Thank you very much for such a profound and wonderful speech. I think all the participants who have heard this presentation, they will be definitely benefited from your presentation, sir. Now I request Professor Piyush Trivedi, sir, to present a memento and a certificate to speaker, sir. Sir, please. Now, I request Dr. Vijay Singh, organizing secretary of the conference, to please to present a memento and certificate to our chairperson, Professor Piyush Trivedi, sir. Thank you very much, sir. Thank you once again. Thank you very much, sir. Now, lunch is waiting for you all. Lunch is behind the auditorium, so this is time for lunch. Please have a lunch. We will resume after 1 hour, exactly after 1 hour from now. It's time for lunch, so please have a lunch. Sir, 1 hour is 1 hour, 1 hour. Forty-five minutes maximum. 45 minutes for lunch. बोल दिए for underground OD presentation, I request Dr. Man Singh to please
we studied at the National Institute on Drug Abuse. However, on the way to the forum, we discovered that they were profoundly hyperactive. So one of the main symptoms of ADHD. So here is the wild type animal. Here's the knockout. This is just a measure of how much they're moving around. And the knockout is profoundly hyperactive. And they show very slow habituation. Not only are they really active, they stay active for a lot longer than other animals. And then this was our initial interest. When you give them cocaine, they actually don't respond to it, which is a separate story I'm not talking about today. At about the same time, another laboratory in North Carolina also made a dopamine transporter knockout, and they got the exact same thing. And that's what's shown here from one of their papers, is they're really hyperactive. They then went on to investigate whether or not you could reverse that hyperactivity with drugs that treat ADHD. So they showed that. So you can reduce the hyperactivity here with methylphenidate, you can reduce it with amphetamine or other stimulants. They then tried to suggest that this was related to the effects of serotonin. A lot of these drugs that act as monoamine reuptake inhibitors in the brain, so the monoamines are dopamine, norepinephrine, and serotonin. A lot of them are very non-selective. They affect several different neurotransmitters. So that's a question from the beginning. Which one of those is more important? They actually suggested that it was serotonin based on this data, and this was published in Science. And the first time I saw it, I thought I would have rejected the paper if I was a reviewer. The reason for that was that their conclusion was wrong. One of the things I try to teach my students is when you look at data, look for things that don't make sense. Look for things that stick out. Look for things that are unusual. And in most of these graphs, there's a reduction in locomotor activity. But with the tryptophan, there's no activity. And I found that very suspicious. So when there's no activity, what does that mean? It could mean they're sedated. It could mean they're dead. I don't really know what that means because there's no other representation of the animal's behavior here. So I was suspicious of that, and we ended up investigating it. And what we found was that there were adaptations in the animals that probably accounted for this spurious finding. So, and it should have been obvious from the beginning because we knew very well drugs that increase serotonin function do not treat ADHD at all. So it was sort of ridiculous for them to go and assert that. But nobody bothered to question them on it. A few years later, we actually published this data that kind of showed that I was probably incorrect. And again, I'll sort of skip over some of this. So we then did a technique called microdialysis. Microdialysis allows us to remove a little bit of extracellular fluid from a part of the brain. We then put it into an HPLC and we measured the levels of different, well, whatever we want, usually neurotransmitters. And we did this, and again, our interest was cocaine, so I'm going to sort of skip over this. The part that was interesting here was this, is that in the prefrontal cortex, there's very little dopamine transporter in the prefrontal cortex. So this is a cortical projection that goes to the striatum. So that means that the function of dopamine in the striatum is profoundly affected, but it's not affected at all in the prefrontal cortex, and this can create an imbalance. And that imbalance has been described in a variety of what we call frontostriatal disorders. So they are disorders where there's impaired connectivity and function involving prefrontal projections to the striatum and then alterations in behavior. So we decided to use this technique, which I usually try to trick my students. So we set up this technique, and the interesting thing here is that if you have a sound that occurs from near the same source that is not startling, it will reduce the magnitude of the startle. This is something called sensory motor integration, and it's important in a variety of psychiatric disorders, including schizophrenia, mania, and ADHD. So just for time, I probably won't explain it too much more, but there's been some suggestion that it's impaired in ADHD. So this was our first good indication, aside from hyperactivity, that this was potentially an animal model of ADHD. So this is the magnitude of the startle. This is how loud the sound is. As the sound gets louder, it's a more effective stimulus that reduces startle. And this is our knockouts. So they are profoundly impaired at this. Now, the interesting thing is when we give them methylphenidate, they get better. When you give the wild types methylphenidate, they get worse. And this is referred to as the paradoxical stimulant effects in ADHD. Stimulant effects make people with ADHD better, but if you don't have ADHD, they actually make you worse. So again, this seems to parallel in a couple different ways. Now, I suggested before that that other laboratory said that serotonin was involved in these effects. We actually discovered that norepinephrine was involved in these effects. So again, here you give nisoxetine, which is a norepinephrine, selective norepinephrine transporter blocker, and it restores their behavior. Has no effect in the wild type animals. Try and go a little bit faster. So then we next showed by direct brain injections that the prefrontal cortex is where the drug was acting. So again, in these animals, they don't have dopamine transporters at all. So this is not acting at the dopamine transporter, it's acting at the norepinephrine transporter. So then we did some immunohistochemistry to look at activation of CFOS. There are a few interesting things here. But the key thing that we showed by looking with double and triple labeling is that it was these drugs were activating neurons in the prefrontal cortex that projected to the nucleus accumbens, and they were glutamatergic neurons. So, and that's the triple labeling. We then started to look at the neurons in the prefrontal cortex, and they were abnormal in a variety of ways. So they have reduced spine density, that is, reduced inputs that are regulating them. They had lower levels of BDNF, and a few other things. Now, this is one of my favorite tasks. It is a very simple task. It involves a circular platform. And this, I thought, started to get at something more fundamental for ADHD. So this was what I called risk assessment. Amit, how many for two? Okay, so I'm going to speed up a little bit. So this is one of my favorite tasks. And it's an interesting one in a variety of ways. You put an animal on this platform and you can see the wild type animal barely goes over to the edge. They are very nervous. They are prey animals. They don't like being exposed out in the open in a brightly lit environment. They really don't like that. Now, look at the DAT knockout. First of all, its posture is completely different. It's not pushing itself into the floor to try to disappear. It is just sauntering over to the side, not too bothered, sticks its nose over the edge, and then eventually thinks it's Spider-Man and tries to climb underneath the platform and falls off. Wild type animals never ever do that. So again, and this is what happens. And the interesting thing here too, and this, I had a long argument with the reviewers back and forth about this paper, because they claimed that this was an impulsive behavior. And I said, it wasn't. And we gave in and used the word impulsivity in the paper to get it published. I didn't, my collaborator did, who was doing this. I love him, but that was a mistake. But it can't be impulsivity, because it's not that you put them on the platform and they immediately wander over and fall off. They think about it. This is time. They're falling off after 30 or 40 minutes. They understand what's going on. They think about it, and they still do something dumb. And this is very much like those ADHD kids that I was describing. It's not just that they're impulsive, it's that they have impaired risk assessment. They do intrinsically dangerous things. Now, again, another interesting thing here is we give them methylphenidate and it fixes it. So here's another drug showing the same thing at a drug that also treats ADHD. And this is a little model that we created of what we think is going on here. This is this interplay between dopamine and norepinephrine neurons, where there really isn't any dopamine transporter here, even in the wild type animals, but there are these net transporters on nearby neurons, and this is where our drugs are acting, we think. We did some additional pharmacology showing that this function was important too. So again, I'm going to sort of skip through. Oh, okay. I left that data out somewhere. So other drugs that also treat some ADHD symptoms like nicotine also work in this model. So we really wanted to get at impulsivity a little bit more, and particularly because I didn't think the CAR test looked at impulsivity. So we used this task called the 5-choice cell reaction time task. And we actually went to a more complex version, which is called the continuous performance test. And basically, in this box, there are lights. The animal has to watch. One of the lights will come on. It responds correctly. It gets food, or actually strawberry milkshake. They love strawberry milkshake, so we use that instead. But it also has to wait until the light goes on. So there are a variety of types of errors the animal can make. It can go before the lights turn on. It can respond inappropriately. So there are different types of errors. Some of them reflect impairments of attention, some of impulsivity. So here, I'll just skip over here. We didn't really see any attentional effects in this. What we really saw was this, is that they would emit a lot of premature responses. So here is amphetamine. So amphetamine is an Adderall, a standard drug used to treat ADHD, and it reverses the impairments. Now, embedded in the diagram that I showed you a little while ago was the position of the 5-HT1B receptors. We felt that they were in a position to regulate this function. So we used a 5-HT1B antagonist, and it also reversed the deficits as well. We next did something called the Iowa gambling task. So in this task, the animal has choices. On every trial, it can potentially be rewarded, again, with a strawberry milkshake. But some of the choices are more risky than others. But embedded in the task is this other thing where you have premature responses. And, oh, okay. All right, where was that? So here again, are impairments and premature responses in this model. So again, this is a second task where we're showing potential impulsivity. So since I'm running out of time, so the DAT-KO mice appear to be a very useful model of ADHD. They show what we call face validity. They show all of the symptoms of ADHD, hyperactivity, impaired impulse control, attention deficit, and impaired executive function. One of the things I didn't mention is that the minus complete knockouts, we couldn't test in that last task because they were so impulsive, their behavior was so disrupted that it just didn't work. So we were using heterozygous mice. But I didn't show you all of the data here, but we actually tested those 5-HT1B antagonists in about 4 of our tasks, and they reversed the impairments in 4 of the tasks. So the 5-HT1B receptor antagonism potentially represents a treatment avenue for ADHD. However, one of the things in terms of the next stages I want to do with this project is like several people here, I need a chemist to work on the project. because the original drugs that were developed are not selective enough for the 1B receptor, and their pharmacokinetics isn't optimal. So there could be some chemistry work that would really improve this project as well. All right, most of the work here was for my graduate student, Yasir Saber, who got his PhD not too long ago. The work was recently started up again by Sarah Beecroft, who's actually a PharmD student, but she's doing research in my laboratory at the moment. And these are my long-term collaborators from NIDA that I started with this. There's a very long list of people who contributed to a lot of the older data, but Yasir was responsible for most of the new data. So anyway, thank you very much, and I'll take questions.
Thank you. Thank you very much, Professor Scott, for an awesome presentation. We got time for only a couple of questions.
Thank you so much for an awesome presentation. My question is this 5-HT1B receptor antagonist, and you mentioned it's not very specific.
Sorry?
Is it very specific inhibitors that
Oh, the inhibitor?
Yeah.
So the question was the SB compound that we used that was the 5-HT1B antagonist. Is it specific? That's an interesting question. When a lot of drugs were originally made, they were considered to be specific. But the specificity at certain points was, well, they're 10-fold selective. Amit would be appalled by a 10-fold selectivity. That can be useful sometimes when you're using it as a tool, but not very useful perhaps when you're bringing something to clinic. So, and the other thing that's happened in terms of selectivity is that over time we've discovered a lot more targets. So it was selective against the original serotonin receptors we knew about in about 1992, but then more of them were developed. And most of the drug development for this target for 5-HT1B antagonists occurred a long time ago. So what we really need, I think, is a new generation that is more specific and has better PK properties for being a drug.
Yeah, so it was a follow-up question to that one, and you answered, some is this the only target that you would target, or there are other targets that you would target?
Well, no, there are potentially other targets too. So actually, I'll skip back quickly. Like everybody else, I prepare far too many slides for 30 minutes. So this is our kind of circuit diagram. And the 5-HT1B receptors are in a couple different positions to modulate this descending circuit that we think is abnormal in the prefrontal cortex. But there are other 5-HT1B receptors and other cholinergic receptors that might be useful as well. In particular, alpha-7 receptors. And for a while, people had been developing alpha-7 receptor agonists for the treatment of schizophrenia. Because one of the things that you see in schizophrenics is they all smoke. And there have been a variety of explanations for this, but part of it is that it's actually therapeutic. And you see the same thing in ADHD. So in ADHD, people who have ADHD smoke at twice the rates of the general population, at least in the United States. Smoking rates are much higher in India, so that's not quite as clear here. But there are a couple potential targets in that circuit that we could go after.
Time for one more question. Any questions? If not, we conclude seminar of Professor Scott. Thank you very much.
Thank you very much, sir, for your magnificent lecture talk. I am very sure that all the participants will be benefited. Now, I invite Professor J.B. Ramapuram, sir, to please come on stage to present a certificate and a memento to Professor F. Scott Hall. Thank you very much, sir. Now I request to Dr. Raghuveer Sayya, sir, to please come on stage to present a certificate and memento to Professor J.V. Ramaprom, sir. Thank you very much, sir. Our next eminent speaker is Professor Pia Bogal, madam. So before inviting him, I request Ms. Shruti to come on stage and please read the citation.
Thank you, sir. A biochemist and leading authority in electron spin resonance spectroscopy, Professor Pia Vogel's research interest include structure, molecular dynamics, and protein-protein interactions of nucleotide binding proteins. At SMU, she is the founding director of the Center for Drug Discovery, Design, and Delivery. Dr. Vogel has over 40 peer-reviewed publications in leading journals and was recently issued a patent on molecules that reverse multi-drug resistance in cancer. She has also contributed to 4 textbooks in the area of biochemistry and biophysics, as well as 1 in nanotechnology. She has received $1.5 million in grants to support her research, including major grants from the NSF and NIH. She also holds an honorary degree from the Shanghai University in Taiwan, China. Thank you.
Thank you, ma'am. Now I would now request to Professor Pia Bogal, ma'am, to please come on stage to present her presentation. So for this, we have amongst us Professor S.K. Jain, sir, who will chair the session. Professor S.K. Jain, sir. Ma'am.
So, I invite Professor Pia Vogel for her deliberation. I'm sure that our participant delegates will be benefited by her deliberations. Without wasting much time, I request Professor Pia to deliver her lecture. Okay, thank you very much.
Thank you very much for the warm welcome. I think India is the only place where you can get a bouquet of flowers before you even get your work done. So thank you very much. I also want to thank the organizing committee on all the hard work, on getting me here, and congratulate you on all the hard work in getting a conference of this magnitude organized. It's been a really pleasure to be here. So why am I even here? I'm not a pharmacologist, I'm not a pharmacist, I'm not a toxicologist, I'm a simple protein biochemist. So I'm going to take you way down to the molecular level of, how things happen. But the reason I think I can belong here is because I'm dealing with a medically pretty relevant protein. So what we're talking about is a protein that causes chemotherapy resistance in cancers. Okay, so let me see if I can turn that around a bit. So especially metastatic and recurring cancers, to a huge degree, find ways on how they can just evade the chemotherapeutic toxic effects. And there's a lot of different reasons, and tomorrow you're going to hear from Dr. Tiwari, I think, of other ways on how cells can do that. But since I'm a protein biochemist, I'm specializing on one protein. So one of the ways that chemotherapeutics can, or that, cancer cells can become resistant to chemotherapeutics is that they just simply, make more of a protein that uses ATP, the cellular energy currency, to export the chemotherapeutics out of the cells. So as soon as they're transported out, and if there's a lot of this made, there's no therapeutic concentration of chemotherapeutic left in the cells, and the cancer cells just simply don't die. Those are actually really important membrane proteins. They're not just there because they pump out chemotherapeutics. They're there because they keep our cells free of toxins. They have a very broad substrate. Yeah, that's a weird word. Substrate specificity is a broad spectrum of chemically completely unrelated molecules, toxins that they pump out of cells. And so you find them in the membranes of many of the important tissues like breast, testes, ovaries, and so on. And they also are an important component of the blood-brain barrier. And as I said, they need ATP to cause a conformational change, so a really huge change in its shape so that they can accept the toxins on the inside of the cell and then release them to the outside of the cell. So because of that, and because they have evolved to export so many different toxins, they create what is called multi-drug resistance. It's not just the resistance of a cancer cell against one chemotherapeutic, it's against just about all of them. And it makes it real hard. It has been really hard to design pump inhibitors. So I'll show you the next slide, but I'll state it here. We've been knowing for more than 40 years that these pumps are associated with chemotherapy-resistant cancers, but still there is no medication in the clinics. The way that initial studies were done to try and find inhibitors was to have a 96 well plate or any such kind of assay, put a multi-drug resistant cancer cell line on there, give them chemotherapeutic, give them other small molecules, and just see which of the cells are going to survive. Okay, so the ones where the chemotherapy resistance was overcome meant we had an inhibitor. But what they did not show, those experiments, was what mechanism of inhibition was actually used for that. Was it that the pumping was somehow blocked, or was it that the inhibitor competed for pump cycles? If that happens, then of course the chemotherapeutic is going to accumulate in the cells. Or is it that somehow ATP energy utilization was changed? So because, we have this really broad spectrum of compounds that can bind in the drug binding areas, it had made it really hard to block that part, that mechanism of inhibition. So we decided that maybe we can block the energy harvesting from ATP and make inhibitors for the pump like that. Well, if you want to find an inhibitor, if you want to design an inhibitor, you need to know what the structure of the protein is all about if you don't want to go in blind. So at the time we had, let's call them cousins of that, one of those pumps in form of X-ray structural models available. None of them is human, okay? The closest one is the eukaryotic one, that's the mouse, and all the other ones are bacterial. But what you could see from those structural models is that the proteins that are related have very strong differences in their shape, okay? So here, Up top, those globular domains, those are the nucleotide binding domains. That's where ATP binds and gets hydrolyzed. And down here, this is in the membrane. So if you turn this by 90 degrees, you see that it's from all sides. This is completely close to the outside of the cell. Okay, so this is a position where some toxin can bind from the inside of the cell, but it's close to the outside. Now, if you go further here in the transformation, you can find a final structure where the nucleotide binding domains are very closely interacting. And this is actually the state where ATP can be hydrolyzed. So that's where energy can be harvested. And if you now turn that structure, you see that now the drug binding domain is completely open to the outside. So we thought, okay, so maybe what we have here is a catalytic cycle. Maybe this is what happens also in the cell. So what did we do? We did not have a structural model for the human protein, but we had this known structure. We took the one that had the highest resolution of the bacterial cousin. And we did have the amino acid sequence of the human pump. So, well, we fit the human protein sequence onto the bacterial structure, and we got a very nice structural model. I want to emphasize that. This is all just models, okay? This is not at this point just what the computer shows us, and we have to be careful not to believe too much of what the computers show us. We have to always make sure that we can support those. So we had, as a result, however, a nice model on how we could potentially start and design inhibitors that could block ATP binding. We also, I'm not going to go into that in great detail. We also were able to, through computational ways, to mimic those transitions between the different states. And we can dock through in silico docking techniques, transport substrates that are known to be transport substrates. And then we can see, so okay, and that actually worked. We have a lot of biochemical work done on that protein. So we know that different transport substrates bind at different parts of the drug binding domain. It also shows you how big it is, right? You can actually bind both of them at the same time, and that's no problem whatsoever. That thing can really change its shape down here a lot. And then we can just see how when you run the protein through those structural transitions, how the substrate gets extruded down to the bottom. It's kind of squeezed out. So we were really happy to see that, right? That made it look as if we really know what the enzyme is doing and how it is doing it. So, as I said, most people for like 40 plus years have been looking to find inhibitors. And because it has been so unsuccessful, more people have been starting to call it an undruggable target because, just nothing was done. But we think that most of the inhibitors that were initially found were actually transport competitors. So they would compete for transport cycles, and with that keep the chemotherapeutic in there. So as I said, our hypothesis was if we can block the ATPase motor, and then we can find maybe small molecules that can tightly bind to the ATP binding domains. And then we could screen out molecules that also interact with the drug binding domain. And so that may get us to an inhibitor that now can really inhibit the pump, not just have a competition happening. And maybe that's a way to make a better clinical product in the future. So we did some huge database searches. Okay, we used like 5 million plus. In the meantime, it's like 3 times 5 million different drug-like compounds that you can find on databases. And we dock them with docking, in silico docking techniques. Those programs are available, and try to find small molecules that were, well, supposedly binding to the nucleotide binding domain. Then we redock those to the drug binding domain, and we subtracted all those small molecules that also interacted down here. So what we wanted was inhibitors that only knock out ATP hydrolysis, but don't get pumped out themselves. Okay, so we wanted to make sure that we get them specifically there and have, with that, probably only a need for a lower amount of therapeutic. So in the initial round, that's the 5 million, we got about 180,000 hits for strong interaction with the ATPase motor part. And then when we subtracted all of those that also interacted with the drug binding site, only 300 were left. So you can see how broadly, you know, the molecule shapes can be that still interact down there. You know, we screened almost, well, 170 something thousand out. So again, we still have to see whether whatever we find in the computer actually is true also in the lab. So the drugs, some of the small molecules that were identified computationally could be purchased, and we assayed them in the lab. Being a protein biochemist, I started out with a protein. Now, this is actually the hardest part of the research because this is a human membrane protein, and it is darn hard to get human membrane proteins isolated from the membrane so that they're still, and have them still active. But what we wanted to see was, you know, is our hypothesis correct? Do we have inhibition of the ATP hydrolysis activity in the presence of those identified compounds? And indeed, we found some that we concentrated on. So those are the initial 4 that we have, and those have been the earliest patented ones. And if you look at them with a chemist's eye, you can see that they're structurally pretty different. From the docking, we had assumed that the 3 of them bind at least close to the nucleotide binding sites. So those are 34, 49, and 19. And 1 we chose to look at because it's at an allosteric site, so somewhere far away from the nucleotide binding sites. And those make usually the best inhibitors, because if you get too close to the ATPase domains, you can have a lot of side effect because a lot of enzymes use ATP. So yeah, we used those 4 compounds in ATP activity assays, and just looked at concentration dependence, and yes, all of them inhibit ATP. This is all done with a purified enzyme that we have purified from an engineered yeast strain that overexpresses the human protein. That's a long process, and yeah. Also, I have this different technique, the electron spin resonance spectroscopy, and I'm not going to go into it at all, but I can synthesize an ATP analog that can help me measure how much of ATP is being bound to the protein. So we could see that of the 4 compounds, 3 actually inhibited binding of at least 1 of the ATPs. So it's got 2 ATP binding sites, and 1 was knocked out or maybe occupied. And 29 did not seem to inhibit ATP binding. And if you remember, 29 is the one that's allosteric, so that's not anywhere close to the nucleotide binding sites, while all the others were hypothesized to be close to the nucleotide binding sites. So that all made us real happy, because it seemed that the predictive power of that method was pretty good. But then we had to go in and see whether or not we have an effect also on cells. Okay, can those compounds get into the cells, and do they do something in a more in vivo, but even though it's still in vitro environment? For that purpose, we had 2 different cancer cell lines. Those are prostate cancer cell lines. The open circles are, they correspond to a newly diagnosed prostate cancer. So it still is pretty sensitive. This is a logarithmic scale of a chemotherapeutic. This is viability of the cells. So they're, they still die at relatively low concentrations of chemotherapeutic. And then we have this derived cell line that mimics chemotherapy-resistant cancer. And for this one, you need 1,000-fold more chemotherapeutic to kill it. So a patient could not survive that. I mean, there's no way. The chemotherapeutic is way too toxic here. So you couldn't kill the cancer without killing the patient. But then when we added our compounds to it, 3 of the 4 actually were able, if they were added to this highly resistant cancer cell line, they brought the sensitivity back to the naive, to the early cancer cell line that we started with. Except for one, that was compound 19, and we found out later that actually it has a couple hydroxyls with a pK that would probably make that compound negatively charged, and so it probably never entered the cell. So that's something where we can still play around with if we had chemists available. Okay, and we did a lot of, and I'm going to just go very quickly, and I'm going to skip the other project completely. We could show through some different assays that indeed in the presence of our compound, this is specifically on compound 29 now, all that I'm going to say, that a transport substrate actually accumulates in the cell in the presence of the inhibitor while everything stays dark. It gets pumped out right away without the inhibitor. We could show that on cancer spheroids as well. So again, in the presence of our compound, we get the transport substrate accumulated in the tumor. And we could also show that the tumor, those microtumors in the presence of chemotherapeutic does not die, while in the presence of chemotherapeutic and our compound, it dies. We did a lot of other assays. In this assay, we show that there is no cell migration in the presence of our inhibitors. In this one, we show that if we have, those are colony formation assays, so in 2 different ways here. So in the presence of chemotherapeutics alone, so these are 2 different types of chemotherapeutics, paclitaxel and vinblastine, the cells are happy and survive. Here the purple means that they're viable. In the presence of the inhibitor alone, they're viable, but in combination with our inhibitors, all cells are dead. And that worked for all the inhibitors except for 19, which didn't get into the cells. We could also show, and I'm not going to get into that, that actually those are not transport substrate, the 4. That's exactly what we wanted. We wanted some that only inhibited ATP hydrolysis, but did not, but were not transported themselves. And this is the last slide that I want to go into in more detail, because I think this can have some really huge effects. So in this experiment, it's a long-term exposure experiment. We have those little spheroids, cancer spheroids, in the presence of DMSO. They're happy and alive. And then we incubate them for a total of 72 hours, either in the presence of our inhibitor alone or in the presence of the paclitaxel, the chemotherapeutic alone. And you see, well, there is a little bit of cell death happening in the presence of 29 of inhibitor, but absence of chemotherapeutic, or the other way around. But the cells are all dead in the combination assay. So that's what we expected. What's really cool, though, is that we could, just do the experiment where we just incubated with a chemotherapeutic for 3 hours. Well, 3 hours don't kill the cancer cell. 3 hours incubated with a combination of paclitaxel and inhibitor also is not enough time to kill the cancer cells. But if you incubate with the chemotherapeutic and the 29, the inhibitor, for 3 hours, just like in this experiment, then wash away the chemotherapeutic and just leave in the inhibitor, and then just continue it to follow up to get to the 72 hours that we had in the previous experiment, all the cancer cells had died. So in other words, all the time that was needed was a very short exposure to chemotherapeutic and inhibitor together, and then the chemotherapeutic could be taken away, and the inhibitor was still able to continue to lock in the chemotherapeutic in the cancer cells and get the cells to die. So a lot of other things we could show that it's not cancer specific. We do it in other cancer cell lines, so all of that has worked out beautifully. We tried a little bit on doing some medicinal chemistry computationally. I'm not going to get into it, but we identified a few variants of that compound 29 and ran them again through the same subtractive docking as before, and came out with a few hits that were somewhat better in binding, and they did what we wanted them to do. They were not transport substrate. They inhibited ATPase. They did not bind to the nucleotide binding domain like the parental compound. And as compared to another one where we did not do the computer-assisted selecting, but just try to look at the binding sites by eye and try to figure out if that works, we did not get quite what we wanted. So, but those are not good enough yet, and that's, again, where we need a medicinal chemist. Those are too big, even those up here that do what we want. They're too big, they're too hydrophobic, they would be really hard to really develop further on as drugs. So we need help in getting some medicinal chemistry, somebody who can look at it and say, well, you need this group to make it work. We're also looking at the second resistance pump, and I'm not going to go into it, but we want to know if that breast cancer resistance protein, which is also involved in chemotherapy resistance, if we can find inhibitors for that. We have a whole screen of it. We do the same thing as before. Do we get accumulation of a transport substrate in the presence of the inhibitor? And yes, we have a lot of hits there. We used it at different cell lines, and we're currently preparing cell lines. That's a breast cancer model, cell line. So we're currently making different types of cell lines that express different levels of the pump, so that we can see how resistant they are actually and how the resistance can correlate to the expression level of the pump, and maybe get to a system that we have cell lines that we can then use in xenograft models to see if we can get into in vivo. Okay, and those three are new kids in town. This is my current graduate student who's done was responsible for most of the breast cancer work, together with Ria, an undergraduate researcher. Simran just joined, and those two are graduate students who just joined the lab this spring. And well, just thanks to my collaborators and to my funding sources, I've been really fortunate to have a lot of private donors, because as I said, the NIH doesn't think we have a druggable target here, and it makes it hard to get money that way. But so far, I've been able to keep it all running. So just at the very end, thinking about the agreement that was reached this morning with this university and the University of Toledo. SMU is a beautiful campus. We have quite a few graduate students in our science programs. We're always looking for more, and I hope we can have some kind of an agreement that maybe have some of the students from here come over to us or the other way around. And that's where I'm going to leave it. So thank you. I think I'm pretty much in time, right?
Professor Pia has finished well in time. Only say 2-3 questions I invite from the audience. If there is any questions. Okay, from students.
One question you should.
Please.
Madam, actually, we don't know in depthly about your presentation, but I want to know about some one information. What is a generic drug binding that generate the NDR? Why?
Say that again, but why is
In your slide, there was mentioned generic drug binding site create MDR, multi-drug resistance. So why, madam? What is the reason?
Okay, so these pumps have evolved from bacterial precursors, right? And it's the same mechanism, except the bacterial proteins, they use that mechanism to import substrates or whatever, or nutrients. The eukaryotic ones have evolved to keep our cells clean, to export all kinds of toxins. So they are very plastic. They can make the drug binding domains, they can make very large conformational changes. I was just saying this morning, this pumps out A beta, the Alzheimer protein. So it pumps out really large substrates. It pumps out cyclosporin. It gets choked up a bit, so it may need a couple cycles, but it could just take very different kinds of molecules and pump them out, because it can just adapt to different shapes of transport substrate.
Okay, thank you.
Thank you, Professor Pia, for an excellent presentation. I have one question regarding the binding mechanism of the compound to the ATPase allosteric site. Can you explain what exactly happens through in silico studies? Is it stabilizing the transporter in a particular conformation wherein the ATP is not able to leave?
Yeah, this is a crazy protein, okay, just to say that out front. So in order for ATP to be hydrolyzed, the two ATP binding domains have to come really close. And in fact, the aspartate that activates the water for hydrolysis from this nucleotide binding domain has to interact with this nucleotide in order to have hydrolysis happen. So the protein has to get into just the right structure. And my hypothesis is that 29 just sits in there as like a monkey wrench, right? And it doesn't let that conformational change happen. And so ATP doesn't get hydrolyzed and it gets stuck in that position.
My other question is, why compound 29?
Well, we have a lot of other ones, okay? This was the first one. This is the one I can show you the structures. And it actually was such a nice story. But we have a lot of other ones that we could do. And we also have some evidence that the other 3 of the initial hits do not interact with other ATP hydrolyzing enzymes or ATP utilizing enzymes. So those may be just as great a lead to go on with. We just have, with limited resources and limited hands, you can only do so much.
Yeah, compound 29 was literally too big for a lead molecule. Because
No, 29 itself is not too big. 29 has around 300 or something, and it also, the hydrophobicity is okay. But the variants that we made, and we try to get variants that have higher affinity, because the affinity is also not good enough here. We're in micromolar range. We want to get it into nanomolar range. So to make the affinity higher, what happened was that hydrophobic portions were added to it. And you can look, if you look at the binding pocket, there is a hydrophobic pocket there. And I think that's what made them better binders, but made them worse drug candidates.
Last one, did you test your compound on normal cell lines?
Test it, test.
Test the compound on normal cell lines, what effect it has?
Effect on normal cell lines.
Yeah, that was what I was going to say. That's the question that should have been asked. Yes, no, they're not toxic to other cell lines. And what's interesting too is that they also do not increase toxicity of the chemotherapeutics to normal cell lines. That was another worry, right? But I think that the expression rate of those pumps in the normal cells is not high enough so that the inhibitors are going to make a big difference. So.
Thank you very much.
Yeah, Tom.
Any other queries or question? Karthikeyan, please.
Good evening, ma'am. Ma'am, when we perform in silico studies, actually there are some hydrogen bondings which usually occurs between the protein and the small molecules. But there are some times when we perform molecular dynamics of that protein structure, the protein leaves that particular amino acids and they show bindings in another amino acids. So is that results are acceptable when we perform such type of studies?
The way I see those docking studies are just as hypothesis generators, okay? I don't believe the docking. It looks as if it looks good. And so it takes the huge spectrum of potential candidates down to a smaller spectrum that we can kind of handle in the lab. And of course, we're going to miss a lot of potential good ones, but I just don't see a way on how else we can, at least for me, for my money, I don't know how we could else just screen them down to a small feasible amount. But I never ever just believe what the computer tells me, what the drugging tells me. It always has to go into the wet lab and needs to be tested.
So, well, we are running short of time. You have one more query? Okay, last one.
Yes, sir, last one. Madam, why the people are increasing the time of performing the molecular dynamics? Nowadays, why the people are increasing the time to perform molecular dynamics? Previously, the people are performing picosecond, the results are accepted. Now the people are asking to go for 100, 200 nanoseconds.
The proteins are very dynamic. So the longer you can watch it, the better I think you can really estimate how well the interaction actually is, right? You don't just look at a snapshot. You look at whatever is happening in the meantime. You have, even with the nucleotide binding domains, you have a lot of dynamics happening. And so I think that's why a longer time watching what is happening and getting all the different delta G values and averaging those out is going to give you a better, a better hypothesis, maybe. Okay.
So, thank you, Professor Pia, for such a nice informative lecture. It is really
for our participants and delegates who have come across the India. So now I would like to invite Professor S.K. Jansa to please to present a certificate and memento to Madam Pia Vogel. Moving forward, I would like to invite Professor Sunil Prajapati sir to please come on stage and to please to present a certificate and memento to Professor S.K. Jain sir. Thank you very much, sir. Of course, we are very delighted to have such a wonderful dignitaries amongst us. Now, remaining expert talks will be conducted tomorrow morning session. And our next event is oral presentation for PhD scholar. We will resume here after 15 minutes. We have a little short break. Kusma is here. So I invite Pratibha to please come on stage and for her presentation. And the session will be chaired by Dr. Vivek Tomar sir, who is Associate Professor in SR College, Jhansi. He is the Director in SR College, Jhansi. Come on stage, Pratibha. And
A very good evening, everyone. So it brings me a delight to be here and present my part of PhD work because I am the alumni of this university.
because of time constraint, please complete your presentation within 5 minutes, and 1 or 2 minutes will be for questioning.
So I will be talking about antifibrillation effects of ionic liquids, and I will be trying to show that there is a role of hydrophobicity in inhibiting the fibril formation. So as a protein is synthesized, it forms the native conformation through various folding intermediates. And these conformations are maintained by interactions such as covalent and non-covalent. That may be disulfide bonds, hydrogen bonds, ionic interactions, and hydrophobic interactions. Any perturbations in these physicochemical conditions of the protein, such as pH, pressure, temperature, and co-solvent conditions, may lead to partially unfolded states of the protein that may expose the hydrophobic side chains of the protein. These partially unfolded states of the protein may form oligomers and which may further form amorphous aggregates and also amyloid fibrils. If we arrange these species into the energy landscape diagram, we obtain a rugged funnel. And to understand what is the role of hydrophobicity, we have chosen a model protein, which is lysozyme, and it has 6 hydrophobic clusters. We have chosen this protein basically because cluster number 5 and 3 perform the long-range interactions to form fibrils. And there's also a loop region that connects the alpha and beta domain of the protein, which is highly amyloidogenic region. So we have chosen the co-solvents. Which have different functional groups with varying hydrophobicity and are highly water soluble. These are ionic liquids such as salts containing cations such as imidazolium, pyridinium, and anions such as halogens, trifluoride, and tetrafluoroborate. They have multiple applications in various fields as shown here. And we have chosen 9 ionic liquids with imidazolium head group and varying alkyl side chains that shows that they have varying degrees of hydrophobicity. And 4 more ionic liquids which have alicyclic head groups as shown here in first 2 structures, and 2 aromatic head groups in another 2 ionic liquids. And next we have seen the mixed ionic liquid effects on the fibrillation patterns. So these are the combinations we have used. This is the first combination where we have taken a shorter alkyl chain with longer alkyl chains, and also with aromatic and alicyclic combination. Another combination we have chosen here, and we have taken the ratio of these ionic liquids in this way. So next we have checked for fibrillation effects, and we could understand that there are 2 types of mechanism taking place in the fibrillation process, which is lag independent, where monomer proteins directly bind to form elongated structures such as fibrils, and in another case it is lag dependent kinetics, where primary nucleus is forming from monomers, and this primary nucleus again aggregates to form a elongated fibril. So here are the results. We can see here in the left side graph, we can see that with the increasing concentration, there is a decreasing elongation rate, and with at the higher concentrations, there is a lag-dependent kinetics observed. And same is observed in the case of aromatic as well as alicyclic ionic liquids. When we mixed the 2 ionic liquids at a time and checked the fibrillation effects, we could see the similar effects here as well. Then we checked for the morphology of fibrils, and we found that various fibrils that were branched in nature were clumped together to form blob-like structures. This is the diagram for lysozyme fibril in buffer, and when we added ionic liquids to the protein, it was also forming the similar morphology. There was no difference in the morphology when it was either lag independent or lag dependent kinetics. Next, we checked for the aromatic and alicyclic ionic liquids, and we found that in case of pyrrolidinium, piperidinium, and pyridinium, there was long unbranched ionic liquid, several micrometers long. But in the case of imidazolium bromide, we found that it was similar to lysozyme fibril in buffer. While in mixed co-solvents, we found that fibril forms formed were long, unbranched, and several micrometers long. Then we checked for the apparent fibrillation time, which is what we have understood as total time taken for a native protein to form an elongated fibrillar structure. And we found that with the increasing concentration of protein, fibrillation time was enormously increased. And in case of longest alkyl chain, the fibril formation was completely inhibited, 500 millimolar concentration. And in case of mixed ionic liquid experiments, we observed that the fibril formation was enormously delayed. Then we performed the molecular dynamic simulations to understand the molecular level interactions, and we used lysozyme in a box, and we used this is the protein PDB ID we used, and we used the Gromex suit to perform our simulations. We used OPLS force fields and TIP3P water model, and all the simulations were performed in the presence and absence of ionic liquids. Ionic liquids were taken in 50 and 200 millimolar concentrations, and mixed ionic simulations were performed in 1:1 millimolar, 1:1 ratio in 100 millimolar concentration of each ionic liquid. We took the protein in a box of a certain size surrounded by the ionic liquid molecules and solvated by water molecules, where we energy minimized it and kept the protein for 1 nanosecond at 1 atmospheric pressure. And then we again incubated the protein at temperature 300 Kelvin, that is room temperature, and we performed the simulation for 100 nanoseconds in the case of individual ionic liquids, and in case of mixed ionic liquids, we performed the simulations for 200 nanoseconds, while analysis was done for last 80 nanoseconds, and in mixed ionic liquids, we did 150 nanoseconds analysis. So we counted the number of water molecules and ionic liquids around the protein in the case of 200 millimolar concentration of simulations, and that was done in the case that it was done in the cutoff distance of 0.62 nanometer. And we could see that number of water molecules were enormously decreased as the alkyl chain length was increased. And in case of aromatic and alicyclic, we found that number of water molecules were decreased in the case of aromatic as compared to alicyclic ones. And we can see here, as you can see here, this in the other side, that ionic liquid numbers were also increased in the case of longer chain and alkylic ionic liquids and aromatic ones, as you can see in brown one and red one. Then we also checked for the hydration fractions, that is determined to know the distribution of water and ionic liquids around the protein. That was calculated by the ratio between fraction of water molecules and ionic liquids averaged for the analyzed frames. And we could see that with increasing alkyl chain, the hydration fractions were decreased, and also in the case of aromatic ionic liquids, the fractions were reduced. So next we checked that how is the residue-wise interaction of the protein with these ionic liquids. So here is a short video. We can see with increasing alkyl chain, the number of interaction sites are more and more. This green and pink color shows the more interaction sites, and the number of residues are also shown alongside. Next, we checked for these aromatic and alicyclic ionic liquids, and we can see that aromatic ionic liquid shows the more interaction sites on the protein. So overall, we identified four interaction sites on the protein for these four ionic liquids. Then we checked for the number of ionic liquids and water molecules, and in all the cases, we found that longer alkyl chain ionic liquids reduced more water molecules around the protein. And here also we can see hexyl alkyl chain could has more numbers around the protein. So here in all the cases, we have checked the similar results. And then we checked that if any role of aromatic and alicyclic is there, and we keep the alkyl chain similar in both the cases. And we found that when aromatic and alicyclic ionic liquids are taken together, they both occupy the similar binding sites and compete equally with the binding sites on the protein. And hydration fraction also shows the similar results over here. So interaction sites, residue-wise interaction sites on the protein, we can see here that when both are together, MIC occupies more interaction sites on the protein, while in case of HMIC and MIC, HMIC occupies more regions because it is more probable to bind to the protein. While in case of BMIC and pyrrolidinium, there is equal binding sites on the protein. So overall summary of the experiments is octyl, hexyl, and pyrrolidinium shows maximum delay in fibrillation at higher concentrations of protein. And in octyl methylimidazolium chloride, it completely inhibits the fibrillation at 500 millimolar concentrations. There is more number of water molecules that were removed in the vicinity of the protein with the addition of longer alkyl chain and aromatic ionic liquids. Either they were present individually or in combinations. While ionic liquids with longer alkyl chain and aromatic ionic liquids compete with alicyclic and lower alkyl chain ionic liquids to interact with protein. So this is the institute where I had been working. And this is my lab group. He is my PhD supervisor.
Thank you, Pridva. If you have any questions from audience side, please, you may ask only 1 or 2 questions because of time constraint.
Ma'am, while you growed your this molecule in GROMAX, after the was there any conformation change after you have visualized that in the solvent pocket?
Yeah, when I was simulating only protein, there were no conformational changes throughout the time. But when ionic liquids were added to the protein, in last few nanoseconds, I observed changes.
So was there any change in the binding site or binding affinity of that?
Yes, yes, we found that. That's why we came up with the result by interaction sites.
Thank you so much.
Any questions?
Wonderful presentation.
Thank you.
My question is why you have chosen the 310 Kelvin temperature for performing your experiment? And my second question is, have you checked your RMSD after the molecular dynamics?
RMSD. Yeah, that was the first step. We have to decide that protein is stable or unstable. If there is any fluctuation in the protein through time, we will go for further analysis. So RMSD is always the first step of analysis. And yes, about your first question, 300 Kelvin, that is room temperature. So we prefer to study on room temperature.
Okay. Actually, I have a doubt when you have performed the molecular dynamics after attaching the different groups and you have used in the GROMAX, you have to check the RMSD, whether there is any fluctuation in the alpha chain and beta chains.
Can you please repeat the question?
My simple question is, have you checked the RMSD after molecular dynamics or not?
Yes, after MD simulation only we check.
It's a proper?
Yeah, in the case of only protein, we observed no fluctuations. There was no changes in protein throughout the simulation. But with the addition of ionic liquids, we observed few changes. That's why we went for further interaction studies.
Okay, thank you very much.
Thank you, Pradipha, for your nice presentation. Our next participant is Ms. Anita Chauhan is here. Anita, come on stage, please, for your presentation.
Evening, everyone. Good evening, everyone. This is Anita Chauhan from Indira Gandhi National Open University, New Delhi, and I'm here to present my work on in silico prediction of ERBB2 GRB7 interaction as a molecular switch to activate NSCLC, non-small cell lung carcinoma. So please be patient with me for next 6 to 7 minutes. So NSCLC accounts for fourth common cancer in worldwide, and it accounts for 85% of all lung cancer cases. The reason for taking this cancer for study was that it is highly asymptomatic, and at the time patient is diagnosed with NSCLC, he is already in the metastatic stage. So causative agent so far is not known for NSCLC. So I have got two ideas from NSCLC research. One was that the target itself changes through mutation while it is changing during the mutation. Just a second. The target itself changes through mutation, and it finds the alternate pathway. And it finds the alternate pathway to regain the tumor pathway without target, and sometimes specific networks and their associated hubs, they can be used as a cancer biomarkers. Using these associated things, we have taken the research forward. So in this presentation, we will be seeing how we have identified the differentially expressed genes in NSCLC. We have seen their cancer and stage-specific expression, and two network modules have been identified in NSCLC joined through a single marker. So this is the methodology, what we have done during this research. So I'm going to skip this. I'll explain this methodology because of time constraint. I'll explain with the results as well. So what I have done, it's short. I will tell you how we have done it. We have done literature-based text mining. We have identified some NSCLC genes. From these genes, we have validated them from microarray datasets and taken some common genes with the evidences. 85 genes have been selected, and from these genes we have seen their gene interaction in 9 different tissue types, and we have taken a lung specific interaction of 5 genes. So further we have worked on these 5 genes only. We have taken these 5 genes for tissue and cancer specific co-expression, because we are not finding only one target, we are finding a module of targets, a network of targets with hub genes. So these were the results we have got. We have got 336 genes which we have retrieved from literature mining, and 181 genes we retrieved from this microarray validation process. So this is the volcano plot of research. So you can see that the green structure which we can see, these are the significant genes which are present in NSCLC. So we have taken all genes which are positively and negatively expressed both, but with the two-fold expression. And after that, we have seen their gene interaction in different 9 vital tissues. And from these was the interactions which were present in lung only, because we wanted to consider the targets which are lung specific. So because we are not finding one target, we are finding a network of targets. So what we have done is we need to find the co-expression of these network. So this is what we have done is we have find the tissue specific co-expression network of these genes. So we can see that these 5 genes are highly significant in breast and lung and their respective cancer. So here this diagram graph is showing the expression of these genes in different vital tissues and their respective cancers. So we can see these two clusters are forming there. One is breast and lung and their respective cancers. So in clustering also, we could see that on tissue-based clustering, we saw that breast and lung are forming a cluster. They are expressed in breast and lung. Specifically, we are not taking breast for this time. We are taking only lung-specific tissues. So what we have done is we have selected the interaction which are present in lungs only. So this was the interaction we have used for our further research. So this interaction is here, it came from string. So this interaction is forming a cascading network. These 5 genes were interacting in A and B. You can see they are in normal and lungs, how they are interacting. So this was the main motive we have identified in NSCLC. I have put a yellow line over there. Below yellow line, there is PAK1 keratin 7 interacting with ERBB2, and above line is for NSCLC. In NSCLC, ERBB2 is interacting with GRB7. So ERBB2 is going to be the hub of genes, hub of network, of genes. That means it is highly interacting, and if it take out this gene, the network will collapse. That is a hub gene. So in this case, ERBB2 is a hub gene, and it's interacting with GRB7 and PAK1. Why we are taking PAK1 and GRB7 only? Why we are not taking any other gene? The reason being is the line of evidences. The green line, different colored lines you are seeing is the evidences for the prediction of this interaction. Every color of this line gives different type of evidence. For example, green shows the co-dominance, black shows the co-expression, experimental evidence is given by red line. So number of lines increases means number of evidences are increasing for a particular prediction. So because this was the molecular basis, we have not done this for so far. So now we want to see the molecular basis for this interaction. Why PAK1 is interacting with ERBB2 and how it is enhancing the interaction of GRB7 with ERBB2. So this was what we have done is interaction GRB7 and ERBB2 was structure was available in PDB. We have found the PAK1 interacting with ERBB2. So PAK1 has a consensus sequence PXXPR, right? So PAK and ERBB2 interaction, there was not any structure present in PDB. So what we have done is we have taken a structure of beta PIX, it is a row guanidine. We have taken a structure of SH3 domain of beta PIX. So PAK1 interacting with SH3 domain of beta PIX. From that is why we have compared our ERBB structure. We have aligned it with the beta PIX with pairwise alignment. We got a 43 residue long sequence. So for this reason, we have predicted a model for 43 residue long ERBB2 using Swiss plot. And after that, because we want to find the binding of PAK1 with ERBB2 model, so we have just done the regular procedure of energy minimization for PAK1 and model, because there could be any kind of transformation, any kind of distortion in the structure. So we need to balance the structure after. That is why we do the free energy minimization. So this was what we got after docking. So we have done autodock vena here. So we could see that PAK1 is binding to ERBB2 in tryptophan group. It is the tryptophan is a bigger group, and it is making a groove for binding of the external protein. So PAK1 is binding to the pocket. It is binding through the arginine group of arginine residue of the consensus sequence, and when it binds to the ERBB2, it alters the ERBB2 pathways, which enhances the binding of GRB7. So both PAK1 and GRB7, they are binding to the ERBB2 in different regions, right? So when GRB7 binds to this region, so after that, it will take part in the ERBB pathway in cell regulation and cell migration, and ERBBT, it will exert an repressive control on GRB7. So the conclusion of the study is we have found the genes which are differentially expressed in NSCLC. They could have multiple interaction with other genes and which can promote cancer, carcinogenesis. Another thing is we have found an interaction PAK1, GRB7 and ERBB2, that is a transforming connection between lung and normal cancers, and ERBB2 that can act as a regulator of transition from normal to cancer state. Thank you. Any questions, please?
Any question?
Yes, sir.
Good evening, madam.
Yeah, good evening.
Ma'am, you have done, or I can say you have used Autodoc Vena for docking.
Yes.
Is it the right platform to perform virtual screening or docking?
Is it right platform to do virtual screening?
Is it a right platform to do virtual screening or docking? If we are docking,
Doc.
it's a small.
See, yes, sir. Are you talking about Autodoc Vena, right?
Yes.
So, sir, what in virtual visualization, if you are saying virtual docking and docking, so I guess because I have done this Autodoc Vina for many studies earlier also. So, for docking purpose, if you want to do, you might have heard about flexible docking and rigid docking, right? So, when you are docking, for flexible docking, Autodoc Vina is good. For smaller platform as well, for bigger platform as well. For flexible docking, it will show good results.
But I think there must be Autodoc 4.2 is better for it.
These are the modulation. Definitely, obviously, when you these softwares, they will keep on, developing. They will keep on improving the things. Autodoc Vina itself has 4 models now. These are 4.1 to 4.2, and now it has come 6.2 as well. Right.
Yes, but mostly the virtual screening when you have more than hundred or thousands of compound, or we can say more than 10 compound.
Yeah.
So you can use Veena. It will perform the work in a very small number of time.
Mhm.
But when we are doing the specific docking with a single compound, then I think it is better to use Autodoc 4.2 instead of going for Veena.
I don't think so. I don't have find any problem with doing Autodoc Vina so far. And the reason being because virtual screening is when you are screening many inhibitors or many ligands you have to dock with your receptors, then it's okay. But for a single docking also, I didn't find any problem with that.
Okay, can you tell me, because I don't know how we can make a grid box through a site in Autodoc Vina?
A grid box. So, see, when you want to make a grid box in Autodoc Vina, you need to give the dimensions first, like where you want. There are 2 options. One, you can use script language, where you can give the X and Y coordination for that. Otherwise, another thing is you in Autodesk Vina, you have a graphic user interface also there. You can just drag your this mouse and you can give the pocket, the main pocket of the, where is the active site of that protein. So you can, where you want to dock your particular ligand to the receptor. So there are 2 options. Either you script it or you can just drag the grid to the active site.
Okay, but I have seen that while I am using the Autodoc Vina, I have to write the script. There is a no GUI for Autodoc Vina.
There was no GUI.
Yes.
So you don't have GUI, but you are facing problem with that.
Yes. But the
So what you can do is just I'll give you a small solution to you. What you can do is you can find you have your protein with you. What you can do is you can open your protein in notebook, just any notepad, right? You can find out the heteroatoms, the ligand part where it is, right? So from that point, there are residue numbers written over there. Either whenever you download any PDB structure, there is a paper referred to that structure which is submitted with along with that structure, right? So what you can do is you can take out that paper as well. In that paper also there will be some residue sites. The active sites will be written over there. Either you do that or you can find it from the notepad file. You have opened your FASTA file in your notepad. From that point you can take out your active site residue, and when you run your if you are not having any graphic user interface for that, in script you can give the grid file as a input where you can write the dimensions at y and x coordinates, and that will be done. Anything else? Any more questions? Thank you.
Thank you, Anita. Our next participant is Ujjwal Gupta. Ujjwal Gupta is here. Ujjwal, please come on, guys.
So a very good evening to everyone present here. Myself Ujjwal Gupta, the student of Naipar Rai Bareilly in MS from Pharmaceutics, going to present my topic nanohybrid system for the delivery of the treatment of the amyotrophic lateral sclerosis under the supervision of Dr. Avesh Kumar Yadav. So these are the contents. So moving on to the introductions, the nanohybrid systems are the chemical conjugates of the 2 or more of the nanosystems. Now ALS is mostly called as the motor neuron disease, and it is also affects the spinal cord. The one of my carrier, that is fullerene 360, it is also called as Buckminster fullerene or buckyball, which is a unique carbon shaped molecule. So mainly in the conventional dosage form, there is a problem that they cannot cross the blood brain, which is a very complex barrier. So by the nano formulation, I am overcoming this. So this is the representation of a pathophysiology of the ALS. So mainly we can see here many of the cellular mechanisms that are taking place, like glutamate release, excitotoxicity or apoptosis. So these are the main mechanism of the ALS. So this is the drug profile, the riluzole that I am using in my research work. So it is a BCS class 2 drug and having a half-life of 12 hours. But the problem is this half-life is because of the 4 repeated dose that you give to a patient at every 4 hours. And it is a hydrophobic drug. As you can see, it is a BCS class 2 drug. So these are the objectives of my work. First is to optimize and characterize my nanohybrid system and to conjugate the fullerene with a BSN nanoparticle and to prolong the nanohybrid system circulation time and to perform various in vitro studies. So this is the hypothesis of my work. First, I have taken the carboxylated fullerene and then I will be conjugating it riluzole loaded Bowman serum albumin nanoparticle. So after the the mode of delivery will be through the intranasal route, as we can bypass the blood-brain barrier through this route. So the delivery will be through the trigeminal nerve and olfactory nerve directly into the brain. So this is the rationale, like in the case of fullerene, we have a small particle size and the functionalization is very easy. And the fullerene is said to have neuroprotective and as well as antioxidant properties also, which is beneficial in the cases of neurodegeneration diseases. So this is a method I am using for the preparation of BSN nanoparticle. This is a dissolution method. So in the first step, mostly the 50 mg of BSA dissolved in the 10 ml of water. Then I am adding 20 ml of acetone for the phase separation and 8% weight by volume glutaraldehyde as a cross-linker agent, intramolecular cross-linking takes place. So after that you can centrifuge and get the nanoparticles. So this is the schematic representation of formation of a riluzole loaded nanohybrid system. First, I have got the fullerene that I will convert into carboxylated fullerene by the reaction of H2SO4 and HNO3 in the ratio of 3:1. After this, the carboxylated fullerene will be attained, and with the two coupling agents I am using, EDC and NHS. After this, we activate the carboxylic group of the carboxylated fullerene, and then the amine group of that is present on the BSA nanoparticle will react to COH group of a fullerene, and further it will form a amide bond. So these are the resultant discussions. So first I have scanned my drug for the over the wavelength of 200 to 800 nanometer, and the lambda max was found to be 220 nanometers. This is the standard calibration curve, and R square was found to be 0.9982. This is a particle size and the zeta potential of a nanohybrid system. So the particle size is here around 210 nanometer, which is very good for the intranasal drug delivery and the entrapment efficiency. And when we talk about the drug loading, it is around 97% and 9% respectively. So this is a same image of the nanohybrid system. Mostly in last slide, this slide we have seen the particle size is around 200 nanometer, but the DLS only shows the average particle size. But in the same, we can actually see the particles. We can mark one by one the particle, and here you can see a particle of 238 nanometer and 224 also 228. So this is the FTIR spectra which I had obtained. Like in the fullerene, you can see a peak around you can see a peak around 527 nanometer and 1160. These are due to the movements of the carbon atoms in the structure. And when we come to the second spectra B, that is of carboxylated fullerene, so mainly we can see a peak around 744. That confirms the presence of COH group. That means the fullerene is carboxylated. And then we see in the third spectra C, that is of Bowman serum albumin nanoparticle, we can see a amine at 1540, the OH at 3620, and the COH of the protein at 1744. But in the final spectra, we can see the amide formation at 1651 and the amine stretching at 1523 and amine bending at 3281 and CH. So this is a CD spectroscopy because CD spectroscopy is most important in the case of protein because we have to see whether the protein structure is changing or it is remaining the same because it's more important while we conjugate or something. But here we can see the structure remains the same. The two bands around 208 and 222 is same in both the spectra. So this is the XRD representation in the ribosol and fullerene. There are almost three characteristic peak, but in nanohybrid system, we can see there is a broadening of a peak and little amorphicity is also shown in the spectra of nanohybrid system. That is due to the BSA nanoparticle conjugation. This is an in vitro release profile of the drug. We can see the blue line shows the plain drug release and it is hydrophobic, so it only releases nearly about 33% in 48 hours. But when I loaded the drug in the nanoparticle and conjugated it with a fullerene, so the nanohybrid system shows a release of around 76% over a period of 48 hours. This is a DPPH assay I have done. It is for the antioxidant property. So we can see here in nanohybrid system, we get the synergistic effect of antioxidant properties of the both ribosol as well as of fullerene. So the final antioxidant property is around 55%. So these are the references I have used. So I would like to acknowledge my supervisor, Dr. Avesh K. Yadav, Director NIPER Raebareli, Professor Subni A. Saraf ma'am, Vice Chancellor Bundelkhand University, Professor Mukesh Pandey sir, organizing committee of this international conference, PhD scholars and lab mates of my lab, Central Instrumentation Facility of NIPER Raebareli, Institute of Pharmacy, Bundelkhand University, Jhansi, Indian Institute of Technology, Kanpur, and at last the Ministry of Chemical Fertilizer, Government of India for providing me fellowship throughout my work. So this is the research team at NIPER Raebareli. In between here is my supervisor, Dr. Avish K. Yadav, and other batchmates. Thank you. Any question?
My question is, why you are using two nano carriers? Can use the only one or not?
Yes, I will tell you about that. Yes, I can use fullerene alone, but fullerene is a kind of options for our body. There is a process of optimizations. Like the fullerene will, as it is not, will be not accepted in our body because it is a foreign particle for our body. So with conjugating with it a BSA nanoparticle, because BSA is also present in our blood. It is a blood protein. It's a kind of blood protein, so it will hinder the process of optimization and it will target the system to the brain.
Another question is, you can go another BCS classification like a BCS third or fourth or one. Yes or not?
What? Please repeat your question.
Yes, I am asking about you are using a BCS class second class and you can use a BCS class 1 or third or fourth. Yes or not?
Yes, but mostly in NIPERs we target BCS class 2 and BCS class 4 drugs because they have a problem of solubility or permeability. BCS class 1 and 3 does not have a similar kind of a problem.
Okay, thank you.
Thank you everyone.
Thank you as well for your nice presentation. Now we have amongst us Ankita is here. So Ankita, please come on stage and share your presentation with us.
Very good evening to everyone present over there. Myself, Ankita Kishore, PhD Scholar, Institute of Pharmacy, Bundelkhand University, Jhansi, under guidance of Dr. Alok Kumar Mahor. So I have chosen my topic today for the presentation, Artificial Intelligence, Positive and Flip Side Highlights in the Era of COVID-19. So first of all, go through the introductory part, that what is the artificial intelligence. So there we can say that this artificial intelligence is actually the simulation of human intelligence processes by the machines. And it is basically involving the computerized systems. So the next question is how this artificial intelligence is actually working. So this is actually working by absorbing massive quantities of label training data. And after collecting these kind of training data, these are examining the data for correlations and for the particular patterns. And then after deciding the particular patterns and correlation, these utilizing these patterns to forecast the future states. So this artificial intelligence is basically focuses on 3 points. First is the learning processes. In this learning process, they are collecting basically the data and choosing the algorithm, various types of algorithm. Through these algorithm, we can convert these information into the actionable information. Now, second one is the reasoning processes. In the reasoning processes, in this step, this is focusing basically on choosing the correct algorithm due to which we can target or we can complete the specific task. Third one is the self-correction processes. In the self-correction processes, there is continuous updation or auto-find in the algorithms due to which we can finally ensure that the specific type of concern we are going to acceptable finalize the information or not. Now, the next one is there are basically 3 terms we are using: artificial intelligence, machine learning, and the deep learning. So artificial intelligence is basically the broad term, and this machine learning is the subset of the artificial intelligence, and this deep learning is the subset of the machine learning. In the artificial intelligence, in which data, information, and human intelligence are transferred to machine during these processes. In the machine learning, this is basically involving all the algorithm and solve the corporate changes using the data and computer algorithms. In this deep learning, these are actually uses the neural networks, and these neural networks is actually like the neurons in our brain, as our brain is having the capacity to finalize and to decide whatever the situation is, what the decision we have to make. In the same manner, this deep learning is actually working, and this deep learning is the core application of the artificial intelligence, which is right now using in the healthcare systems. Now, there are some simple examples has been given for the artificial intelligence, machine learning, and the deep learning. In the artificial intelligence, the most common example we can see that we are using the household things like Alexa. We are using varieties of questions we are asking to the Alexa, and through this artificial learning, this Alexa is giving the answer. In the machine learning, it is actually using the algorithm. So in the machine learning, if we put one cats and dogs images, so this data has to be decided through the algorithm in the machine learning. That data is processed, and after giving some queries to this algorithm or to this machine, it is able to identify that the given image is for the dog or for the cat. In the deep learning, one example is there that we have provided as a data images of a different type of retinas. So due to the neural network of deep learning, it uses and create one healthy image of the eye. And this healthy image of the eye retina, this deep learning can compare with the diseased eye, and can after the comparison, it can give the output that whether the given input is of the diseased retina or of the healthy retina. Now, going through the COVID-19 pandemic, we all know about this, that it is actually right now also going on, global disease outbreak brought on by the coronavirus 2, and it causes severe acute respiratory syndrome, that is SARS-CoV-2. Virus was first identified in outbreak on December 2019. WHO has declared public health emergency of international concern on 30th January 2020 and a pandemic on 11th March. As of the date of 15th Feb 2023, according to the WHO, pandemic has caused more than 673 million cases as well as 6.85 million confirmed deaths, making it most deadliest in the history. So by using the artificial intelligence, how we have dealt with this situation. Some examples are there, like Blue Dot, contact tracing platform, open voice medicine, model architecture, digital services, search engines, clinical use of AI-guided chatbot, InfoVision's AI, COVIDNet, and drug repurposing. Going through these one by one application, one is the Blue Dot. This Blue Dot is actually one epidemic intelligence system that is actually one startup, Toronto-based startup, that has, and CEO and the founder of this Blue Dot is Dr. Kamran Khan. This Blue Dot is actually using the NLP, that is natural language process and machine learning. It compiles data from many sources. It is actually collecting the data from different sources like health companies and digital medias and different type of health reports. These are having the ability to quickly handle a large amount of information, means they are updating their information at every 15 minutes around 24 hours. And his CEO Khan, he's given one beautiful statement that we don't use artificial intelligence to replace human intelligence. We basically use it to find the needles in the haystack and present them to our team. Means they are not just bypassing the human intelligence by using this artificial intelligence, but we are interconnecting them, and we are utilizing in more powerful way. Now, second is the contact tracing platform. This is actually the Bluetooth-based applications. And two examples are given here, that one is the Arogya Setu app and another is the COVID Safe app. This Arogya Setu, we have much familiar about this because we all have used it. This Arogya Setu app has been implemented by the Indian government, and this COVID Safe app has been implemented by the Australian government, and both have been developed in the Singapore. Open voice medicine model architecture that is designed by Myshoots Institute of Technology. Here, what they have used that through the online, through one application, the smartphone application, through the forceful breathing or forceful coughing, they have identified that whether this coughing sound is of a healthy person or is of one infected person. So they are using vocal cord changes and muscle deterioration through their identification. This is actually one smart for how this app is working. They are collecting the data through the deep learning framework. These sounds has been identified and classified, and then final decision has been made that two differentiation they have been made, one for the COVID-19 patient and another for the healthy persons. In the performance evaluation step, if shallow breathing is there, yes. Continue. Yes, sir. If shallow breathing is there, there's no much more differentiation between the healthy and COVID-19 patient. But if deep breathing we are doing, then there are some differentiation we can see. Now, digital services, hotlines and online questionnaires are there. We can see that hotline, when we are going through the social media, when we are going through the hotlines, there are some certain platforms are giving through which some numbers are given. On that, contacting on that numbers, we can easily contact to the concerned person and can given our query. Now, this is WHO data board that which giving a lot of answer to the journalist asking question. They have also given the hotlines numbers. Search engines or symptom analyzer. Their WebMed symptom checkers are there, Mayo Clinic symptom checkers are there, through which we can easily analyze our symptom and can predict that we are having the infection or not. Now, the clinical use of AI-guided chatbot, like pop-ups, many times pop-ups are there through which we can text our queries to them. And these queries is actually they are using for enhancing their knowledge, and by enhancing their knowledge by decision making, they can use or utilize it in a more productive way. In this way, chatbots are working, that we are giving in form of the text or sound to them messages. By deep learning, they will convert into the natural language processes. Infravisions AI. This is actually using to study the CT scans within 10 seconds. Otherwise, when we are going through the manual reports reading, then it will generally using taking the 15 minutes, but it is completing it within 15 seconds. Now, COVIDNet. COVIDNet is also one open source and can compare the X-ray of COVID-19 patient and a healthy patient. So this A diagram is for the COVID-19 X-ray, healthy patient X-ray, and this second diagram, second X-ray is for the infected lungs. So in this way, this COVIDNet can provide during the utilizing the artificial intelligence can show the results. Drug repurposing. Like remdesivir we are using for the drug repurposing. So there also our AI has played very important application, and here varieties of information has been given, and due to molecular docking we were discussing. So there this type of techniques we can use and can give the results that whether our drug can be repurposed or not. There are some other applications like early warning and alerts can be given by using different type of apps such as Blue Dot, tracking and predictions like Arogya Setu app, that data dashboard like WHO data dashboard is there. Diagnosis and prognosis through the symptom analyzers, and treatment and cures, social control, and Google's DeepMind, some artificial intelligence applications are there. Conclusion. So actually conclusion is this, that is still there are a very much lack in the information that is we have to provide on this artificial intelligence platform, because either there is too less data or too much noisy data is present, due to which exact decision is we are not able to make. What are the future prospects? That more data can also be available there. This can increase the ease of work for the healthcare professional. This can also reduce the workload, and more data available for the professional and educated people. In somehow we can also deliver it to the general people who are not able to understand this. So this is some references that I have used. Thank you. Any questions?
No question. Thank you, Ankita, for nice delivery. This was fourth presentation. So fourth presentation session has been chaired by Dr. Vivek Tomar, sir. Now for next sixth presentation, the session will be chaired by Dr. Laukus Dwivedi, sir. He is a eminent faculty member of Bundelkhand University, Jhansi, in the Department of Biomedical Sciences. So I would like to invite on stage Deena Prakash. She is here. Deena, please come on stage and deliver your presentation, please.
Good evening, everyone. My name is Deena Prakash. I am a research scholar in IGNOU. My topic is hepatoprotective activity of Selaginella bracteris against acetaminophen-induced toxicity in Vistar rats. Okay. So to start with, my plant is Selaginella bracteris. It is a lithophytic pteridophyte, mainly found in Aravalli Hills. It is locally known as Sanjeevani. It contains variety of phytochemicals like alkaloids, flavonoid, tannin, saponin. It is used to treat various ailments like constipation, dysentery, fever, jaundice, urinary infection traditionally. So, okay. Paracetamol is acetaminophen is also known as paracetamol, which is a common drug as antipyretic drug, which is prescribed by doctor normally. So, paracetamol acetaminophen is metabolized in liver producing by cytochrome P450 enzyme producing NAPQI, a toxic metabolite, which deplete the antioxidant system of liver, which thereby causing liver damage. Okay, material method. Plant extract was used for therapy. As acetaminophen was used as toxicant. Silymarin was used as positive control. So this is the study plan of the study. In that study plan was divided into two type of study: acute study and subchronic study. In acute study, paracetamol was given at 2 gram per kg dose postorally. Plant in therapy group, plant extract was given at 250 mg per kg, 500 mg per kg, and 750 mg per kg. Positive control was used as at 50 mg per kg dose. After the finalization, acute study was done for screening out the plant extract dose. After the finalization of the dose of plant extract, we proceeded to subchronic study, in which the paracetamol was given for 21 days for 20 mg at 20 mg per kg. Plant extract was given 750 mg per kg after toxicant. Similarly, Silymarin was given at 50 mg per kg for 5 days after toxicant. Now, these are the tests which were evaluated for the liver function, for liver toxicity results. So in acute study, male vista rats were divided into 6 groups. First group was control group. Second was experimental control. Paracetamol was given in this group only. Group 3 to 5 was after paracetamol administration. Plant extract was given for 24 hours at 3 doses. Last was a positive control group. After 24-hour administration of all the doses, sacrifice was done and blood biochemistry, tissue biochemistry, and histology was performed. So this is the slide of showing the overall effect of plant extract on hepatic marker in paracetamol-induced hepatotoxicity. Due to the paracetamol caused the increase in AST, ALD, LDH, and bilirubin. These are the hepatospecific marker which was released due to the damage caused by paracetamol. And with the following treatment of plant extract between 250 to 750 mg per kg dose, these enzymes level were decreased in dose-dependent manner, which was similar to silymarin group. Now, for acute study, we performed only lipid peroxidation and reduced glutathione tests. So in paracetamol caused an increase in lipid peroxidation, suggesting that plasma membrane damage in the hepatocytes, and which was recovered in dose-dependent manner. And however, reduced glutathione is an antioxidant. It was depleted due to toxicity, which was then increased with the help of plant extract at higher dose. The maximum effect was shown at higher dose of plant extract. Now, these are the histological image of liver, in which B is the toxicity group, C, D, E are the plant extract group with respective doses. Last one was silymarin group. You can see that in APAP, focal necrosis and cellular infiltration was there. But with the treatment of plant extract, these toxicity characteristics were recovered with similar to silymarin group and normal group. To conclude the acute study, SBEE extract showed dose-dependent recovery at 250 mg per kg, 500 mg per kg, and 750 mg per kg. Acute against paracetamol toxicity, acute one. Out of 3 doses, 750 mg per kg was effective. Therefore, 750 mg per kg was further evaluated for subchronic study. So in subchronic study, male vista rat were divided into 4 groups randomly. In group 1, control group was there. In group 2, paracetamol with 20 mg per kg dose post-orally was administered for 21 days. Group 3 was after 15 days of paracetamol toxicant administration. 5 days treatment was given at 750 mg per kg. In last group, similar administration was there for 5 days. Silymarin was given. Now, after 24 hours of at last administration, sacrifice was done for evaluation of blood biochemistry, tissue biochemistry, pro-inflammatory cytokine, and histology. Now, this is the slide showing the overall, overall effect of blood-specific hepatomarkers. Now, here you can see that red bar is paracetamol-induced toxicity 1 group, so in which you can see the level of AST, ALT, LDH, ALP, and bilirubin were increased. But after the treatment of the plant extract and silymarin, the effect of toxicity decreased. Now, LPO-GSH also, APAP caused the increase in lipid peroxidation, which was again decreased with the help of plant extract and silymarin. Reduced glutathione was depleted again. It was increased or elevated due to with the help of plant extract. Now, oxidative stress marker. These are the oxidative stress marker. SOD catalase, glutathione peroxidase, and glutathione reductase are the remarkable oxidative stress markers. In liver, they have the very important role for regulation of antioxidant activity. Now, SOD catalase, GPX, and GR, every enzyme were depleted in paracetamol toxicity group. It was recovered with plant extract between 60 to 70% protection, which was similar to silymarin group. This is the histological image of these 4 slide images are the histological image at 100x magnification. Now you can see the A is the normal control. B is the toxicant group. You can see the increased sinusoidal space and focal necrosis also, and the disarrangement of central vein. Now you can see is the plant extract treatment group. You can see that all the focal necrosis was decreased, sinusoidal space was filled, and normal hepatocyte structure was there. D was the Silymarin group. You can see it is similar to the normal group. Now, this slide showing the ultrastructural histology of SBEE against subchronic toxicity. You can see that nuclear membrane in normal control group, nuclear membrane is clean, very sharp, and mitochondrial number was also was there. But with the toxicity, nuclear membrane was disarranged, and mitochondrial number was decreased, and cellular vacuolization was also there. Now with the treatment of plant extract, you can see that clear nuclear membrane was seen, mitochondria was also seen, and ER also seen. Now, which was compared with the Silymarin group. Therapeutic. This slide shows the therapeutic effect of SBEE on pro-inflammatory marker cytokine tumor necrosis factor alpha. Since tumor cytokines are increased in case of any inflammation or any disease, so hereby also TNF alpha was also increased in APAP, which was then again recovered in plant extract and Silymarin group. Now, to conclude the overall study, we can say that 5 days of therapy with plant extract at 750 mg per kg showed a pronounced hepatic recovery in APAP treated group, which might be due to reported phytochemicals like amentoflavone and henicoflavone. It showed anti-inflammation and anti-inflammatory effect by restoring the level of TNF alpha. Microscopic studies revealed that it can have a regenerative activity. I would like to acknowledge, first I would like to thanks Institute of Pharmacy for organizing this humongous international conference. Thank you for making me part of this. Now I would like to I would like to acknowledge my PhD supervisor and PhD co-supervisor and all the persons who helped me in this whole process. References. Thank you so much. Any questions?
Yes. Hello. Madam, my question is to you. Did you perform any LD50 estimation for this study?
Sorry, I can't hear you. Where are you? And
I am asking that, did you perform any LD50 estimation for this study?
actually, it is already reported in paper already that lethal dose of selaginol bipteris was around 2000 mg per kg. इसलिए हमने कम से कम डोज में काम किया है.
Like, कम से कम, not apart from the कम से कम 2000, 200, 250, 500, 750.
Yes, but it was it was like 1000 से भी कम था वो. And profound जो भी effect था वो 750 mg per kg पे ही मिला था. And it was referred to another papers also, research reported papers.
And method of euthanasia?
Sorry?
Method of euthanasia. You have not mentioned the method of euthanasia.
Methodology?
I'm asking about the euthanasia. You have already cleared your CPCM rule, I think, for performing the study.
Yes.
What is your method of euthanasia?
Acha, euthanasia, okay. So we gave the chloroform, and then cervical dislocation was done, and then we retrieved the blood from retroorbital puncture, and then opening of a rat was done to retrieve the liver. Okay.
From where you get the approval of your animal study?
I would like to thank Bundelkhand University and Animal Facility for providing me certificate, like this facility to do my experiments.
Okay, thank you.
Thank you.
Lauku sir, you have to ask question?
Yes, sir.
Okay. Dina, please come on stage once again.
Yes, you have taken the ethanolic extract. So is this a pure ethanolic extract or you?
Pure ethanolic extract.
So you have got that 100% pure ethanol.
Yes, sir.
Then you are using a 100% pure ethanol. So your extract is in a dried form or is it in a semi-solid form?
सेमी सॉलिड फॉर्म मतलब बहुत ही लिक्विड ही सा मतलब हाँ इसका थोड़ा ऐसा स्ट्रक्चर था मतलब.
So how you maintain the drug accuracy, dose accuracy?
Oh, okay. So we dissolved the, first we made the stock solution, and then after that, we calculated the study, like doses from the stock solution. No, you're not asking this. We dissolved the plant extract in water and other soluble solvents. It's not clearly soluble in water, so we have to like give it 24 hours to dissolve with water. Like thoda sa. No, it's not completely soluble in water. Okay, so जब हमने इथेनॉलिक एक्सट्रैक्ट बनाया था तो उसको जब हमने वाटर में डिसॉल्व किया था तो हमने उसको कम से कम 24 घंटे के लिए रखा था, जस्ट टू एंड शेकर पर रखा था, जस्ट टू डिसॉल्व इट इन वाटर, बिकॉज और कोई तरीका ज़्यादा इफेक्टिव नहीं होता है.
Thank you, Dina Prakash. Now we have our next participants, Divya Vist from IGNU. Divya, please come on stage.
A very good evening to all. Myself, Divya Best, research scholar, PhD biochemistry, and I'm here to present my PhD research work on titled Nephroprotective Effect of Polyalthia longifolia against cisplatin-induced toxicity in vitro and in vivo model. Here is the overview and objective of this whole study, where I have used 70% ethanolic extract of Polyalthia leaves, and in vitro study, MTT assay was performed, and for in vivo study, biochemistry and histological analysis was done for assessing the nephroprotective effect of this extract. Coming to the plant, Polyalthia longifolia is a tall evergreen tree, which is commonly found in India and neighboring subcontinent. You can easily find this plant even in your own campus. It has rich pool of phytochemicals and is known widely for its therapeutic use, as well as utilized in Ayurveda and other traditional medicine system in India and neighboring countries. Here are some diseases again which it shows therapeutic effect. Coming to the toxicant, cisplatin. Cisplatin is a popular anti-cancer drug which is used as a chemotherapeutic agent to treat wide range of solid tumors. It is highly effective, but its medicinal use is limited due to its adverse effect on patients like ototoxicity, neurotoxicity, nephrotoxicity, vomiting, nausea, etc. Here we are focusing only on nephrotoxicity. So in case of nephrotoxicity, the drug reaches to the kidney through bloodstream where it get accumulated in renal cell and cause cellular dysfunction, oxidative stress, generation of free radicals, which later cause tubular injury, necrosis, apoptosis, decrease in filtration rate of kidney, ending in acute kidney injury, which on prolonged exposure or repetitive dosage turns into chronic kidney diseases, leading to kidney failure in patients. Here are the material and method used. So 70% ethanolic extract was prepared after defatting it, and chemicals, all the chemicals, drug were used of analytical grades. For in vitro, HEK293 cell line was used, which is human embryonic kidney cell line. And for in vivo study, male Vistarat were used. All standard guidelines and procedures were performed while executing these experiments. Now the results. For in vitro study, we had started with cytotoxicity assay to check whether this extract is toxic or not, and we found that this extract is non-toxic and safe to be administered. Then for in vitro protective effect of Polyalthia longifolia leaf extract, it was found that PL leaf extract is significantly showing protective effect in significant dose-dependent manner, and the result were comparative to that of positive control silymarin, which is a well-known nephroprotective drug in market. So both these results concluded that in vitro model, the extract showed nephroprotective or protective property against cisplatin toxicity, which further validated in vivo model. So for in vivo model, there were 6 groups with 6 animals each. The first group was a normal control. Second was experimental control, which was given toxicity of cisplatin only once. Group 3 to 5 were therapy of plant extract, for which cisplatin was given on day 1, and for next 2 weeks therapy was given. And the last group was positive control, which was given toxicity of cisplatin on day 1, and for 2 weeks silymarin therapy was given. After last administration, that is on 16th day, the sacrifice was done using chloroform as and euthanasia and cervical dislocation. Then the blood samples were collected through retroorbital puncture, and tissue organ were collected for further blood biochemistry, tissue biochemistry, and kidney histology. Now the body weight and kidney weight of the rats were also observed during this study, and it was found that body weight of the rats given cisplatin toxicity per se decreased during the course of this experiment, which can be due to the loss of appetite, dehydration, and internal toxicity, including GI tract toxicity, nephrotoxicity, and other toxicity already reported. But also kidney, in contrast to that, kidney weight was found to be increased in cisplatin intoxicated group, which indicate inflammation and drug accumulation in kidney. Both these effects were reversed significantly by the plant extract in dose-dependent manner, and the result reduced similar to that of our positive control. Now we also have assessed in blood biochemistry the kidney function markers, which include urea, uric acid, creatinine, and albumin. These their level were found to be altered in toxicity, cisplatin toxicity group, but was later on reversed by all 3 doses of plant therapy, and the result of highest dose 600 mg per kg was found to be maximum efficacy. Now, lipid profile marker were also assessed, and it was found that triglyceride and cholesterol level were highly increased in toxic group, which was reduced and recovered by Polyalthia longifolia leaf extract therapy in dose-dependent manner. Also, since we had said that cisplatin cause oxidative stress in kidney, so oxidative stress marker were also analyzed in this study. LPO and GSH test were performed. In this, LPO level was found significantly increased in cisplatin toxicity, indicating cellular membrane damage, which was reduced, recovered through all 3 doses in dose-dependent manner, indicating that Polyalthia longifolia leaf extract has antioxidant property, which I had earlier presented for its antioxidant property, and it has significant antioxidant properties. GSH level was reduced in cisplatin toxicity group, but later on increased considerably in all 3 doses in dose-dependent manner. These are other 4 tests of oxidative stress marker where SOD catalase, glutathione, peroxidase and glutathione reductase level decreased considerably in cisplatin toxicity, showing it oxidative stress, but its level was recovered significantly by the therapy of PL extract in dose-dependent manner, and the results were found similar to that of positive control as well as normal group. To validate all these results, these were similarly reflected in that of histological analysis of kidney, where there was considerable damage, gaps, and spaces in both glomeruli as well as renal tubules, and necrosis was visible. But these damage and alteration were reversed in all the 3 doses at image C, D, and E, and the last one is positive control group, And the highest doses of PL extract showed significant effect of recovery in tissue histology, which was similar to that of positive control silymarin. To conclude the whole study, it can be said that in vitro, the Polyalthia longifolia leaf possesses protective effect against cisplatin cytotoxicity in HEK293 in vitro model, and also in vivo model, it showed significant nephroprotective activity in dose-dependent manner against cisplatin toxicity in Wistar rats. It has reversed both the blood biochemistry, tissue biochemistry levels, and the recovery was also visible in histological observations. So Polyalthia longifolia can be considered as a nephroprotective agent and can be used in future as an alternative medicine as a co-therapy with cisplatin to prevent nephrotoxicity in patient given chemotherapy of cisplatin. Now I would like to thank each and every person which was involved in this study and made it a success. I would like to congratulate and thank Bundelkhand University Jhansi Institute of Pharmacy for organizing such a wonderful event. And also I'm going to thank them for organizing the CPCA approval for this particular study, as this study has been done in Bundelkhand University itself. Thank you. Any questions?
If anyone is have any question.
Nice presentation.
Thank you.
When we induce this nephrotoxicity using...
Can you please be a bit loud?
Okay, when we use cisplatin induced model, so there is a problem with cisplatin that usually there is a vomiting sensation whenever the patient take this.
Yep.
Is if you are giving this medicine to a lab rat, is this the possibility with the same?
See, we had observed certain, like physically certain toxicity effects in rats also. Like there was induction of loose motion in the toxicity group, not exactly vomiting, but there was loose motion in Wistar rats with toxicity group. And there was considerable loss of appetite as they were not eating and not drinking water, and there was considerable loss of body weight in toxicity group also.
Okay, thank you.
My question is, when you have given the dose cisplatin, have you checked the bioavailability also?
The bioavailability of cisplatin?
Bioavailability of the drug cisplatin in the Mr. rat? Given the orally, na?
No, sir. We had given it intraperitoneally.
You given intraperitoneal?
Yes, sir.
Okay. So when you are injecting the drug through intraperitoneal cavity, is it available 100% in the blood?
No, sir, but I had checked the bioavailability through literature by other paths also. So the maximum efficacy was with IP or IV. So we chose IP for this particular method because there were enough literature available to test
What is the bioavailability of the drug when we are giving through intraperitoneal cavity?
Sir, I believe it was above 50%. But I'm not,
You have not checked it.
no, I have not checked it myself. We had followed the literature only.
What is the given in the literature? It is below 50% or above 50%?
So it was found to be above 50%. So most effective pathway was this only. Otherwise, the toxicity will be really high and that will be lethal. So to test that dose, that pathway was the only appropriate pathway.
Okay, so what was the final dose of your plant extract, which was showing most effect?
600 mg per kg, the highest dose in this particular.
So why you stopped yourself in 600 only?
So
What is the LD50 of this?
okay, so the LD50 of this plant is 3,240.
Why you have not chosen some more than 600?
So we were going through a range which, so basically the idea was that this was the range in which all the other pharmacological activity were tested. So we were testing in the same group. And then there was another group also for higher study, but we thought that it will come in this range only as the already.
The question is like, when you are observing that when we are increasing the dose, the effect is increasing. if suppose you have got the highest effect on your highest dose, that was 600. The simple question is that why you have stopped yourself on 600 only. You may go beyond when you know that the lethal dose 50 is 3 or 3000. Then why you stopped yourself on 600 only?
So I'm getting the point, but actually the whole like experimental design would register earlier, and we were not aware whether it's gonna show effect on 600 only or
But
not.
I think when you had planned your research, you must have studied your literature.
Yes, sir.
And in literature, like in your previous presentation, when Ms. Dina was presenting, the same question I was believed to ask. When you studied in your literature that yes, it was 750 or 600, which is having the highest effect, या लोगों ने ऐसे ही यूज किया था, तो आपको जब उसमें इतना इफेक्ट मिल रहा था तो आप उसके ऊपर भी जा सकते थे. उसके बाद आप देखते थे कहाँ तक ये जो है वो आपका इफेक्ट बढ़ता चला जा रहा है और कहाँ से डिक्लाइन होना शुरू होता है. आपने तो वो जो ऑलरेडी लिटरेचर में दिया हुआ था उसी को फॉलो किया सिंपली, उसके ऊपर जाकर के नहीं देखा.
Yes, sir.
ना मेरा एक सजेशन है,
Okay.
प्लीज टेक इट एज अ पॉजिटिव नोट.
Sure, sir.
आप थोड़ा सा बढ़ा करके भी दे सकते हो. थैंक यू सो मच.
You wanna ask question? Sir, she wanna ask question again. Oh, then it is fine.
We have, like, I have also picked 1000 mg per kg dose, but the shows the significant, it was not significant at all. वो सिमिलर था 750 mg per kg के, और थोड़ा सा क्योंकि मेरा प्लांट हाँ वो एक्चुअली सब क्वेश्चन वाज सीरियसली नॉट, जो भी क्वेश्चन यहाँ से आ रहे हैं ना वो बहुत तो सुनाई कम दे रहा है. That's
Didn't mention in its slide ना, you should mention. ये आना चाहिए बिकॉज आप अपने आप डिसाइड नहीं कर सकते कि यहाँ पर हमको रुक जाना है.
that.
अदरवाइज फिर इनोवेशन क्या होगा उसमें.
True, sir.
ठीक है मैम, थैंक यू.
Okay.
Our next participant is Mohammad Yasir from MIT University, Uttar Pradesh, Lucknow campus. Mohammad Yasir, please come on stage.
सर ये क्या दीजिए, मुझे ना नोट्स देखना है यहाँ पे. हाँ यस, वो दिखेंगे ना उसमें. नंबर ऑफ स्लाइड्स 22. ओके ओके. गुड इवनिंग. Good evening, respected teachers and my dear friend. I am Mohammad Yasir from Amity University, and I am going to present about the recent topic, that is accelerated theranostics, adapting biomaterial. Actually, when we talk about theranostics, it is usually a combination of two words, that is theranostics and diagnostics. So when till now what we are focusing on the treatment of the disease separately and the diagnosis of a disease separately. But when the invention of theranostics happens, now we can simultaneously detect or diagnose as well as we can treat the disease. So in today's section, I have a few points that I have to cover, and the contents are introduction, theranostics biomaterial, imaging bio modalities, carriers of biomaterials, application, conclusions, and references. So when we talk about the definition, you can see in the definition there is a image and the therapy. So it is a novel process when we talk about the theranostic. So it is an interrelationship of a diagnostic imaging with the treatment therapy. So we will concentrate on some diseases. Right, we will start from the cancer disease. When if you look into the cancer cells, so there is a certain receptors on the cancer cells over which the drugs are usually attached. So it is usually you can assume that a cancer is a home, and if you want to steal the home, you have to open the door. And if you open the door, if you have a team of robbers, you can enter into it easily, and you can easily destroy that particular assets. So here you can see that we can attach a diagnostic agent as well as the therapeutic agent simultaneously. And if you look into the therapeutic circuit diagram, it will look like this. There is a radioactive compound, as we have studied in the radio-labeled compound in the B pharmacy, and there is a linker, as well as there is a targeting molecule which will act as a key of opening the cancer cell gateway, and the target protein, which is the receptor over the target cancer cells. So we will have a small video representation so that we can easily understand the concept of theranostic. So if we see this, you can see we have an example of prostate cancer. So let's go. Yes. Yes, so here is the example of a prostate cancer. So when you look into the prostate cancer, you will find that when the advanced stage of prostate cancer occurs, so there is a widespread spread of the cancer cell throughout the body. Okay, so it is very difficult to wash out the body with the cancer cells. So what usually happens in the prostate cancer, there is an receptors over the cancer cell, which is known as the prostate membrane antigen, specific membrane antigen. And what we are doing, we are attaching the radionucleotide with a theranostic agent. And this you can see in the image that there is a PSMA receptor over the prostate cancer, and this is the advanced stage of a cancer of a prostate where the cancer is spread all over the body. And if you watch it, so what we are doing, we are attaching a radionucleotide over the MSA targeting molecules. So usually what happens, this targeting molecule, when we inject into the body of a human being, it will search for the PSMA receptor and it will bind there. And when you move for a PET scan or a CT scan, you will have the diagnosis image that where are the cells located. But usually what happens when we are using the theranostic approach, we are attaching the drug molecule with a radionucleotide molecule also. So what usually happens, the one radionucleotide is diagnosing the image, and the second nucleotide, when you inject such type of molecule inside the body, it will going to destroy the cancer cells. So what is the benefit of using this? The benefit is it will not destroy the normal cells, it will only destroy the cancer cells. So here, when we use the radioactive materials for the destruction of the cancer cell, you can see that when the radioactive decay happens, so there is a liberation of a different particles, that is a alpha particle, beta particles, and a gamma particles. And we commonly call them as an alpha rays, beta rays, and gamma rays. So when we look into the penetration power of these rays, we found that the gamma rays are the one which easily penetrate through different layers. Like if we consider the layers as a paper, aluminum foil, lead, or water, you can see the neutron ion is the rays which are usually and deeply penetrated, and we are mostly using the gamma rays and X-rays for it. So what is the purpose of this? So here we need a biomaterial. So why we are needing the biomaterial is that we have to transfer our drug molecule so that it can be used as a key to facilitate the radionucleotide that can be able to get inside the cancer cell. So you can see there are a lot of biomaterials are there, and this biomaterial can be used for various engineering purposes like tissue engineering, drug delivery, biology, and chemistry. And this biomaterials can act as a carrier for the delivery of a diagnostic agent. So when we talk about the therapeutic biomaterials, these therapeutic nanomaterials, or we can call as a biomaterial, especially we are considering the nanoparticles here when we talk about the theranostic approach. So these are the multifunctional nanosystems, and they can be used for more specific and personalized disease management. So nowadays, this is an era of a personalized medicine. So this branch of pharmacy, the theranostic is growing fastly. So when you look into the nanomaterial which are commonly used in the theranostics, these are, you can see there are the magnetic nanoparticles, quantum dot, polymeric nanoparticles, carbon-based materials, even the radioisotopes can be used in the field of theranostics. The dual-coated radioisotopes can be used for the treatment of cancer cells. Then you can use gold nanoparticles and silver nanoparticles also. If you see the general diagram that how the things are going on, you can see there are the therapeutic agent, which can be anything. It can be a small molecule, or you can call as a chemical drug. It can be a DNA, RNA gene, or a protein, or a photosensitizer. So nowadays, what we are doing, we are combining the things. So what we do, we are adding the imaging agent also in the nanoparticles. And to facilitate the transfer of the this theranotic agent into the cell, we are using the stealth molecules. So this stealth molecules helps the drug to, or helps that material to easily penetrate inside the drug, the cancer cells. So that there are two things when we talk about the theranostics. So the theranostics will work along with the imaging facilities. So when we talk about the imaging facilities, what we are using, so as we have concentrated our talk in the cancer, you can see there are certain optical imaging platform, there are ultrasounds, there are magnetic resonance imaging, photoacoustic tomography, that is known as the PET imaging. There is a PET imaging also, single photo emission computed studies, and the SPECT, that is known as the SBCET. And when we talk about the benefits of using the nanocarriers, which is generally due to greater effectiveness, we are using this nanomaterials to work as a theranostic. We are using, we can use which we can use for site-specific targeting, including solid tumors from leaky vectors. When we look into the market of this theranostic material, you can see, according to the CAGR's report, till 2023, the market will be 3.9 billions, because the cancer patients are increasing day by day because of the disturbance in the environmental domain. So these are the references which I have covered or used for preparing the slides, and thank you very much for your patience. Yes, any question,
Muhammad
please?
Yasir.
Yes, sir. Good evening, sir.
Good evening. It was a nice presentation. You detailed very well about this theranostic. But it was not an experimental work, I believe. We talk about only the theory of the theranostics.
Yes, sir. It is not an experiment work. It is a theory part.
But I think you are pursuing your PhD.
Yes, sir. But
why you have not presented your experimental work?
because, sir, it is not up to the mark, so I can present in front of you.
But I think we have to consider it if it is not experimental. But it is not experimental. Okay, no issue. I'm not discouraging. I'm
Yes,
just
sir.
saying that
I can explain some points.
Yeah.
Actually, when you have seen this slide, the circuit diagram, just give me 2 minutes. So when you look into this circuit diagram, sir, there is a target molecule which is opening the gate for theranostic agent so that it can enter the cancer cell. So here, what we are trying that recently the people have reported that we can use natural product. Okay, so the benefit of using the natural product for linking in the level, and we will be we will be hoping that we will get a good experimental results.
Thank you. We got it. But are you working in this area only in your PhD?
Yes, sir. In the area of nanoparticles.
So do you have idea about the brachytherapy?
Yes, sir.
Brachytherapy.
No, sir. Sorry.
Because that is also related to this radiation and what radioisotopes you are talking about, that they are used for identification of tumor specific marker proteins, and then
Okay.
to target the tumor specific cells only, a tumor cell only, so that the innocent cell could not be, encountered.
Yes.
So, Okay, thank you very much.
Okay, thank you, sir.
Good evening, everyone. I am Purnima Tripathi, and I am here to present. And the topic of my presentation is design and development of cyclosporin niosomes for the management of psoriasis via dermal delivery. I have pursued my PhD under guidance of Dr. Piyush Bharadwaj, and it is a part of my PhD work. The contents of my today's presentations are as follows: introduction, statement of problem, aim and objective, rationale of the work, work done, results, and conclusion. So, starting with the introduction, niosomes are the non-ionic surfactant vesicular drug delivery system. Since these are made up of non-ionic surfactants, these are non-toxic, biodegradable, as well as biocompatible. The most important thing about niosomes is that these can be used to entrap both hydrophilic as well as hydrophobic drugs. Next is the disease part. The disease I have worked on is psoriasis. It is an inflammatory condition of the skin, and it is characterized by the presence of plaques of thickened and erythematous skin with white scales due to the hyperproliferation of keratinocytes. For severe treatment, it is necessary to give the classic immunosuppressive agents like cyclosporin, methotrexate, etc. The statement problem is we have selected cyclosporin here. Cyclosporin is a very hydrophobic drug with very high molecular weight. Since the main problem with us was delivery of cyclosporin. When we deliver cyclosporin in psoriasis, it creates, when it is administered systematically, it creates number of serious problems like carcinogenicity, cardiovascular side effects, nephrotoxicity, etc. So the alternative route to use cyclosporin is dermal delivery. But the problem in dermal delivery is that cyclosporin is a very high molecular weight compound and we cannot deliver it directly through the skin, especially in case of psoriasis when the skin is thickened, and so the delivery is more difficult. So the novelty of my work is currently cyclosporin is available only in the solid dosage form as a capsules, and the oral route showed severe side effects, as I have just told, the cardiovascular side effects, nephrotoxicity, etc. And to date, there is no topical formulation available in the market. Aim and objective of my study. The main objective of my study is to prepare cyclosporin-loaded niosomes for the effective permeation and retention in the skin tissues for the management of psoriasis. It includes pre-formulation studies, formulation of preliminary batches of niosomes, incorporation of cyclosporin into the niosomes, optimization of the prepared niosomes using box Benken design, characterization of the optimized niosomes, then in vivo studies, and finally, evaluating the efficacy using histopathological studies. Rationale of the study: since it is a high molecular weight and hydrophobic compound, it could not be easily delivered by dermal route. It can poorly penetrate into the skin, and in the trials, it is reported that 80 to 90% of patients who received cyclosporin at a dose of 2.5 to 5 mg per kg body weight for 12 to 16 weeks had rapid improvement. Though it shows rapid improvement when administered systematically, but when administered orally, it shows many side effects. And especially when it is used for a longer duration, it has proven to be carcinogenic. The SPAN 60 is the non-ionic surfactant that I have utilized for the preparation of niosomes, and cholesterol is used as a stabilizer, just as cholesterol stabilizes the cell membrane in our body. Here also cholesterol stabilizes the membrane of niosomes. So the proposed work is pre-formulation studies, optimization of the formulation, and characterization.
Pre-formulation studies include organoleptic properties like color and odor. Melting point was determined, and the reported value was similar to the observed value was similar to the reported value. Next, FTIR studies were performed to characterize the spectra and the purity of the compound, and the reference peaks and the observed peaks were almost similar. Next is the calibration curve of cyclosporine was prepared using acetonitrile and water with a system of 70:30 ratio at 210 nanometer in a UV detector, and the flow rate was 1 ml per minute. Retention time I obtained was 4.6 minutes, and R-square value was 0.9984. Niosomes, next is the method or preparation of niosomes. Niosomes were prepared using thin film hydration method. Here we mixed drug, surfactant, and cholesterol in 1:1 ratio of chloroform and methanol in a RBF. And then this mixture was fixed in a rotary evaporator. Using the rotary evaporator, the solvent was removed. A thin layer of the film was formed on the RBF, and this film was hydrated using phosphate buffer pH 7.4 at 60 degrees Celsius for 30 minutes with gentle spinning. Later on, it was probe sonicated for 5 minutes to homogenate it and obtain a good size range. The composition of the preliminary batches were span 60 was used in 100, 50, and 150. Different amount cholesterol 100, 150, and 50. Hydration time was varied from 10, 20 to 30 minutes. And the results we used to interpret the preliminary data were the vesicle size and percent entrapment efficiency of cyclosporine. Later on, the formulation was developed using the experimental design. The minimum and maximum values were put into the Volkswagen design, and the next the formulations were developed according to the design. Total 15 formulations were developed using 3 independent variables. The 3 independent variables were ratio of cholesterol is to surfactant. The low level was 1:2, medium was 1:1, and the highest was 2:1. Hydration time was the second independent variable. It was capped at 30, 45, and 60 minutes, and amount of drug was the third independent variable, which was taken as 10 mg, 20 mg, and 30 mg. The 3 dependent variables I selected were vesicle size, polydispersity index, and percent entrapment efficiency. The table shows the data of niosomal suspension that I have prepared. So these are the different, the first X1 shows the independent variable. Ratio of cholesterol is to span 60. In the varying ratios, X2 is the minutes, the hydration time in minutes. X3 shows the amount of drug in mgs. And the later 3 column shows the dependent variables or the responses I obtained for size, percent entrapment efficiency, and PDI. This table shows the summary of results of regression analysis for responses fitting to quadratic model. For all the 3 responses, quadratic model was the fitting model. Since the value of p-value was less than 0.005, the p-value for y1 was less than 0.001. For y2, it was equal to 0.0004, and for y3, it was less than 0.0001. Summary of regression equation for responses is as follows. For the first response, size y1, mathematical equation clearly shows that the first factor, cholesterol is to span 60 ratio, the second factor, and the third factor all had a significant effect on the first response. As the from the equation, it is very clear that as the concentration or the ratio of cholesterol increases, the size decreases. The reverse is also true. We can say that as the ratio amount of span 60 increases, size decreases. This is because of the long chain of span 60. Let us move to the next response, y2. Here also the equation is very clear. It shows that all the 3 responses all the 3 factors had given a negative response on the y2. That is the PDI. And the third is percent entrapment efficiency. Here we can see that the first factor X1 has given a negative response, whereas factor X2 and X3 has given a positive response in the percentage entrapment efficiency. First response, as we see, as the amount of cholesterol increases, percent entrapment efficiency of cyclosporin decreases. This is because both the cyclosporin and cholesterol are lipophilic in nature, and they compete for entrapment inside the vesicles. Whereas the factor X2 and X3 both increases the entrapment efficiency when it increases, just because the factor X2 is the hydration time, and when the hydration time increases, the cyclosporin gets more time for the entrapment. So it gets the more amount of cyclosporin is entrapped inside the vesicles. And as the amount of drug increases, it also increases the percent entrapment efficiency. These are the 3D response graph for vesicle size, as I have explained just using the equations. This is the first one for vesicle size, first response Y1. This is the second response PDI, 3D response curve, and this is the response curve for percent entrapment efficiency. The optimized batch formula was lipid is to surfactant ratio 0.456. Drug was used in a quantity of 30 mg. Hydration time was kept at 30 minutes. Results, predicted values and obtained value of the responses were as vesicle size was in predicted value was 187.8 nanometer, whereas the obtained value was 180.5 nanometer. PDI was 0.141 predicted value, and the obtained value was 0.156, and percent entrapment efficiency predicted value was 95.36, and the obtained value was 93.2. So as we can see, there is not much difference between the predicted value and the obtained value. Next is the drug permeation profile. The two graphs that we can see here, the blue line shows the permeation of cyclosporin from the dispersion, whereas the red curve shows the permeation of cyclosporin from the niosomes. It is clearly evident from the graph that the permeation of niosome increases when it is given using permeation of cyclosporin increases when it is given in the niosomes, as compared to when it was simply given as a simple dispersion of cyclosporin. Though if we see the complete permeation, it is very less. Only maximum 13% of drug could permeate the skin through the niosomes. Next is skin deposition in dermal kinetics study by tape stripping method. So first we performed the permeation studies, and then afterward the permeation is both the permeation as well as skin deposition studies were performed using excised goat skin. So first we performed the permeation studies, later on we used the skin that we utilized for the permeation study, and then we used the tape stripping method to see how much amount of drug is deposited in the stratum corneum. After stripping the skin, later on the remaining amount that we were having with us of the skin, we cut it into the small pieces and put it into the methanol and stirred it for 30 minutes, so that the drug present in the viable epidermis could extract out, and then the content of drug present in that viable epidermis was determined. So from this graph, it is very clear that the percent drug deposited in the SC for cyclosporin niosomes was 3.1%, and for cyclosporin suspension was 3.1%, and for cyclosporin niosomes it was 15%. Whereas the amount of drug deposited in viable epidermis from the cyclosporin suspension was 7%, and cyclosporin niosomes was 35.8%. Whereas the amount of drug left on the skin from cyclosporin suspension was 83%, and cyclosporin niosomes was only 35%. So we can say the maximum amount drug that we have given was deposited in the viable epidermis, which is actually the site of action of psoriasis, and minimum amount of was permeated across the skin, which was our main focus, since we do not want any undesirable effect of cyclosporin in our body. That's why we have shifted our route of administration from oral to the dermal delivery. So since the amount of drug permeated using niosomes was very less, only 13% of drug could permeate the skin, and maximum amount of drug was deposited in the viable epidermis, this delivery system proves to be effective. Next, we performed in vivo studies in the using anti-psoriatic plaque model. Here, we used Swiss albino mice and the psoriasis was induced using imiquimod. 62.5 mg per kg body weight drug was used per day to induce the psoriasis for 7 days. We took total 4 groups of cis albino mice. First group was the control group. To that group, we neither induced psoriasis nor we given any treatment. To the next group, we induced psoriasis, but treatment was not given. And to the last 2 groups, to the third group, we have both given, we induced psoriasis as well as we given treatment. To the third group, the treatment was simple of cyclosporin suspension. And to the fourth group, treatment included niosomes of cyclosporin. So for the first 7 days, we induced psoriasis. Conclude कर रही हूँ मैं. This slide shows the histopathological examination. The first slide shows the normal control group. Next is disease control, where it is very clear that all layers of skin are disrupted. Whereas in case of third and fourth, some normalizing was started. In the third group, skin was somehow normal, but in the fourth group, we can clearly say that it was regaining almost to the normal shape. Thank you. Any questions?
Any question, please? Okay, Priyanka, it was a nice presentation.
Thank you, sir.
You detailed the things very well.
Thank you.
My question is, like the cyclosporin, for what purpose it is used? Means what is the mechanism of action of the disease?
Sir,
this
actually,
drug?
sir, basically it is a immunosuppressive agent.
Uh-huh.
It is used for transplant. जैसे for example kidney transplant होता है, तो उसमें हम देते हैं to suppress the immunity of the body.
But how it is suppressing the immunity?
So just 1 minute. So it suppresses the T lymphocytes.
How?
So it is an calcineurin inhibitor, sir.
Okay, so what the calcineurin does?
So calcineurin activates T lymphocytes. So since it inhibits calcineurin, T lymphocytes are suppressed.
So this drug must be used in some other autoimmune diseases.
It could be used, sir, but it is mostly used for transplant or organ transplant.
No. I mean,
Or in case of psoriasis, psoriasis is an autoimmune disorder. So to suppress the immunity, it is used here.
transplant is a one thing, means where we need to suppress the immune system.
Right,
I
sir.
mean, voluntarily.
Okay, sir.
But sometime when some autoimmune diseases are there,
Right, sir.
I mean, for some other autoimmune diseases also cyclosporin may be used, na?
Right, sir. May be used, sir.
Okay. So you found that this niosome patches, they are working well because they are releasing the drug properly. So you have checked the toxicity also? I mean, it is when you are applying to the niosome, it is not inducing any kind of toxicity?
No, sir, there was no toxicity. Like skin toxicity, I can say there was no toxicity. We have not performed any other special toxicity test.
Okay. But I think you should perform and
Yes, sir.
see that secondary, tertiary lymphoid organ toxicity.
Right, sir.
Because when availability is increasing, what kind of impact it is having on the lymphocytes?
Definitely, we should, sir. Actually, the main thing is the marketed formulation is capsules, which is given orally, sir. So its oral administration causes severe issues like nephrotoxicity and cardiovascular side effects. And since we are giving it dermally, only 13% of drug was permeated. So as compared to the other marketed formulations,
But dermally means only it will act locally, na?
so we want it to act locally.
In case of psoriasis, na?
Right, sir. In case
I'm
of
talking
psoriasis.
about some other autoimmune.
So for that, we have to.
Okay, thank you, ma'am.
Thank you, sir.
हेलो मैम एक क्वेश्चन पूछना था पूछ लूँ क्या
Yes, sir. Thank you.
As you have mentioned, this is niosomal suspension.
Ji, sir.
I would like to ask which type of suspending agent you have added for this formulation.
Sir, I've simply dispersed it in the system. It was not soluble actually.
Actually, you have bud suspension, niosomal suspension.
Yes,
Okay.
yes, sir.
So I am just asking, if there is suspension, niosome is suspended.
So niosomes were suspended, so in it's actually the volume of, let me explain you, sir. Since the volume of total suspension was very less, so whenever we utilized it, we dispersed it and used it immediately.
No. My question is just which type of suspending agent you have used it? Because you are using for the transdermally, I
Yes,
think dermal for the dermal system.
Dermal, sir, yes, sir.
Okay.
Yes, sir. So we have not used any suspending agent.
Just a minute, sir, because I was supervisor for this work. I would like to clear here that there is no need of any suspending agent because we are not here to prepare a stable suspension of niosome, sir. So I would like to say, sir, that there is no need of any suspending agent in this preparation, sir.
No. My question is because as I know, suspension, if there is a suspension, then there should be some suspending agent.
Sir, it is not necessary. Suppose we are having some particles, suspending. Sir, let me do clear, sir. Sir, for stable formulation.
For curiosity purpose, I asked which type of suspending agent you have used. That's my question. You have not used, that's okay.
I have not used.
पक्का?
हाँ जी, sir.
कोई तो यूज किया होगा.
से पूछिए पक्का है सर, कंफर्म. हमारे सुपरवाइजर. Thank you.
Okay. Thank you, sir.
Thank you, Purnima, for nice presentation. Our next participant is Priyanka Rathore. Is Priyanka here? Okay. Priyanka Rathore from pursuing her PhD from Institute of Pharmacy, Bundelkhand University, Jhansi. So please come on stage, Priyanka.
Good evening, everyone. Good evening to all the respected dignitaries. I am Priyanka Rathore, pursuing PhD under the guidance of Dr. Alok Mahur, sir, from the Institute of Pharmacy, Bundelkhand University, Jhansi. Today, my topic is I'm going to give a presentation on herbal versus synthetic pharmaceuticals for the better management of attention deficit disorder. As this is the theme of the conference, also, I have chosen this topic because in the modern era of technology and environmental changes, many behavioral, neurobehavioral changes are arising, especially in the children. And this is a neurobehavioral disorder characterized by the persistent pattern of inattention, hyperactive, and impulsive behavior. This is a disorder which is also called attention deficit hyperactivity disorder, ADHD, and it is mostly found in the children and can persist into the adulthood also. The basic risk factor is not clear, but certain etiology which I have reviewed from the many review articles. These are the general factors, neurotransmitters, environmental factors, prenatal factors, psychosocial factors. From the genital factors, we can say if in a family there is a history of some autism or some mental disorder, then there can be a chance that the children can have this ADHD disorder. Basically, neurotransmitters are responsible for this disorder. If there is a low level of neurotransmitter, that is the dopamine and norepinephrine, then there is certain changes in the brain, and the person, children, adult can also be the case of ADHD. Environmental factors, because certain environmental changes, some technology, children are nowadays basically busy in keeping the mobiles. They are not having that playing grounds. They are not busy in spending their time in studies as they previously used to do. So these are certain environmental factors also which cause the children some distractibility. Perinatal factors we can say, because during the pregnancy, if the lady is smoking or they are having the alcoholism cases, then the perinatal factors definitely cause the ADHD in the child. Social factors, like nowadays, the family is basically a nuclear family. We are not, children are not growing in the joint family. So some psychosocial factors, their mother and father both are working. So in nowadays, these factors are also causing these changes in the brain anatomy of the child, and they are more and more distracted. They can't focus, they can't concentrate on their work, and these changes that we can see during in their school, in their studies, and in their behavior. Primary school children in India, basically around 11.32% are affected by this disease. Most prevalent this disease in males, like boys. Globally, this disorder affects 2 to 7% of the children. And India, it is higher than the global estimate of 11.32%. The highest prevalence is found in 9 years of age. And in India, this defect, this disorder in children can lead to the adolescence also. Basically, 2 to 8% of the population in adults is affected by this disease. 9% of the school children are also affected. The prevalence rate on several factors, depending on several factors like gender, family, even country, and in the worldwide ADHD children chart, you can see. The signs and symptoms of this disorder leads to inattentiveness. The child can be distracted. They can't focus very well. They are very hyperactive. They are impulsive. They behave and they work without thinking about the results. Some antisocial depression changes are also seen in the child, in the adult. Some focal and associated control problems are also there. Basically, the diagnostic tool is not there to identify or diagnose this disease like the biomarkers, but basically we use the machine learning and deep learning techniques. Machine learning involves some MRI changes in the brain of the child. Deep learning depends on generally depends on the psychological questionnaires, some psychiatric questionnaires, counseling of the child. With these tools, we can diagnose this disease. The brain function and structure, how is it responsible for affecting this disease? As you can see, I have given here two diagrams of the brain. First one is the normal structure of brain, where is the frontal lobe, parietal lobe, occipital lobe, temporal lobe. As we can see, the frontal lobe, these are responsible for the emotions and neurons which are depending on the cognition and decision-making power of the child. When there is a decrease in the level of neurotransmitters like norepinephrine and dopamine, this basically changes the mass of the frontal lobe, and there is the reduction activation of prefrontal and striatal region, and these leads to the changes that the child cannot be able to make some decisions, that they cannot focus, they are distracted. For basically the treatment and management, how can we treat the disease, how can we stop this disorder in the child? It depends on the behavior therapy, special education, social skill training, effective parenting. With these tools, we can stop or we can decrease the effect of this disorder in the child. Some medication, synthetic pharmaceutical drugs are also available. Basically, the stimulant which decrease the hyperactivity and impulsiveness and increase the attention and boost the neuron growth in the brain of the child. The drugs which are responsible, which are stimulants, which are used under the trade name Ritalin, that is methylphenidate. It is available in immediate and sustained release doses form, but they are having the low bioavailability. Similarly, the other drugs I have also mentioned here, that is the dexmethylphenidate, amphetamine, dextroamphetamine, the combined amphetamines like less dexamphetamine. Similarly, some non-stimulant drugs are also used, that is atomoxetine, clonidine, guanifacin, desipramine, which are generally the sympathomimetic drugs, and they oppose the smooth muscles contractions. Like this, the other drugs also, bupropion and other drugs, which some supplements are also used, which can enhance with the stimulants in the therapy of this disorder. But now, my main concern about the herbal treatment. As in the morning also, respected Mr. Anurag Sharma sir also said that Ayurveda is a field, herbal medicines, they can also make some changes, and they are also having the potential to treat so many disorders where as in the child, these synthetic pharmaceuticals, they can cause the adverse reactions. And our herbal medicines, they are having the potential, they are very beneficial to treat such type of disorders. Like tulsi, lion's mane, it is a type of mushroom. Their main constituent is Herinium and Erinaceous. They are the neuron boosters like valvetamine. They contain the L-dopa, which generally make up the amount of the dopamine in the brain. Similarly, the pycnogenol, which is a flavonoid antioxidant extracted from pine bark, some herbal teas, spearmint, lemongrass, and like this, ginkgo biloba. They are also help in improving the memory and increasing the mental sharpness. Similarly, like Brahmi, it is also used in the China and as a water hyssop. The herb is made from leaves. It is also using as a brain tonic from a century. Green oats, these are also the having calm nerves and treat the stress and anxiety. Ginseng, pine bark, these are also some herbal medicines. So there is an advantage of herbal medicines over the synthetic pharmaceuticals. Some pharmaceuticals which are not tolerated by the children, herbals can be easily tolerated by the child. Natural and green therapy will be beneficial for the child. Antioxidant properties present in the herbal medicines, they are also effective in reducing the toxicity of synthetic pharmaceuticals. So at last, I would like to conclude that combining the herbal medicine plant with nutritional supplement will be the promising treatment for against the attention deficit hyperactive disorder. Further development of herbal plants and different disease formulation improve the effectiveness, and proper use of these herbal plants during the pregnancy also improve the prenatal factors. So with these words, I would like to, these are the references I have gone through, and thank you so much.
Thank you, Priyanka. Please. Priyanka, what was your study design?
No, sir. This is the review. I have gone through some articles and I have just make the presentation. This is a review work. Whatever the things I have seen and I have inculcate in my presentation.
You are pursuing your PhD on the same topic?
No, sir. This is not my PhD topic.
You have interest in this area?
Sir, actually, this topic is in the theme list of the conference. So I have used this topic, and my PhD work is not in that list. So I have not used that one.
Again, that's a problem is same that there is no experimental data presented here in this presentation. And the second thing I could not understand that how the machine learning can be used for diagnosis of attention deficit disorder. Please,
Actually, sir, actually, I have seen some papers, some articles, research articles. That I have seen some researchers have work on the brain of animals, and they just give the drug, and they find out they gone through the MRI, and in children, and so they inculcate that in vivo studies in the human volunteers also. So we can use that MRI data as an input and find out the changes in the brain.
Where is
So
the machine learning in this?
that is machine learning.
Where is the machine learning in this?
So machine learning, because we are using some scanning techniques, na, like computer scanning techniques
Scanning
also.
techniques we are using since decades for understanding the damages or any kind of things
Yes, sir.
on the brain or any kind of cells. That is not a machine learning.
Sir, actually what happened, they have given in that review, their research articles also, that they are diagnosing using the machine learning. So I have used that thing. And from that work, I use these, that whatever the inputs we are digitally processing, so I use that word, the machine learning.
But I think one has to be understood ना, means machine learning है क्या और वो कैसे आपको attention deficit disorder दूर करने में मदद कर सकती है।
Yes.
एक बड़ा अच्छा आपने बताया, इसलिए एक बड़ा ऐसी चीज है जो बड़ा attractive type था, जिसको जानना जरूरी है कि machine learning से कैसे attention deficit disorder दूर किया जा सकता है। आप प्लीज थोड़ा सा और समझें और हमें भी समझाएं।
सर मैंने पहले भी कहा था कि एज अ बेसिक डायग्नोस्टिक टूल इज नॉट अवेलेबल फॉर आइडेंटिफाइंग दिस डिजीज, सो दे यूज सम बिकॉज दिस इज द डिसऑर्डर विच ट्रीट्स which deals with the brain functioning and brain structure, so they continuously use that MRI technique.
Suppose you are doing a HRCT scan for understanding the damages caused by the coronavirus in your lung cells,
Yes.
is it the machine learning?
Sir, if the diagnostic tool we are using the CT scanning, so we can.
It is not a machine learning.
So maybe my perception is different from that, but whatever.
But you please take.
I have gone through that
Okay, ma'am.
article, so
Thank you.
I have mentioned here. Thank you, sir.
Ma'am, I have two questions. Like one, sir, I've already asked about machine learning and how artificial intelligence. How can you use an ADHD? So, sir has already asked that question.
Yes, sir.
So another question is dyslexia and dysgraphia related to ADHD?
What?
Dyslexia in kids and dyslexia
Dyslexia.
and dysgraphia. They are two common diseases. You know, it's not disease, it's a disorder.
Ma'am, but dyslexia will not come under the attention deficit disorder.
Okay.
Means if the attention deficient disorder, you can understand these are the hyperactivity when the child won't want to concentrate or focus on the things. They don't want the
Dyslexia
people
and
can see
dysgraphia are
them.
the same, ma'am.
Huh?
Dyslexia and dysgraphia also, children, they cannot focus, they cannot retain things.
Ma'am, it is a type of neuronal disorder, but this attention deficit disorder, we can say it is a neurobehavioral disorder.
Dyslexia and dysgraphia are not this behavior, you mean to say that?
Maybe ऐसे तो फिर
alright,
we
you
can
said
say
like-
the autism also
Alright.
is the journey, as I have as I already
Autism is a genetic, you know, there is a genetic mutation in autism that I can understand. But in dyslexia and dysgraphia, these are not genetic related disorders, right?
Ma'am, we cannot put dyslexia under the attention deficient disorder, but they both are the brain related disorders. That only I can say right now.
Alright. Okay, so you said these ADHD, they doesn't have any other symptoms. Other symptoms like other diseases, like cancers or whatever diseases we are dealing with, then how do you feel like you are going to find some Ayurvedic medicine for ADHD if ADHD is not a disease? You yourself said this.
Ma'am, herbal medicines, they are having.
If ADHD is not a disease, then how can you find a medicine for that?
ADHD is a disorder, na?
You said in your words like ADHD.
I'm not saying that it is a disease. I am saying that dyslexia and attention deficient disorder, these are separate things.
In your
Both
slide
comes,
number 3, you
both
said-
comes under the brain dysfunctioning, but we cannot say ADHD is the synonym of dyslexia. Yeah, dyslexia will come under the
In
ADHD.
slide 3, you said you have written that ADHD has biological some
Hyperkinetic
symptoms and all, but in this slide only you are saying like we are finding some.
No,
yeah,
I have not written their dyslexia. I have written that
I'm
it's
not
the
saying dyslexia. Leave that.
Okay.
I'm talking about ADHD now. So you have said like ADHD has some biological symptoms. They cannot, the kids cannot concentrate.
Okay,
and all.
sign and symptoms.
Yeah. So
Okay.
they have the symptoms, all right, and after that you are saying like this is a disease, but you are finding some medicine for, as if you are finding some medicine for a disease.
So sign and symptoms are also seen in the disorder, na? I am not going through you, but you are really about to ask.
Sorry, ma'am. Actually, I need to interrupt in between.
Yes, sir.
With your kind permission.
Yes, sir.
No issue. I think attention deficit is a kind of thing where, like she told about, somewhere it is observed that dopamine or some those kind of like serotonin, dopamine, those neurotransmitters which are, I mean, delighting our mood. And then when we are in delighted mood, when we are in pleasant mood, then obviously we can focus better on the things and that we can grab the attention very well. So sometimes what happens that some neurotransmitters which are, I mean, affecting on the limbic system, brain limbic system. If those neurotransmitters are decreasing due to some any reason, that may be social reason, that may be, I mean, environmental factors, that may be chemical factors, then such kind of problems may be generated. Means like attention deficit is not a kind of particular disease.
Yes, sir.
It is just a kind of observation sometime. So when it is getting in higher quantity, that one is not very well, I mean, able to pay the attention on even for a short while also, then it may be a kind of disorders. Otherwise, you cannot measure at what level you can say that we are having the attention deficit.
Okay, yes.
Again,
Yes.
so in that way, I think she's talking about that if we are using such kind of Ayurvedic medicines or herbal medicines or
Or in case of
general
dopamine, yes.
which can, I mean, affect on the limbic system, which can increase the serotonin and level of the dopamine, which are making our mood pleasant, then obviously it will have some good impact on our nervous understanding. Thank you, ma'am.
Yes, sir.
Thank you.
Thank you.
Thank you, sir.
For today's session, last but not the least, our next participant is Brajesh Mishra from IGNU. Brajesh, please come on stage, please. After this presentation, felicitation program will be held. All the chairperson are here, to whom we have to felicitate. Our faculty member will be felicitated. Will felicitate to our chairperson, like Rahul Deo is here, Dr. Pradeep Niranjan is here, Dr. Laukus Dwivedi is here.
Good evening to all. This is Brijesh Shivhare, and I am here to present my PhD, a part of my PhD work. And the topic entitled hepatoprotective and nephroprotective potential of sarcanic extract against lead acetate induced toxicity in rats. So the aim of from starting to the aim, to evaluate the hepatoprotective and nephroprotective potential of sarcanic extract against lead acetate induced liver and kidney damage in vivo study. The objectives are assessment of hepatoprotective and nephroprotective activity of sarcanic extract by in vivo study. Second is to check prophylactic activity, and third is therapeutic effect of the sarcanic extract. So the plant taken is Sarca indica, which is commonly known as Ashok plant, or you can say Sita Ashok. The ethanolic extract is used. A combination of plant parts is used for making the ethanolic extract, and this is of 80% ethanol. And the chemicals used for the leads of AR grade and the diagnostic kits for serum biochemical parameters analysis, and the star rats of weighing 140 to 180 gram of 6 to 8 weeks were used which kept in a optimum condition under the standard guidelines. having the same temperature. The temperature is 25 plus 1 is 2 degree with 12 hour of light and dark cycle, and animals were fed commercially with available pellet diet and water ad libitum. For this the for using for any study on rats, we have to approve by the ethical committee. So my study is done on the rats by the ethical approval by the Institute Animal Ethical Committee of Bundelkhand University. So the study of the study is divided on 3 groups. First is pilot study I have done. Then second is subchronic study, and the third one is for chronic study. In my pilot study, the rats were divided into 7 groups, n is equal to 5. In this group one is for control. Group 2 is for lead acetate with with a dose of 60 mg per kilo body weight, which was given post orally, as lead acetate is soluble in water. That's why I choose for orally. And the group 3 give the standard drug, that is NAC, N-acetyl cysteine, with a dose of 100 mg per kilo body weight. Now the next 4 to 7 groups are divided into on the basis of different concentration of plant extract, with a range of 500 to 2000 mg per kilo body weight. Next, after the last administration of the dose, after 24 hours, the rats will be sacrificed by the method of chloroform and cervical dislocation. After that, the blood was taken for retroinverter, and the serum biochemistry was done for liver injury and as well as for kidney also. The first slide was showing for liver disease or the liver injury. Next one is for next study after the dose optimization. Then we go for subchronic study for 3 weeks. In this, 4 groups are divided, in which first is control, second was is the toxicant, that is lead acetate, and third one is the toxicant plus standard drug, and fourth group is toxicant plus plant extract. After that, necroscopy and serum biochemistry and tissue analysis were also done. For checking the prophylactic activity. Now this is study 3 for chronic study for 5 weeks for checking the therapeutic effect of the optimum dose, which was obtained from the pilot study. And in this also the groups are divided into 4. Control, second is the toxicant, that is lead acetate. Third one is lead for first 2 weeks lead acetate, the toxicant will be given for group 3 and 4 animals. After 2 weeks, for continuous 3 weeks, the extract and the standard drug was given just to overcome the toxicity caused by the lead. After the study completed, the necropsy was done, and the blood and the serum biochemistry analysis and tissue analysis was done. So these are the results on which there are this is the combined oxidative hepatic marker shown, which cause the control. In this, the lead, the toxicant will cause more effect. Is it cause more toxicity, which leads to the increase in levels of oxidative enzyme markers in as AST, ALT, ALP, and LDH. As the different doses was given in this, the optimum dose which was finalized by my pilot study was of 500 mg per kilo body weight of the plant extract, which was given for further subchronic and chronic study. So in this, the level of the level which was the increased level of the oxidative enzyme markers was recovered and overcome by the plant against the toxicant. So this is the enzymatic antioxidant enzyme activity was also done. In this, the level of the antioxidant enzymes were decreased due to the toxicity of the lead, which was overcome and as similar or comparable to the control after the dose of the plant extract. And after this, the histology was also done. In this, the first A slide was for as a control, and the toxicity is caused by the in which the cytoplasmic vacuolation was formed due to the toxicity, which was overcome by the dose of these D, E, F, G are the different dose of my plant extract, in which the dose 500 mg per kg was used for the next against toxicant for prophylactic activity as well as the therapeutic activity. The same method is used for kidney also for nephroprotective effect. Earlier was the for hepatoprotective, and same procedure was done for kidney also for nephroprotective activity also. In this, after the sacrifice of the rats, the blood was taken for the serum biochemistry, and for the tissue antioxidant assays was done also. And the same chronic and same pilot after the dose fixation of pilot history, same the subchronic and chronic studies was done by the optimum dose, which was 500 mg per kilo body weight. And the results shown in this the findings were the as the level of the as the level of oxidative stress marker was increased by the toxicant given to rats, in which was overcome by the plant extract as compared to control as well as standard drug. This was also done. The antioxidant enzyme activity was also which is more similar to the standard drugs as we compare with the with different dose of the plant and with toxicant also. The both prophylactic activity and the therapeutic activity was shown the better results as compared to the higher dose. The histology of the kidney tissue histology was described as the evaluation in the first figure. This is as this is control. The second one is for toxicant given. The third is for the standard drug used in the market, and D, E, F, G are the different concentration of my plant extracts, which are given on the separate concentration, which gives more better results in the 500 mg per dose, 500 mg per kilo body weight. And the next two, the subchronic study describes the prophylactic activity, which overcomes the toxicity caused by the lead acetate in the rats. And in the chronic studies, the toxicity caused by the toxicity cause will overcome due to the decreased level of antioxidant markers, which was overcome by plant extract. So, the conclusion of the extract is the Sarca indica extract showed the recovery of 4 doses against hepatotoxicity and nephrotoxicity caused by the lead acetate, which is a highly toxic molecule, which cause many symptoms in human body. Out of 4 doses, 500 mg per dose was found to be more effective rather than that. Therefore, 500 dose was further evaluated for subchronic and chronic exposure for lead acetate. After 5-week study, therapy of Sarca indica extract showed pronounced recovery against lead-induced oxidative stress, cellular injury, and antioxidant defense systems. Microscopic studies indicated the extract has improved the structural integrity against lead and liver and kidney structure, as which was done by the toxicity caused in the rats. Next, the reference, I'll go through this. Thank you.
Thank you, Mr. Brajesh. It was a nice presentation. You were a bit nervous during the presentation, it was seems, but no issue. It happens. Okay. Very simple question I just want to ask. Means you took 4 doses, na?
Yes, sir.
Please go on your conclusion slide. You took 4 doses, 500, 1000, 1500, and 2000.
Yes, sir.
And you are writing that extract showed recovery in all 4 doses. Recovery of what?
Recovery of cellular disruptions. The
Cellular disruption was recovered by under all 4 doses.
all 4 doses, the extract is not much more harmful for the tissues, but the 500 mg
Question is very simple. Means you induced the hepatotoxicity and nephrotoxicity.
Yes, sir.
And through lead acetate.
Yes.
Then you tested the drug in four doses, 500, 1000, 1500, and 2000.
Yes, sir.
And in all these 4 doses, the tissues were recovered.
Yes, sir.
Then why you choose only 500 for further study?
So 500 is much more effective that, as compared to as the higher doses, it will cause some more cellular damage, because
You were saying no. Please don't mix it. Please make it clear. Earlier you told that the tissues were recovered in all 4 doses.
Yes, sir.
Then why you opted only 500 kg per 500 mg per kg dose? Why you have not opted 1000 or 1500 or 2000 for further study? First thing. Second thing, what kind of toxicity was caused by the lead acetate in the kidneys? I mean, what kind of toxicity? Where the damage was observed? Like in liver, you showed like the enzyme level was rising. How you assess the toxicity establishment in the kidney after giving the lead acetate dose?
Sir, sir, as described in this, the spacing and the necrotic cells and the solid structure, the Bowman's capsule is the tissues ruptured by the toxicant, which makes spacing in the kidney tissue. As compared to control, there is no much more degeneration of cells.
You mean to say the space is because of the degeneration of cells?
Yes, sir. As the
Degeneration of cells of which part of the kidney?
It will harm the metabolic pathways of the kidneys.
नहीं यार ऐसा नहीं. Metabolic pathway, कौन सा metabolic pathway?
Sir, like in जैसे glomerulus जो bowel है.
Glomerulus is different thing. Bowman capsule is different thing. Bowman capsule is the part of nephron. Glomerulus is the part of blood vessels. Damage कहाँ हुआ?
Sir, I am checking the whole organ. I am not using a particular part.
तो किस
That's why I am checking the whole histology for that.
अच्छा, you took whole kidney and then you checked it on the damage in the tissues of whole
Yes,
kidney.
whole kidney.
अच्छा, स्पेस बढ़ गया तो इससे क्या होगा? इससे क्या नुकसान होगा अगर स्पेस बढ़ गया तो?
जो इसका
ये गैप जंक्शंस बढ़ गए. गैप जंक्शंस बढ़ गए हैं या सेल्स जो है डिस्ट्रॉय हो गई हैं?
सेल्स डिस्ट्रॉय हुए इसलिए गैप जंक्शंस बढ़ गए.
चलिए ठीक है. ओके, थैंक यू.
Thank you, sir.
Thank you, Rajesh. Now this is the time to felicitate our chairperson of different session. I request all the chairpersons, Dr. Rahul Dev Yadav, Dr. Pradeep Kumar Niranjan sir, Niranjan, Dr. Dharmendra ji, Dr. Laukush Dwivedi ji sir, please all chairperson, please come on stage. And I also request to our faculty members, those who are present here, please come on stage to felicitate our chairpersons. So thank you very much, all the chairperson and faculty members, organizing teams, dear students. We will resume here tomorrow at 9 o'clock. The breakfast will be started at 8 to 9 and 9 to 10 poster presentation in the department, and 10 onwards the expert lecture session will be here. So be present at sharp 10 o'clock over here. And as I told you that from 8 to 9 is a breakfast tomorrow morning. So we will resume at 10 o'clock here tomorrow. Thank you.
You can play the music.
With our animation videos, you can start visualizing the data structures and algorithm concepts. Build the algorithm and learn how.
Or into secure placement of the mask. Okay.